on October 7th, 2021. I would like to introduce and acknowledge the excellent contributions of the staff in my division and the great team that I have in preparing for this meeting. Ms. Kathleen Hayes is my co-DFO, providing excellent support in all aspects of preparing for and conducting this meeting. Other staff who have been contributing significantly are Ms. Monique Hill, Ms. Karen Thomas, and Ms. Christina Wood, who also provide excellent administrative support. Would also like to express our sincere appreciation to Mike Kaczynski in facilitating today's meeting. Also, our kudos to many FDA staff working hard behind the scenes, trying to ensure that today's virtual meeting will also be a successful one like all the previous VRTAC meetings on the COVID topics. Please direct any press or media questions for today's meeting to the FDA Office of Media Affairs at FDAOMA, one word, at fda.hhs.gov. The transcriptions for today's meeting is Ms. Linda Giles and Erica Demham. We will begin today's meeting by taking a formal roll call for the committee members and temporary members. When it is your turn, please turn on your video camera, unmute your phone, and then state your first and last name. And then when finished, you can turn off your camera so we can proceed to the next person. Please see the member access slides in which we will begin with the chair. Dr. Arnold Monto, can we please start with you? Thank you.
Yes, thank you, Prabha. I'm Arnold Monto. I am Professor of Epidemiology and Public Health at the University of Michigan School of Public Health. And I've had a long experience in vaccines, respiratory disease prevention at the University of Michigan. Back to you, Prabha.
Thank you. Dr. Cohn?
Good morning, everyone. I'm Dr. Amanda Cohn. I'm a pediatrician at the Centers for Disease Control and Prevention with expertise in vaccine preventable diseases and vaccine policy.
Thank you. Next, Dr. Chatterjee.
Good morning, everyone. My name is Archana Chatterjee. I am a pediatric infectious diseases specialist. I'm also the Dean of Chicago Medical School and Vice President for Medical Affairs at Rosalind Franklin University. My area of expertise is in vaccines.
Thank you so much. Next, Dr. Meissner. Cody, we can't hear you.
There you go, Cody.
Thank you, Prabha.
Thank you,
Mike. My name is Cody Meissner. I'm a professor of pediatric infectious disease at Tufts Children's Hospital in Boston.
Thank you. Next slide, please. Dr. Gans.
Good morning, everybody. I'm Dr. Haley Gans, pediatric infectious disease at Stanford University, and my area of expertise response of vaccines, both children and adults with normal immune.
Thank you, Dr. Gans. Dr. Kurila next.
Good morning. Michael Kurila. I'm the Director of the Division of Clinical Innovation at the National Center for Advancing Translational Sciences within the National Institutes of Health. I'm a pathologist by training. My expertise is in infectious diseases and vaccine development.
Thank you, Dr. Kurila. Next, Dr. Offit, Paul Offit.
Yeah, hi, I'm Paul Offit. I am a professor of pediatrics in the Division of Infectious Diseases at Children's Hospital Philadelphia and the University of Pennsylvania School of Medicine, and my interest is in the area of vaccines and vaccine safety. Thank you.
Thank you. Dr. Annunziato.
Good morning. I'm Paula Annunziato. I lead global clinical vaccine development at Merck, and I'm here today as the non-voting industry representative.
Thank you, Paula. Next, Dr. Pergam.
Hello, everyone. I'm Steve Pergam. I'm an associate professor at Fred Hutchinson Cancer Research Center in Seattle, Washington, and the University of Washington. And my expertise is in infectious disease in immunocompromised patients.
Thank you, Dr. Pergam. Next, Dr. Fuller. We're going to be introducing our temporary voting members. Dr. Fuller.
Good morning. I'm Dr. Edith Fuller. I'm an associate professor of microbiology and immunology at the University of Michigan Medical School and also faculty in the STEM initiative of the African Studies Center. And I'm a virologist by training as well as implementation science in the community.
Thank you, Dr. Fuller. Next, Dr. Rubin.
Hi, I'm Eric Rubin. I'm an infectious disease physician. I'm at the Harvard T.H. Chan School of Public Health, Brigham and Women's Hospital, and the New England Journal of Medicine.
Thank you. Next, Dr. Hildreth.
Good morning. I'm James Hildreth. I'm the professor of medicine and president and CEO of Meharry Medical College. I'm an immunologist by training, and I study how the immune system responds to virus infections. Thank you.
Thank you, Dr. Hildreth. Next, Dr. Hawkins.
Good morning. Dr. Randy Hawkins, physician in private practice, internal medicine and pulmonary medicine, Charles University. I'm a consumer representative.
Thank you. Mike, can we have the next slide, please?
Good morning. My name is Jeanette Lee. I'm a professor of biostatistics and a member of the Winthrop P. Rockefeller Cancer Institute at the University of Arkansas for Medical Sciences, and my area is biostatistics and clinical trials. Thank you.
I lost connection, so next, can we go through the next slide, please?
Good morning. This is Mark Sawyer. I'm a professor of pediatrics and a pediatric infectious disease specialist at University of California, San Diego, and Rady Children's Hospital, San Diego. My area of expertise is in vaccines.
Thank you, Dr. Sawyer. Dr. Nelson?
Oh, I'm Dr. Michael Nelson, professor of medicine at the University of Virginia and chief of the Asthma, Allergy, and Immunology Division there. I'm also president of the American Board of Allergy and Immunology. My interest and work in vaccines centers on adverse effects and originated during my military career at Walter.
Thank you, Dr. Nelson. Last but not least, Dr. Melinda Wharton.
Good morning. I'm Melinda Wharton. I'm an adult infectious disease physician by training, and I serve as the Associate Director for Vaccine Policy at the National Center for Immunization and Respiratory Diseases at the Centers for Disease Control and Prevention.
Thank you, Dr. Wharton. We have a total of 19 voting and 1 non-voting members today, and I will now proceed with the reading of the conflict of interest statement for the public record.
Dr. Treya, we have a couple other people to introduce. Dr.
Oh, I'm
Levy,
sorry.
Dr.
Okay, thank you. Dr. Levy, we can hear you.
Dr. Levy, are you muted on the top of the screen? Go ahead and
Good morning. Good morning, Dr.
There you go.
Good morning, everyone. My name is Ofer Levy. I'm a physician scientist who directs the Precision Vaccines Program at Boston Children's Hospital, and I'm a professor of pediatrics at Harvard Medical School.
Thank you, Dr. Levy. Dr. Moore, Patrick Moore.
Good morning. I'm Pat Moore. I'm at the University of Pittsburgh Hillman Cancer Center. I'm a professor here. My expertise is in molecular biology and epidemiology, and I specifically study epidemics as well as new human cancer virus.
Thank you. Next, Dr. Perlman.
Yes, my camera's not turning on, so I don't know why that is, but I'm Dr. Stanley Perlman. I'm at the University of Iowa in the Department of Microbiology and Immunology and a pediatric infectious diseases specialist, and my expertise is in coronaviruses.
Thank you. All right, today we're going to be joining by Dr. Peter Marks, who is going to make a presentation also later in the first FDA introduction, please. Dr. Marks, you want to introduce? and thank the committee.
Hi, I'm Peter Marks, Director of Center for Biologics. Thanks.
Thank you. I think now I would proceed to reading of the conflict of interest statement for the public record. The Food and Drug Administration is convening today virtually October 14th, 2021, the 169th meeting of the Vaccines and Related Biological Products Advisory Committee under the authority of the Federal Advisory Committee Act of 1972. Dr. Arnold Monto is serving as the acting chair for today's meeting. Today on October 14, 2021, under topic 1, the committee will meet in open session to discuss the emergency use authorization EUA of Moderna Texas Incorporation's COVID mRNA vaccine for the administration of a booster dose following completion of the primary series to individuals 18 years of age and older. The topic is determined to be a particular matter involving specific parties. With the exception of the industry representative member, all standing and temporary voting members of the WERPAC are appointed special government employees, SGEs, or regular government employees, RGEs, from other agencies, and are subjected to federal conflicts of interest laws and regulations. The following information on the status of this committee's compliance with the federal ethics and conflict of interest laws, including but not limited to 18 U.S.C. Section 208, is being provided to participants in today's meeting and to the public. Related to this discussion at this meeting, all members, regular government employees and special government employee consultants of this committee have been screened for potential financial conflicts of interest of their own, as well as those imputed to them, including those of their spouse or minor children, and for the purpose of 18 U.S. Code 208, their employers. These interests may include investments, consulting, expert testimony, witness testimony, contracts and grants, cooperative research and development agreements or CRADAs, teaching, speaking, writing, patents and royalties, and their primary employment. These may include interests that are current or under negotiation. FDA has determined that all members of this advisory committee, both regular and temporary members, are in compliance with federal ethics and conflicts of interest laws. Under 18 U.S. Code Section 208, Congress has authorized FDA to grant waivers to special government employees and regular government employees who have financial conflicts of interest when it is determined that the agency's need for a special government employee services outweighs the potential for a conflict of interest created by the financial interest involved, or when the interest of a regular government employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Based on today's agenda and all financial interests reported by the committee members and consultants, there have been one conflict of interest waiver issued under 18 U.S. Code 208 in connection with this meeting. We have the following consultants serving as temporary voting members: Dr. Ovita Fuller, Dr. Randy Hawkins, Dr. James Hildreth, Dr. Janet Lee, Dr. Ofer Levy, Dr. Arnold Monto, Dr. Patrick Moore, Dr. Michael Nelson, Dr. Stanley Perlman, Dr. Eric Rubin, Dr. Mark Sawyer, and Dr. Melinda Wharton. Among these consultants, Dr. James Hildreth, a special government employee, has been issued a waiver for his participation in today's meeting. The waiver was posted on FDA website for public disclosure. Dr. Paula Annunziata of Merck will serve as the industry representative for today's meeting. Industry representatives are not appointed as special government employees and serve only as non-voting members of the committee. Industry representatives act on behalf of all the regulated industry and bring general industry perspective to the committee. Industry representative on this committee is not screened and does not participate in any closed sessions if held and do not have voting privileges. Dr. Randy Hawkins is serving as the temporary consumer representative for this committee. Consumer representatives are appointed special government employees and are screened and cleared prior to their participation in the meeting. They are voting members of the committee. The guest speakers for this meeting today are Dr. Sharon Elroy Price, Director of Public Health Service under Ministry of Health located in Jerusalem, Israel. Dr. Ron Milo, a Professor of Plant and Environmental Sciences Department at the Charles and Louis Gartner and a Professional Chair at the Weizmann Institute of Science in Rehovot, Israel. Disclosure of conflict of interest for speakers and guest speakers follows applicable federal laws, regulations, and FDA guidance. FDA encourages all meeting participants, including open public hearing speakers, to advise the committee of any financial relationships they may have with any affected firms, its products, or its known its direct competitors. We would like to remind standing and temporary members that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to inform the DFO and exclude themselves from the discussion, and their exclusion will be noted for the record. This concludes my reading of the conflict of interest statement for the public record. At this time, I would like to hand over the meeting to our chair, Dr. Arnold Manto. Dr. Manto, take it away. Thank you.
Thank you, Prabha. We got through this very promptly, so we're right on time. To start the meeting and to tell us about the roadmap for the day, I'd like to introduce again the director of the center, Dr. Peter Marks, who will give us the introduction of the topic. Dr. Marks.
Thanks very much, Dr. Manto. Good day. I'd like to welcome you to this 169th meeting of the Vaccines and Related Biologics Products Advisory Committee meeting. First, I do want to take a moment to thank our staff, the sponsors, and our advisory committee members for devoting the time for considering the important topics at hand today. The theme of today's meeting is focused on the topic of the use of additional doses of the authorized or approved COVID-19 vaccines to boost immunity in order to prevent adverse outcomes from COVID-19. We'll hear updates on the results on the effectiveness and safety of the deployment of booster vaccines in Israel. We'll consider the issue of boosters for the Moderna and Janssen or Johnson & Johnson vaccines, and we'll discuss the results of a study in which a booster from different manufacturers were given to individuals who had received different primary series for their initial vaccination. If I can have the next slide. The spectrum of COVID-19 ranges from asymptomatic infection to death. And in between these is a range of infection ranging from mild to severe, including severe disease requiring hospitalization. Vaccination is most important for preventing severe outcomes from SARS coronavirus 2 infection, such as hospitalization and death. However, in considering the value of vaccination, one may also need to consider the potential for morbidity from mild to moderate infection, such as blood clots and long COVID-19. In this regard, we now know from recently published studies that vaccinated individuals can develop long COVID-19 if they experience breakthrough COVID-19 infection of any severity. These issues may need to be considered in discussions of the value of booster vaccinations. The next few slides, if I can have the next slide, show the relative preservation of effectiveness of the vaccine over time. Most of the evidence is based on neutralizing antibody titers or real-world evidence on symptomatic infection. And the data that I'll show you comes from real-world evidence, but there are other data as well. Separating waning effectiveness from reduced effectiveness against the variants, such as the Delta variant, can be challenging. And what you'll see on all of these slides is that the vaccines are still very effective against serious outcomes, such as hospitalization. So on the right of each of these slides, you'll see the hospitalizations, and the left, you'll see the overall infections. Like on the next slide. So here,
for the Pfizer-BioNTech vaccine, you can see that over time there is still relative preservation of the effectiveness of the vaccine against preventing hospitalization, yet there seems to be a decrease over the course of time against overall COVID-19 that was observed. And that occurs across the various age groups. There's a suggestion from some studies that it may happen most in older individuals. If I can have the next slide. A similar trend is seen with the Moderna vaccine here. Things are reversed here when you're looking at this, but on the right you see again the flat orange line at the bottom shows that hospitalization remains an event that is well prevented by the vaccine, whereas there is a somewhat trend of that orange line upwards showing that there seems to be some waning of protection against the overall observed COVID-19. And if there's the next slide. Similarly, here, again, now reversing again, you can see here that on the right, hospitalization from COVID-19 with the Janssen vaccine is something that is relatively prevented, and that efficacy is relatively preserved over time. And then for overall infections on the left, you can see that the unvaccinated curve in orange and the vaccinated in blue, and the blue does seem to drop off some over time. So the final slide. So just to summarize here, we'll be talking about booster vaccination today, but it's important to remember that the vaccines still provide strong protection against serious outcomes, especially for younger age groups. I didn't show those data, but some of that will be shown subsequently. The vaccine effectiveness against mild and moderate disease does appear to wane over time for the different vaccines. And we do need to account for the fact that mild to moderate COVID-19 can be associated with adverse outcomes such as blood clots and long COVID-19, even in those who have breakthrough infections after vaccination. But it's important not to forget as we move forward that facilitating higher primary coverage of the entire vaccine-eligible population with the initial series of vaccination should still be a key priority. And I'll just thank you today and for today and tomorrow. We greatly appreciate the input that this advisory committee will provide. Thank you again. Thank you, Dr. Marks. Next, we are going to be hearing from Dr. Sudhakar Agnes Othram, excuse me for murdering your name, who is going to present from the Division of Vaccines-Related Products Applications from OVRR. And he's going to give us the background for the day's activities.
Thanks, Dr. Monto. Could you hear me, see me, and then see the slides?
We can see you and hear you very well.
Okay, thanks very much. Good morning, everyone. I'm Sudhakar Agnihotram from Division of Vaccines and Related Products Applications, OVRR CBER FDA, and today I'll be talking to you about Moderna COVID-19 vaccine application for emergency use authorization of a booster dose. Here is the outline of my talk. I'll start with the description of Moderna COVID-19 vaccine and EUA request for booster dose. Then I'll discuss the considerations for emergency use authorization of a COVID-19 vaccine booster dose. And I'll be talking about COVID-19 vaccines available for use in the United States. And I'll be presenting the overview of today's agenda. That will follow with my presentation of the voting question and a discussion question
for the committee. Please note that the part of my presentation pertaining to the second and the third bullet also applies to the advisory committee discussion tomorrow and is relevant for tomorrow's AC discussion. The phase 1 trial of Moderna COVID-19 vaccine started in February of 2020, and Moderna COVID-19 vaccine was authorized for use under emergency use on December 18, 2020. Moderna COVID-19 vaccine is indicated for active immunization to prevent COVID-19 caused by SARS coronavirus 2 in individuals 18 years of age and older. Regarding the dosing regimen, Moderna COVID-19 vaccine is administered as 2 doses 1 month apart. The third dose for administration at least 1 month after the second dose was authorized on August 12, 2021 for use in certain immunocompromised individuals. Each 0.5 ml dose of Moderna COVID-19 vaccine contains 100 microgram of a nucleoside modified mRNA encoding the viral spike glycoprotein of SARS-CoV-2 from the Wuhan strain formulated in lipids. Regarding the Moderna COVID-19 vaccine booster dose amendment, the amendment was submitted to the EUA on September 3, 2021. Moderna aligned their proposed indication with the population that was authorized for the Pfizer-BioNTech booster dose, and the proposed use of booster dose for Moderna COVID-19 vaccine under the EUA is a 50 microgram dose, 0.25 ml volume to be administered at least 6 months after completing a primary series to individuals 65 years of age and older, 18 through 64 years of age at high risk of severe COVID-19, and 18 through 64 years of age whose frequent institutional or occupational exposure to SARS-CoV-2 puts them at high risk of serious complications of COVID-19, including severe COVID-19. Clinical package in the amendment includes safety and immunogenicity data from 171 clinical trial participants who received 50 microgram booster dose of Moderna COVID-19 vaccine approximately 6 months after completing the Moderna COVID-19 vaccine 2-dose series, which is 100 microgram each. Getting into the rationale for the need of a COVID-19 booster dose. The emergence of highly transmissible Delta variant of SARS-CoV-2 has led to considerations of the potential need for booster doses for fully vaccinated individuals. Data from post-authorization effectiveness studies conducted suggest that the currently US authorized or licensed vaccine remain effective in protecting against severe disease. However, some data suggests that effectiveness may be waning against mild disease and against severe disease in elderly individuals. Concerns have been raised that declining neutralizing antibody titers or reduced effectiveness against symptomatic disease may herald significant declines in effectiveness against severe disease. Talking about the emergency use authorization, FDA may issue an emergency use authorization of an unapproved medical product following an EUA declaration if the following statutory requirements are met. The agent referred to in the EUA declaration can cause a serious or life-threatening disease or condition. The medical product may be effective to prevent, diagnose, or treat the serious or life-threatening condition caused by the agent. The known and potential benefits of the product outweigh the known and potential risks of the product. And then, if no adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition prevails. I will now be talking about the COVID-19 vaccines available for use in the US. Pfizer-BioNTech COVID-19 vaccine, or Comirnaty, is licensed for use as a 2-dose primary series in individuals greater than or equal to 16 years of age. Pfizer-BioNTech COVID-19 vaccine is available under EUA as a 2-dose primary series in individuals greater than or equal to 12 years of age, and a 3rd primary series dose is available under EUA for use in certain immunocompromised individuals. The booster dose of Pfizer-BioNTech COVID-19 vaccine is available for use at least 6 months after completion of the primary series in individuals greater than or equal to 65 years of age
individuals 18 through 64 years of age at high risk of severe COVID-19. In individuals 18 through 64 years of age whose frequent institutional or occupational exposure to SARS-CoV-2 puts them at high risk of serious complications of COVID-19, including severe COVID-19. The Moderna COVID-19 vaccine is available under the EUA as a two-dose series in individuals greater than or equal to 18 years of age, and for use as a third dose in certain immunocompromised individuals. The Janssen COVID-19 vaccine is available under the EUA as a single dose in individuals greater than or equal to 18 years of age. Getting into the benefit-risk considerations for booster dose. The available data should support the effectiveness of a booster dose, specifically against currently circulating SARS-CoV-2 variants. That is, benefit of the booster dose should be considered relative to the benefit provided by previous vaccination with a primary series. Available data should at minimum characterize the most common adverse reactions associated with the booster dose. There are uncertainties regarding risks, example myocarditis, that are also considered and would be further evaluated during post-authorization surveillance. FDA's evaluation of the safety and effectiveness data of a booster dose of the Moderna COVID-19 vaccine and additional input from the VRBPAC is essential for weighing the known and potential benefits and risks. Getting into today's agenda, we are currently in the FDA introduction, which will then have a 5-minute Q&A session, and that will be followed by a presentation of data relevant to the need of the booster dose from Dr. Al Roy at Ministry of Health Israel and Dr. Milo from Weizmann Institute Israel. There will be a 15-minute break after that. Then there would be a sponsor presentation titled Safety and Immunogenicity of a 50 microgram booster dose of mRNA 1273 Moderna COVID-19 vaccine to be given by Dr. Jacqueline Miller from Moderna Therapeutics. This will be followed by FDA presentations from Dr. Tina Monju and Dr. Hui Li Wang. There will be a 10-minute question and answer session after that, followed by a 30-minute lunch break and an open public hearing for 60 minutes and a 15-minute break. There would be additional Q&A session regarding the sponsor and FDA presentations, followed by the committee discussion and voting. Here is the voting question for the committee for today's AC. Do available data support the safety and effectiveness of Moderna COVID-19 vaccine for use under EUA as a booster dose, just 50 microgram of mRNA 1273, at least 6 months after completion of a primary series in the following populations: individuals 65 years of age and older, individuals 18 through 64 years of age at high risk of severe COVID-19, and individuals 18 through 64 years of age whose frequent institutional or occupational exposure to SARS-CoV-2 puts them at high risk of serious complications of COVID-19, including severe COVID-19. We also have a non-voting discussion question for the committee. Considering the information presented today and at the meeting of the VRBPAC on 17 September 2021, including the updated information on effectiveness of mRNA COVID-19 vaccine, please discuss whether available data support use of a mRNA COVID-19 vaccine, that is Pfizer-BioNTech or Moderna, booster dose administered at least 6 months after completion of the same mRNA COVID-19 vaccine primary series in the general population of adults in an age group less than 65 years. For the purposes of this question, age groups below 18 years should not be considered. I'd like to thank the advisory committee, supervisors, and management for providing the opportunity to present here. Thanks, and now it is open for Q&A session.
Thank you very much. We have our first Q&A session, and we have a little more time because we're ahead of schedule to discuss what we're going to be doing today and to get going in terms of our thoughts. And Dr. Kurilla has raised his hand.
Thank you, Dr. Monto. Yeah, one question. Can you clarify the relationship between the 6-month booster EUA with regard, does it supersede the EUA that was issued for the immunocompromised, or do both of those stay in effect? It seems like there might be a little bit of confusion because the immunocompromised would also be at risk for serious COVID disease, but that's 1 month after versus 6 months. I'm just wondering how those will play out.
And what about the dose?
I can answer that question. Yeah, thanks for that question. The third dose for immunocompromised is actually 100 microgram dose, and then the 6-month EUA for the booster dose is for 18 to 64 years in individuals who have comorbidities, and then above 64, it is for everyone. For Moderna COVID-19 vaccine, the dose is 50 microgram for the booster dose, that is the third dose. But the dosage for immunocompromised for Moderna is the 100 microgram, which is the third dose. And the immunocompromised may also opt to get another booster dose. That would be 50 microgram. And if anyone else from FDA wants to jump in to answer that question, they're welcome to.
This is Dr. Kurilla. I think I take your point, and I think we've gotten some feedback that when we reissue the fact sheets for the current emergency use authorizations, we'll make it clearer about the distinction between the third doses for the immunocompromised and the issue of a booster for an individual who's received 3 doses as a primary series. And that's a very good point that we have to just make sure we clarify. Thank you for that.
And so just to be clear, for the immunocompromised population, you have changed the primary vaccination sequence then to a 3-dose?
We have not changed it, but we have allowed, it's permissive if a third dose is desired based on the considerations of that individual, such as an individual who has been through solid organ transplant, where there's good evidence that they often don't make a good immune response to 2 doses, that at the discretion of a provider, a third dose could be administered. We note in the authorization that even then, the protection may not be perfect, and that's why we recommend that people still continue to use reasonable precautions such as mask wearing, et cetera.
But the 6-month boost then for them would be a 4th dose. They would still be, you would still consider them eligible under this EUA for a 4th dose?
You know, I think this is one where we probably need to discuss. This is far enough in the future that I don't want to make a definitive statement here. It's something that we do, though, have to cover when we reissue our fact sheets. And I'd be very welcome to have the advisory committee, Dr. Monto, later on have a conversation about that, because I think there is some dialogue that could be had.
And there's potential for a lot of confusion as to who needs what.
Right, Dr. Kurilla, I don't know that we're going to be able to fine-tune the whole program, national program, in the next couple of days. I think they're going to be, we really need to think of broad concepts, especially when we get into our discussion after the vote later this afternoon. Dr. Meisner.
Thank you, Arnold, and thank you both presenters. I think my question is going to be a little bit easier than Dr. Kurilla's question for you, and it's for you, Dr. Marks. You showed 3 slides that demonstrated real-world effectiveness for the 3 vaccines, and could you just remind me? There were vaccinated and unvaccinated curves that were demonstrated there. Who was in the unvaccinated group? Did that group have the same degree of risk factors such as age as the group who were vaccinated? Because they probably weren't from the original trials, right? Because didn't most of the placebo recipients cross over?
So both of those, both for Pfizer and Moderna, those were from Kaiser Permanente studies, and the papers are published in the Lancet. The references are on the slides. They did match for age, disease score. These were from their HMO databases. So these were cohorts that were matched, and our statisticians in looking these over feel that reasonable matching was done. But you know the limitations of all of these. These were covered at the last meeting, the limitations that are present with these studies. Although the one thing that is true is that the studies, though one might see differences in magnitude, they do all seem to trend in the same direction here.
Thank you.
Thank you. Dr. Sawyer.
Thanks. I just want to go into the discussion today with a clear understanding of whether the voting question that was presented is the only question we would deal with. Last meeting, we decided that the voting question, we voted against the overall question that was posed, and then a revised version was presented, and we then voted again on that. And would we follow the same process today?
I'm happy to try to respond to that. I mean, we anticipate having the vote on the question that's there. Wanting to take make best use of the advisory committee's expertise. If the advisory committee did not, if there was a problem with that question that became apparent during this meeting, we would potentially take it upon, if the committee was acceptable, revising it. But it is our intention to vote on the question that is the questions that were presented, the one question that was presented, and to have the one discussion question.
Right, and just note that the voting question derives from the sponsor's request to the FDA, and that's the reason we did what we did at our last meeting. Dr. Offit.
Yes, Sadakar, I had a question that hopefully you could clarify something. One of the statements that was on one of your slides, you stated that because there was a decrease in effectiveness associated with the vaccines over time regarding infection, that likely presaged a decrease in effectiveness against serious disease. But one could argue that decrease in effectiveness against all infection is more likely mediated by neutralizing antibodies, which are going to erode over time, whereas immunological memory is probably more likely associated with protection against serious illness. So I'm not sure why one would argue that one would presage the other.
Well, thanks for that question, Dr. Offit. I can try to answer that question. The decrease in effectiveness against mild to moderate disease can apparently be also driven by the decrease in quality of the neutralizing antibodies that are present. And then that can eventually lead to severe outcomes such as hospitalization, etc. I mean, point well taken that the immunological memory can also play a role in protection against severe disease. But over time, in vaccinology and immunology, when the immune response declines over time, then that can also eventually lead into severe disease. So that is the explanation that I can give. But anyone else from FDA wants to jump in?
So Dakar, you're arguing that arguably immunological memory would decline over time. I mean, and I think that some of the Israeli data show that in the 70 to 79-year-old, that's very possibly true. But I just wonder whether in a younger group that really would be true. But again,
no. Dr. Offit, my suggestion is let's see the Israeli data that they present today, because they may answer some of that question today, I think. I'm sorry, I don't mean to cut you off. Just think it's. I think you may that may be a. I totally take your point, and they may address that today.
Okay, thank you, Peter. Thank you, Sodekar.
Thank you.
Dr. Moore.
I see we don't have anyone presenting from VAERS or CDC on giving us an update on serious adverse events, particularly comparing Pfizer to Moderna mRNA vaccines. Is there a chance for us to get that information before we evaluate Moderna's booster? This is on the primary series, of course, but is there a chance for us to get that information before we evaluate Moderna's booster, or how do we deal with that, particularly with the issue of myocarditis, particularly in males, that suggests that we may want to tailor our recommendation more?
Yeah, thanks for that question, Dr. Moore. We have a presentation from Office of Biostatistics and Epidemiology from Dr. Hui Wong Lee, who will be talking about that. That would be followed by Dr. Tina Monge's presentation. So that would address your question.
Great,
Dr. Marks or anyone else, if you want to jump in. Yeah.
And just to note that our voting question actually is, for the most part, down to 65 years of age. The rest is going to be part of the discussion afterwards, in which we're going to be looking at and can ask some questions about age groups as such. We can unusually have a little more discussion here because I've heard that Dr. Elroy Price is not in place yet to give her presentation from Jerusalem. So any of our committee that wants to ask a few more questions, we've got exactly 7 minutes to give her time to get in place. Yeah, Dr. Meisner, is that you from before or new?
No, it's new, Arnold. Let me try and position myself here. I have another question.
It's all very tricky when you're virtual.
Thank you. Another question for Dr. Marks. So the question from the sponsor relates to individuals 65 years of age and older, people 18 to 60 through 64 who have underlying risk factors. And then my question relates to the third category. It seems to me there's some confusion between people who are at risk of severe disease and people who are perhaps at greater risk of being exposed. And I don't, first, are there any data to say that people at greater risk of being exposed are likely to get more severe disease? And I worry because that's been interpreted as, for example, a person who bags groceries at a grocery store is an increased risk. And that, to me, that wasn't quite the intention of what we discussed during the last meeting. Could you comment on that, and your perspective on that?
So thanks for the question. It's one where I take your point, and we discussed it a fair amount internally. The question is, some people are at greater risk of getting COVID-19, right, because they are just constantly exposed. If they get it, you're right, the grocery store worker, for that infection, there's nothing that says that they would be, because they're a grocery store worker, does not mean that they would get more severe infection than another individual. But it was part of kind of the overall consideration there. Again, if this committee, as they discussed, that was the purpose of the second question today, to allow the committee to refine what we have, and we'll very much value that because we know it's not perfect. And to the extent that I'll just say this, to the extent that we can try to harmonize between the various vaccines to the greatest extent, we greatly appreciate that, because in the practice of this of rolling things out, we think that will make things easiest and create the least confusion operationally. But I really welcome that discussion. That was the purpose of the second discussion question. Thank you, Dr. Meisner.
Thank you, Dr. Marks.
Dr. Rubin.
Thanks, Dr. Monto. I have a question about safety and what the FDA's view of what a reasonable safety sample is for a third dose. The difference between, Pfizer did a relatively small trial and Moderna is going to present the results of a relatively small trial of third doses. Pfizer had all those real-world data from Israel of 1 million of people who had received the vaccine. So how do you think about an adequate sample size to view safety?
Thanks for that question, Dr. Rubin. I can attempt to answer that question. With regards to Moderna, we have safety data from 171 participants and additional safety data from 173 participants as well. So approximately around like 340 participants for a booster dose, which is being reviewed for the emergency use authorization of the booster dose. And then, we believe that, for emergency use authorization, that is adequate for authorization of the booster dose. But if there is anything that anyone else wants to add from the FDA, Dr. Marks or Dr. Fink.
I think the important piece of this to understand is that I think we take the totality of the evidence. I think some of this is understanding what the most likely adverse events have been from mRNA vaccines. And I think probably the major thing we'll be looking at in post-deployment surveillance would be myocarditis. Given the incidence rates of that, I think this is one of those areas where we will look at using our large databases to make sure that the incidence as it's deployed is not excessive compared to what we would expect. And I think Dr. Fink looks like he wants to chime in here.
Thank you. I just wanted to add, and this was discussed several weeks ago with the Pfizer booster dose request, that we did issue guidance regarding emergency use authorization of modified vaccines to increase protection against variants. And even though we're not talking about modified vaccines, the considerations that we outlined in that guidance for booster doses of modified vaccines, we do think are very applicable to these homologous booster doses that we're considering then and also today. In our guidance, what we said is that based on a well-characterized safety profile of a primary series, that a safety database of around 300 or so individuals who received a booster dose would generally be sufficient, provided no signals are identified, to support emergency use authorization of a booster dose. It was very nice that we heard about data from the Israeli experience with the Pfizer vaccine last time. Of course, FDA did not independently review those data. And so they did not, contribute in a major way to our consideration of the risks and benefits for the U.S. population, although we certainly did consider those data in part. So I think what Moderna has provided us today, which you'll hear more about today later, does align with the principles outlined in our guidance for study of booster doses to support emergency use authorization.
Thank you. Final question from Dr. Fuller, because Dr. Price is now ready to go. So, Dr. Fuller.
Yes, thank you, Arnold. So I just want to say to the question that Dr. Cody Meisner asked about the third category of high risk, that at least some of us think that a person who's at a grocery store may not be, we don't know if they're at higher risk for disease, but they're certainly at higher risk for exposure. And I, for one, am grateful that we have that allowance that someone who would like to get the booster is able to do so. So if I understood him correctly, he was concerned that maybe that wasn't what the committee meant. And for one member of the committee, that is exactly what I meant. I would like people like that to have the choice. So just want to clarify that for everyone, or if that's something that we need to talk about later.
Okay, well, thank you all. We've had an unexpected and rather robust discussion of the day's activities, and now I'd like to give over to Dr. Sharon Alroy-Price in Jerusalem, who will give us booster protection across ages data from Israel.
Thank you. With me is Professor Ron Mullo, and I want to use this opportunity again to thank the 4 leading academic institutions in Israel who have helped us do this, create this data and analyze the data. And I am trying to move to the next slide.
At the bottom of the screen,
yeah.
You'll see the 2 arrows here. I'll point. There you go.
Yeah. So we are presenting Israeli data. We have no competing financial interests. I do want to say again that Israel, MOH, Ministry of Health, and Pfizer have data sharing agreement. And in relation to the booster effectiveness, also this data that we're showing now, only the final results of the analysis were shared with Pfizer and was done by these 4 academic institutions independently. And again, I want to say, like we said last time, we're coming here not to tell anyone what to do. We think every country has not just the right, but the obligation to do what is needed for their citizens. This is the decisions we've done for Israel based on our data, and if our data can help anyone else in the world, we're happy to share it, but it's not that we're telling anyone else what to do. So what has happened since the last time we've been here, which was about a month ago? A large majority of elderly population received a 3rd dose. You see in the blue line that over 95% of the 60-plus have been vaccinated with the 3rd dose. And similarly, the other age groups that we've opened gradually by steps is increasing. And so we have nearly in all populations over 50% already with a booster dose. This is a slide we showed last time showing that shortly after starting the booster dose at the age group of 60 plus, we saw a decrease in the number of confirmed cases among that group, whereas the other groups, age 60 and below, were continuing to rise. And when we now are looking nationally at the data, we're seeing now a decrease in the percentage of positive tests, also in the reproductive number. Once we're adding more and more age groups into the booster protocol, and what we're seeing basically is a break in the pandemic curve in Israel. You see here a separation of, in the green line, the people who were vaccinated with a booster dose, the adults who were vaccinated with a booster dose, and in the black line, those who were unvaccinated. And you see that with the beginning of the booster dose, we saw a decline in the infection rate. Here you see the severe cases among those who were vaccinated, and those decreased sharply. But you see at the end of the slide on the right that now we are seeing a decrease also in the unvaccinated population. So the fact that we have high coverage of vaccinated individuals with a booster dose is now leading to a decrease in the overall severe in the overall pandemic curve, but also in the severe cases. And I'll move it to Ron now.
Okay, so I will be continuing from what Dr. Price was presenting to look at the detailed study that we did on those several million booster shots that were given. So the data that I'll be presenting is based on those aged 16 and above who were fully vaccinated before May 2021. These are the people that have been vaccinated at least 5 months prior to the booster dose, and it consists of altogether 4.6 million people who were vaccinated at that, until that time. And this consists of about, during the study period, of about 100,000 confirmed infections, over 1,000 severe illness cases, and over 250 deaths. And this has happened in the study period, which is between August and the end of September, maybe even beginning of October, as you see in the different slides. And this is following the booster campaign. And we'll be looking at the different age groups, as you'll see in a minute. Let me begin with ages 60 and above, and I'll be talking first about confirmed infections. This is complementing the results that were already published in the New England Journal of Medicine and were presented last time. And all the results that I'll be showing you are also shared online through the medRxiv. We'll be looking at time following the third dose, so after giving the booster. That's what you see on the x-axis. And on the y-axis, you see the fold reduction in the rate confirmed to those with 2 doses. Meaning we're taking people that had the third dose and we're comparing them to people who had only 2 doses. And we're looking what happened to the rate of confirmed infection as time progressed following the booster. We expect in the beginning to have some transient effect and some time delay until you start to see the effect, both because of the time response of the immune system, which takes several days, and then also the inherent delay between a time between a person gets infected and the time their infection is confirmed, which in Israel is on the order of 5 days, which is also consistent with the latent period. And therefore, one could look at the time window from day 12 and beyond. And this is what I'll be showing. I should say also that everything I'll be showing you is based on a Poisson regression, where we're adjusting for age, for gender, demographic group, second dose period, and incidence in area of residence. Meaning we're looking at the location, at each location where the people live, and at that given time, and taking as a confounder or something to be adjusted for the rate of incidence at that time frame. What I'll be showing you is based on an observational study, and I want to be very clear here. So observational studies have their limits. We did everything we could in order to adjust in the ways that I'll be showing you, and we thought it is best to try and share it with the community as we do also with you, but to share it for peer review as fast as we could and to put it all publicly available online as I'm presenting, and I'm trying to put all the links to enable everybody, including the general scientific community, to be able to comment on our work. When we take this data and we're looking at what is the level of protection, meaning what decrease in the rate of infection is being observed, we see on the order of about 10-fold or 12-fold, somewhere above 10-fold, overall protection when doing the analysis based on the Poisson regression. You can see that the confidence interval, this is 95% confidence interval, is relatively small. That comes from the fact that we already have, several tens of millions of risk days, both in the non-booster, those that only had 2 doses, and those at days 12 and over. And overall, we're talking about somewhere above 14,000 cases of infection within this age group. In the study period, which is between July 30th, which is the time where the booster campaign has begun for those age groups, and October 4th, this is the last update that we had for the data. So this is presenting the results for the age group of 60 and above. It continues and I think enforces what we also presented last time about the effectiveness of the Pfizer dose in the regimen of 3 weeks between the first and the second dose after 5 months to give a booster dose. Let me move on now to present what we've been observing when analyzing all the other age groups where, as Dr. Price was presenting, most of the Israeli population has now taken that booster dose. So this is a bit of a busy slide. Let me walk you through. I think you also saw this, which is the ages 60 and above. You can see in a similar format what happened in ages 50 to 59, 40 to 49, 30 to 39, and 16 to 29. Again, I'm sorry for the small font, but this is days following the booster, and this is the fold reduction in the rate. I think you can see that the patterns are relatively similar. You can see, by the way, that we have, if for the ages 16 and above, we have 2 months of follow-up. For 50 to 59, we have 2 weeks less because we did it in a serial manner, such that there was some delay, so that we waited 2 weeks until we opened it to the ages 50 and 59, and then about a week for ages 40 to 49, and therefore we have a limited follow-up time for those age groups. But we could see that the effect begins similarly about 12 days following that, and the results are summarized here in terms of the rate ratio for day 12 and over versus the non-booster. And you can see they are on the order of 10-fold protection. You can see the confidence interval, and you can see we have quite a few cases in order to perform the analysis within all age groups, and all the results, as I said, are summarized and updated and are now under revision for publication. I would also say that we see similar patterns across the age groups in terms of both the timing and the magnitude, though not completely identical, which probably would be of interest for further analysis. All of that I've shown you is, again, trying to correct for all of those different confounders for which we have data that are able to do that. And in all cases, we're doing the analysis from the time of booster eligibility, that's for the different age groups because they were one after the other changed somewhat, until the first week of October. Beyond looking at the fold rate of reduction, we also looked at the absolute rate and what happened to them. So what you see here is the confirmed infection rate per 1,000 risk days, and we're comparing between those that took the booster versus those without the booster on the second dose. And you can see in yellow the non-booster, which is on the order of 100 confirmed infections per 100,000 risk days in the prevalence that we had during the study period in Israel. And in green, you can see what happened to the absolute rates for confirmed infections for those that took the booster 12 days and onwards, everything per risk day. Understanding that it's important to try and look at it from different perspectives, we were trying to be as careful as we could, and beyond the approach which is often being utilized in order to study such analysis, the Poisson regression, we also took a second approach based on a different framework, and that is using matching. So basically for every person that took the third dose, we're matching a person that took only 2 doses, and we're following them through time, and we're making sure that the matching is such, as you can see also that is being done in the literature, such that you're comparing properties as much as possible, meaning the age, the demographic sector, all sorts of things as much as you can in order to ensure that the comparison is as similar as possible. And we find that the results also in terms of the rate ratio between the two cohorts are such that we're getting a similar protection as we saw during the Poisson regression. We followed another approach, and that is looking at using a temporal comparison, such as your control group, instead of being the ones that only took the second booster, we have an alternative control where you're looking just at the people that also took a third booster, but you're looking within the time frame of days, which is between 3 and 7 days post-vaccination. So this is the rationale for that, is that one expects little effect of the booster on confirmed infections in days 3 to 7. The reason for that is the combined effect of the delay until the effect of the vaccination of the booster, together with the delay for being confirmed. So even though there is some response already in days 3 to 7 of the immune system, you would not expect that you would already get the symptoms and be confirmed. Therefore, it's another way to perform the analysis. I would say that it's also conservative in the sense that even if there is some effect, or maybe it's not zero, there is some effect, and there are also other effects, which is what's known as the healthy vaccinee bias, which relates to the fact that people that tend to be, that take the booster are those that feel better, because those that do not feel well tend to not come and take the booster. All those would give you some seemingly protection level, which is, you might even be seeing it here. And that would make it such that when you take the ratio for the control group, it means that you get a lower protection than the actual one. But we thought it's important to try and use as many alternative and optional ways. And this is this approach. And let me show you the results. You can see them compared here for the different age groups. This is using the alternative control group, where the control group is 3 to 7 days post-vaccination, when the booster has little effect. And we see on the order of, indeed, somewhat lower levels between 4.8 to 11.2, where I just want to point out, when we're talking, say, about around 5-fold protection, that means 80% lower rate of infections. Okay, so that's not something, it's not, I would say in absolute terms, it's still 80% decrease in the rate for those age groups in terms of confirmed infections. Following our analysis of confirmed infections, we wanted to look further at what happens in terms of severe disease. So let me move on to that. What you see here are results for severe disease, and we've been following the definition of the NIH regarding the resting respiratory rate and the oxygen saturation for the definition of what is severe disease, and we're looking across the age groups. You can see that the numbers are generally, number of outcomes is obviously lower, but still we find that we had, at least in the age group of 60 and above, and even in the ages of 40 to 60, unfortunately, we had quite a few cases in Israel. And you can see here what happened in terms of the rate ratio, and we could see a very significant decrease in the rate of severe cases in those age groups of 60 and above and 40 to 60. In the age group of 16 to 39, the number of cases for the booster group is very low, and therefore there are too few cases to estimate reliably the rate ratio, even though you can see the raw numbers here, where you can see the number of cases and the risk days, the number of cases and the risk days at risk. And all of this is, again, done in the same approach of trying to control for all these confounders as much as possible. This is the analysis on the severe disease in those age groups using the Poisson regression. Here is the same analysis, but now using the alternative control group, where you're comparing the people 12 days over and days 3 to 7 as your control. And you can see here the level of protection that we're finding. So 6.5 for this age group, 3.2 in this age group, and too few cases to estimate reliably in the lower age group. Just to clarify again, say 3.2, that means roughly 60% lower rate, or somewhere above that, actually 70% lower rate of severe disease. These are the changes in terms of absolute rates for severe disease. Again, for 100,000 risk days, you can see that for the non-booster versus the booster, the non-booster versus the booster, there is some thin line here, sorry. And you can see that the numbers, obviously there's a dependence on age, but we see that there is quite a dependence on whether a person takes the third dose or does not take the third dose. Finally, I want to present our results regarding the analysis of death as an outcome in the ages 60 and above in both approaches, both with the days 12 and over versus those with non-booster, meaning only 2 doses, and as a comparison to those with the alternative control for days 3 to 7, we see a very significant protection. We're about 4.8, that's about 80% decrease in the rate of death. For the ages 40 to 59, there are too few cases to be able to estimate those values. So in summary on this analysis, we find that the booster dose improves the protection over the second dose, and also regarding both in terms of confirmed infection and in terms of severe COVID-19, where the exact values of reduction depend on the age group, but I would say overall we see high levels of protection and the decrease in the rate. In terms of severe disease, over 80% decrease in rate ratio over the second dose for the ages 60 and above, and in ages 40 to 60, over 60% decrease in the rate ratio over the second dose. And finally, we see that the booster dose decreases the COVID-19 associated death rate around 3 to 10-fold among the elderly. With that, I want to go back for 2 minutes to the nationwide observations following the booster dose before Dr. Price presents our results regarding the safety of the vaccine across the different ages. So just going back to here, I remind you that in Israel, we're doing the confirmed infection based on PCR testing. So it's both following symptoms and without symptoms as part of contact tracing and for other reasons. And looking at the number of daily cases, we saw them rapidly increasing, which was the rationale for beginning the booster dose administration for the ages 60 and above. And then we saw with a delay of about 2 weeks, which is roughly what one would expect given what we just talked about, we saw a specific decrease, whereas the below 60 continued to increase rapidly. And we also checked within this, let's say, the ages of 50 to 59, 40 to 49, they also continued to increase until later on where booster was administered. I'm showing here values until September, where in September you already started to see the effect of the other booster doses, and if anybody's interested, we could afterwards talk about it further. Here, I think you saw, this is looking at the positivity, percentage of positive tests in Israel. So I think what is interesting to notice is that when we started the booster dose, even though I showed you that the overall number of infections within the group of the ages 60 and above started to drastically decline, the other age groups continued to rise. And as a result, and we also opened to the ages 50 and above and 40 above also in order to protect them. And what we find is that only after opening to more age groups, the absolute percentage over all the population has started to decrease. And now this is from 7th of October, it was 2.6. Now it's actually at about 1.5, much continuing to decrease. Following the booster dose to more age groups and not just as a result of the ages 60 and above. By the way, we're looking here at the percentage of positive tests and not just based on the number of cases, which we could also show you, and that is because there are effects from the high holidays that are taking part in Israel during September, and therefore this is a more robust way to analyze this. Finally, looking at what happened in terms of severe disease, severe cases in Israel during that time period, we saw that following the administration, or within the time where this was the booster campaign began, you can see that in green here are the vaccinated people. And you can see that they were the majority, about 2/3 of the severe cases, the daily severe cases that we had, were of those that were vaccinated. That was a combination of the waning and of the Delta variant. And we saw that after a delay, roughly of 2 weeks, we started to see a stabilization and then a decrease as a result of, as Dr. Price was saying, about 90-something% of those within those age groups have been taking the booster. And there was a continued rise in the number of cases for the unvaccinated such as even though they're only a small population of the people at risk of the adults, less than 20%, they were in charge of the vast majority of the severe cases in Israel ever since. And we started to see a decrease of that, and in the same time when we started to see overall incidence in Israel declining after wide booster adoption in the age of 16 and above, which can be interpreted as the fact that once you had the booster adopted by many age groups, the overall incidence in Israel declined significantly, which is what I've just been talking about. It is now over 5-fold less than it used to be in terms of overall incidence in Israel. And that also started to decrease the number of severe cases also among the unvaccinated in all age groups, including the elderly, as a result of the fact that the incidence is now much lower. And with that, I'll move to Sharon.
I'll take it from here. I'll just want to emphasize something that Ron said, but it was a question last time, and so I want to put a notice on it. The severe definition is the NIH definition. It's not something that is specific Israeli construction. It's something that we're using the NIH definition for severe case, and we have been using the same definition since July 20. So the change in the numbers that you're seeing is not because there was some change in definition midway. The booster is important to see the vaccine effectiveness of the booster, but as important is to see the safety data. And so now we have more data on the safety of the booster in younger age groups. And I will show you the data, the rates of adverse events per million doses within 30 days post-vaccination. It's updated until 4 days ago. And for the youngest age groups, which is 16 and above, we have for 50% of them more than 30 days of follow-up. So for about 50% of them, all the adverse events following vaccination would have happened until by now. There is a limitation to note. As we said last time, the reporting is based on passive surveillance, so we are looking for healthcare providers to report to us. But the myocarditis data we are proactively looking for. So we are calling hospitals and asking for the data. And so this is something that is more hands-on with myocarditis, knowing that this is an adverse event that is connected usually to the second dose of the vaccine in younger males. So the data that you're seeing here is the rate of adverse events by category and age groups. You're seeing on the left the first dose, the middle the second dose, and on the right the third dose. And you're seeing that at least we have the same amount of adverse events, not more. Again, we are aware of the fact, the limitation that could be underreporting, but it's the same system for all 3 doses that we provided. This is the rate of systemic adverse events by dose. Again, the third dose on the right is not higher rates of adverse events. This is the rate of local adverse events. Again, similar, if not lower. Neurologic adverse events in gray is the third dose, and I should have mentioned the number of cases. So for first dose, we have more than 6 million 6 million vaccinees. For the second dose, 5.6 million, and for the third dose, 3.7 million. So it's big numbers, and you see on the gray the rate for neurologic adverse events. Allergic adverse events, similar. We have to mention that between the first and the second, if someone developed an allergic adverse event, usually they would not be given an additional dose. So part of it is those who were allergic to the medication were not given another one, but we're not seeing huge amounts of allergic adverse events post the third dose. And what is more important to us is the serious adverse events. You see here the definition of serious adverse events that result in death is life-threatening, requires hospitalization or prolongation of existing hospitalization, result in a persistent or significant disability or incapacity in congenital anomaly, and other important medical events that required intervention. This is kind of common serious adverse event definition, so we're not defining this in any other nationally accepted way. For 3.7 million booster doses administered, we had 44 serious adverse events reported. And for those adverse events, we have a special committee that looks into each and every case, looks at the clinical data, and defines whether it's connected or possibly connected to the vaccine. And here you have the results of that group. You see here for age 16 to 59 on the green on the left, out of 2.5 million vaccinees, we had 9 cases of myocarditis and 8 cases of perimyocarditis. So altogether 17 cases of either myo or perimyocarditis. And in smaller rates, other adverse events, some of them related, like the allergic reactions, and some of them, like the DVT, that were deemed not connected to the vaccine. And on the left, you see for age 60 and above, out of 1.2 million vaccinees, the adverse events that are seen here, and they were deemed not connected to the vaccines. In one of the cases is still under investigation. And in one case, the causality is possible. So myocarditis, which is the one adverse event that we found connected in Israel and other countries to the Pfizer vaccine, usually after the second dose. So what you see here in this table is the data for the first dose, the second dose, and the third dose, and it's split to female and male and split by age groups. And so what we saw before is really a high number, increased rate of myocarditis among 16 to 19 and 20 to 29 males. This is from the prior vaccination campaign. What we are seeing now with the third dose, you see here the number of cases. This is what I mentioned before. We have 17 cases of either myocarditis or perimyocarditis. And so we're not seeing an increased risk of those events following the booster dose. Saying again, for about half of the younger population, we don't have the full follow-up observation time. We do have them for roughly 50%. So in summary, the booster dose in Israel was effective and so far had safety profiles similar to the other doses. We have improved protection against confirmed infection for all ages 16 and above. We have improved protection against severe disease in ages 40 and above. And I have to mention, we are always talking about the fact that younger people have less tendency to go into severe and critical conditions and to dying. But as you saw in the slides that Ron showed before, we did have mortality and severe cases among the younger age groups who were doubly vaccinated but did not get the booster dose. So it could impact even younger than 40 years old for severe and critical disease and mortality. The booster dose adverse events are not more acute than the first or second dose, and their rate of occurrence is not higher. And I think that we can say when we're looking at all the data, the epidemiological data in Israel so far, is that the administration of the booster dose helped Israel dampen the infections and the severe cases in the fourth wave. So we are now coming out of a fourth wave that was, without the booster dose, I believe, would have put us in a worse place with really high burden on hospitals with severe and critical patients, and we were able to get out of this wave due to the booster dose. Thank you, and we are more than happy to answer any questions that you might have.
Thank you so very much, Dr. Price, Professor Milo, for the presentations. A very good tag team of the two of you going over the material. Dr. Price, you were very careful to say that the side effects of the third dose were no higher than that of the second dose, although some of your data suggested that they might be lower. What do you, not going on record, but just giving your personal opinion about this, given the shorter time and selection that might have gone on, what do you really think about this?
I think it's lower, but I want to be very careful about how I present this because there could be underreporting and there could be a difference between the underreporting of a third dose compared to the first and second. With a new vaccination campaign, the awareness maybe is higher, and with a third dose may be lower. So I'm trying to be very careful about that, but I am very confident about the serious events. I think that the serious events are being reported to the Ministry of Health, and especially the myocarditis cases, which we are actively looking for. We are going out and doing active surveillance on, so we're very confident on those numbers.
Do you have a feeling that young males are holding back from getting vaccinated?
I think that there is some concerns among younger males, even though the fact that it was the publication in the New England Journal of Medicine of both our data and CLALI data that showed that most of the cases are mild and are resolved completely without sequela was important. So I think there could be some concern, but I think we're showing in the data that it's a really rare occurrence and mild in most cases.
Thank you. Dr. Gans.
Thank you so much. I really appreciate you coming and sharing your data with us, and I just want to say it's really beautifully presented and very accessible. I did have a couple of questions, if that's okay. One question is, in catching these quote cases, I'm wondering if you have any mandatory testing. So is there a difference in the way that people are getting tested now? So for instance, we have some businesses that require biweekly testing, and are we just catching people who aren't symptomatic, or are most people just getting these tests because they're symptomatic? So that's one question, just to understand the data. But I think my overarching question, because I think your data is very compelling in the lens that you bring to it. So we aborted this wave. I'm wondering if you could overlay, because I'm sure you thought about this, the idea that many countries show a similar pattern regardless of what they do with vaccination. So there's sort of this wave of epidemics that come and go. So I'm wondering if you can overlay what would have been the natural history of the disease with your data, because it's very compelling. And then lastly, just so that I can throw these three out, do you have any immunologic data that you did sort of side by side with this so that you can start to understand these are the breakthroughs, here's the here's the immunity that we saw at that point, so we can start understanding any immune correlates of protection? I realize that's sort of a side study.
I'll start from the end and see if I can remember all the way through.
A lot of questions to answer.
So ahead, first, about the study. We are completing, hopefully in the very, really few weeks, a family study in which we enrolled family members of vaccinated family members of confirmed cases. We took at the beginning of the study serology, neutralizing antibodies, and for some of them, cell immunity tests, and PCR at the beginning and PCR at the end. And the purpose of that study, the goal, is to try to see if there is some protection level. What is the correlative protection? We don't have the data yet, I can say that we are seeing breakthrough infections even when we have hundreds of a titer of 100, to 30, 300, 400, 500, and people got infected. So we are completing the data now, and hopefully that will be available soon. Because for some people, it will be really important to know what is that correlates. So that was the second question. The first question, and maybe I won't remember the middle one, but the first question was about the policy of testing. So in Israel, the testing policy was after the vaccination campaign is that if everyone who travels abroad, when they come back, they need to be tested when they enter Israel. So that's everyone, vaccinated and not vaccinated. So in that population, which is not representative of all Israel, obviously, it's a very unique population, but many people in Israel travel. We have everyone. For the rest, the recommendations is to be tested when you are a contact of a confirmed case. You have to be tested. Again, it's not really mandatory. There's no mandatory except for travel abroad testing, but it's highly recommended to be tested when you are a contact of a confirmed case. And if you are tested, it shortens your isolation period. So without testing, you need to be in quarantine for 14 days, and you can shorten this to 7 days if you test at the beginning when you just learned about the contact being a contact, and on day 7, and if both tests are negative, you go out of quarantine. So that is the main reasons where people would be tested if they're asymptomatic. Another specific population is the long-term care facility workers, where we do constant testing every week. And for that group of employees, we're doing this for everyone, vaccinated and not vaccinated. So what we saw is really a decrease in positive case. Especially what we can compare really nicely is when we are testing everyone in that population. So, for example, the testing when you come back from abroad, or the testing among the long-term care facility employees, you can see the drop in cases, in confirmed cases with the booster dose. So we had, before we implemented the booster dose coming from abroad, we had hundreds, up to 200 a day cases, confirmed cases coming back, and either being tested positive at the airport or in the 7 days following their return. And this has dropped significantly with the booster dose. I saw Ron raising his hand.
So maybe just to add a sentence on the answer regarding the issue of testing. So I think in that respect, very informative is the alternative control group, where you're looking at the same people, but days 3 to 7 versus days 12 and onward, because that means it's the same people. And I would say that, if they tend to do less tests just after the booster for some reason or another, that would just give you an underestimate. Okay, so together I think that's a very good way to think about this, because now it's the same people that you're comparing in terms of the tendency to go and be tested.
Okay, thank you.
And if you can remind me the second question, I'll try to answer.
Let's move on so we can get some other people. We have a lot of hands raised in a limited period of time. I'm sure we'll get back to the same topics. Dr. Levy. One part question only, please, from now on.
Okay. Thank you to the team in Israel at the Ministry of Health. This was a very informative and important presentation. We need to be mindful, of course, that Israel is a very different country and population than the U.S., and that we're talking about a vaccine that's different from the vaccine we're considering today, but nevertheless, it is a similar mRNA platform, so there is relevance there. I had a question for Dr. Milo regarding his graph depicting the fold reduction in rates of COVID by age. The alternative control group was selected at, I forget, day 3 or so, but why not at day 0? And I didn't understand why the day 0 group already had a 5-fold reduction in risk. The data are very convincing in general, but that aspect I didn't understand. And the question to Elroy, Dr. Elroy, is simply regarding the myocarditis. If I understand correctly, there is some additional risk after the 3rd dose, the booster dose, but the rate of risk isn't higher than the 2nd dose, if I understood that correctly. Thank you.
So let's answer.
So thanks for pointing that out, and we also explain about it in detail in the New England Journal paper and in the Med Archive. But just briefly, what we observed is that on day 0 and day 1, meaning just after you took the booster, it is very rare to also do a test on that day. There's like a behavioral effect that people, just as they're taking the booster, they usually don't go and perform a test as well. And therefore you get an artifactual protection that is just a seeming protection. And we observe that, and you know, we have it in the supplementary material, exactly the numbers, etc. The reasons.
Thank you. And
Dr. Price, the myocarditis?
As for the myocarditis, we've shown that myocarditis could be an adverse event following Pfizer vaccine. So we're not trying to say that after the 3rd dose it's not connected. I'm sure it's connected, but the rate of it is really really low compared to what you would have expected if it was the same rate as after the 2nd dose. And perhaps it's because we're giving this dose 5 months or more later, and so it doesn't have the same response as giving 1 dose and then after 3 weeks the 2nd dose. And in our work group, what we saw in Israel is that most cases were in a few days, 3 to 5 days after the 2nd dose among the younger males. And so maybe this, the fact that we're giving it months after is causing this rate to be actually lower.
Thank you.
Thank you. Dr. Hildreth.
I just got reminded that the second question was interesting. It was about what would have happened if there wouldn't be a booster. And I would just mention in brief that our modeling analysis shows that the number of hospitalizations, severe cases, etc., would have continued to rise very significantly according to all the details that we have. We didn't get very deeply into it, but I just wanted to mention it briefly.
Thank you. Dr. Hildreth.
Good morning. Thank you, Dr. Monto, and thank you, Dr. Elroy Price and Dr. Miller for presenting the compelling data from Israel. The most interesting part of your presentation for me was the fact that the cases began to drop among the unvaccinated once you achieved a large percentage of the population getting the 3rd dose. So do you think that you've achieved herd immunity by getting so many of the people there boosted with a 3rd shot? And part of my question is, what % of those unvaccinated individuals had COVID-19 and recovered? So could natural immunity be contributing to that group as well? Thank you.
I'm sure that people who have been infected with COVID-19 and recovered, and we don't even know about them, like the silent recovered individuals are there. When we're doing serology testing, we find among kids, we find between 5 to 12% of the kids that did not know that they had COVID-19 were tested positive by serology, depending on in the sector. So we do know that there is this population of people who have been infected and don't know that, and they are definitely contributing to the population of protected that leads us to herd immunity. For me, it's hard to say if we're in herd immunity place at the moment. It's easier to say it after, when you look back in retrospect. So when I look back in retrospect on our 3rd wave, we see that we got to herd immunity with the Alpha variant when we still had about 1/3 of our population not vaccinated, mainly kids, and still the wave went down. And this, for me, is the answer of like the perfect depiction of herd immunity, that you still have 1/3 of your population not protected. And if I go beyond my way and say some of them are probably protected and we didn't know about that, 20% of the population is not protected, and still the wave is coming down. So we're starting to see this trend now. I'm hoping we're in herd immunity now for the Delta strain, but I'm not sure we know it yet, but it seems like it's going in that direction.
Thank you. Thank you very much.
Thank you. Dr. Kurilla.
Not my camera. Oh.
Thank you, Arnold. What I'm curious about is obviously the rationale for the booster, because at least with regard to breakthrough infections, is the rather brisk antibody decay rate for neutralizing titer. And I'm wondering if you have any evidence that the third boost, the third dose, the boost that you provided, which some people have suggested may actually serve as a true boost in a prime boost strategy, is that actually impacting the antibody decay rate, or do you have any evidence that the antibody drop-off in neutralization titers is the same after the third dose as it is after the second, and you'll be back in another 6 months needing to boost again? The other aspect of this is, do you know whether or not people who have suffered a breakthrough infection, do they need to be boosted?
I'll give Ron answer the second one because there was a lot of work done and maybe it would be worth showing them. But there was a lot of work done about the recurrent infection among recovered individuals and what is their risk. But I think what we're seeing in serology is that when you give a third boost, the booster dose, you see a rapid increase in the serology, in the titers, and to some extent it's even higher in some studies than the first kind of highest level that people got from the second dose. I think what you asked is the million-dollar question that unfortunately I don't have the answer to. We're hoping that it's not a setting of every 6 months we need to be vaccinated. We know from other diseases that sometimes you need, in the protocol, 2 doses a month apart, and then after 6 months, another booster dose, and you're protected for years. I'm not yet sure what will be the answer here, but we'll definitely look carefully into that and hopefully see the decline or identify the decline earlier this time than we identified in the third wave.
Regarding your other question, I would just say briefly regarding your other question is that we're doing the analysis around the clock about the recovered. And when we're talking about breakthrough, meaning that they had at least 1 dose and then also got infected and then recovered, we see that they have a very good protection overall if they have this combination of being recovered and a single dose, similar to what they have if you have versus people who have a booster. And we hope to wrap all of this up and put it on the Med Archive as soon as possible.
Thank you.
Dr. Pergin, please.
Thanks, Dr. Monto. Just a question about the pediatric population. Since you present all, adult data 16 and older, I'm curious about what patterns you saw within the pediatric population with the booster and declining rates of disease within those under 16. And did you look at timing of when that occurred based on the ages of when boosters were given to the adult population? Thinking, of course, as parents and children in those typical age ranges. Did you see shifts in those declines during the periods when those were given?
So let me say the following. It's a bit complicated in the sense that we had our school year open in parallel in a not very different timing when the booster shots were administered. And therefore, it's not easy to disentangle what happened in the pediatrics in terms of the indirect effect of the protection that they got from the decreasing, say, from the booster to the parents, and the fact that now they started to meet in classes. So it's a complicated picture, but what I could say for certain is that we see that the overall incidence in Israel, as I said, declined about 4 to 5-fold and continues to decline twofold every 10 days. So it seems like this is also pertaining to the younger age groups, the pediatric age groups, that they also see a reproduction number lower than 1 right now.
Thank you. Dr. Nelson.
Thank you. Thank you, Dr. Monto and Dr. Alroy and Dr. Milo. Congratulations on your active surveillance for myocarditis and pericarditis, certainly a topic that will impact our deliberations today. The value of active surveillance was seen as we rolled out the smallpox vaccine to a large number of vaccine-naive individuals here in the U.S. I wonder if you would expand a little bit on your surveillance itself and the ability specifically to detect pre-hospitalization myopericarditis and pericarditis, as well as perhaps subclinical myocarditis and pericarditis. The outcomes of those individuals with less severe disease as well as those with severe disease on a long-term basis still is not yet settled upon. And I'm very interested in what case definitions you use for myocarditis and pericarditis as you call your hospital and your ability to detect these milder forms.
I hope you can hear me because I lost the connection, but I'm still online with you.
I can.
We hear you.
So, great. So we use a definition that is common based on suspected probable cases. It's in our New England Journal of Medicine publication. We have there 2 ways of defining criteria. And so we're classifying. We have a group of cardiologists and a rheumatologist who are defining each and every case based on pain, troponin level, EKG changes, echo findings, MRI findings, or biopsy findings. And so the combination of those 4 categories would lead to someone being defined as probable suspected case, and most cases are probable in our group. What we're doing is all healthcare providers know about the active surveillance that we have for hospitals, which is where we would assume the severe cases would go into. In Israel, myocarditis is a diagnosis that is recommended to be sent to the hospital, sent for hospitalization for observation. So for the most part, if not all, cases should be in our hospitals, and we have communications with all hospitals in Israel and getting their results of hospitalization each and every week for myocarditis cases. We also have IDF, Israeli Defense Force, our Army cases that we reach out to them and make sure that we're not missing that young group that might develop myocarditis as well. And so the data from Israel is actually has the cases from the Army as well in the total representation of myocarditis cases.
Thank you. Dr. Chatterjee.
Yes, thank you very much for your presentations and the answers to the questions so far. My question is around what impact behaviors in terms of the mitigation measures might have had on the epi curves that you showed. In other words, were there mask mandates? Were there other mitigation measures, and was there a way to evaluate those and their impact? Because we know even pre-vaccination, we had these waves, and as the cases would start to go up in the community, people would sort of self-quarantine or not be going out into big groups, and there was increased mask use, and that definitely had an impact on curbing some of those earlier waves. So are you able to disentangle that, the behavioral aspects, with the impact of the booster dose?
So there are mask mandates for since the beginning of our 4th wave. we re-implemented the mask mandates. And except for the mask mandates, we start using a green pass, which means you need to go into certain places using your a green pass that shows you're either vaccinated or recovered individual or have a negative corona test. I have to say that there was no correlation between implementing mitigation steps and the decline in confirmed cases. So we started using the green pass at least a month and a half before we started to show a decline. We would expect numbers to go down if that mitigation step would have worked efficiently. We would have seen a drop in the reproductive number about 2 weeks after. And so I have to say that at the same time, we started implementing booster doses. Shortly thereafter, we implemented booster doses. We didn't see a huge decline with the implementation of the booster dose. But there was some mitigation steps that we took. There was no curfew that was put in place, and until we started the booster regimen, we saw an exponential rise in cases. Now I remember the second question from before, whether this was some normal decline. I think Ron answered this along the way, whether this was a normal decline in the pandemic wave. We don't think so. This was an exponential rise that continued to go up and up and up. 50 to 60% of those infected in the fourth wave were actually doubly vaccinated, that the effectiveness of their vaccine went down to 40%, and so they were part of this wave, some of them getting severely ill and dying. And so there is no question in my mind that the break of the curve now was due to the booster dose. There was nothing implemented at that time, at that period of time that got the curve to break.
Thank you.
And I think, Ron, I don't know if there is a way for us to bring back slides.
Let's move on to a final question from Dr. Meisner, and then we're done with this session. We can't hear you.
Thank you, Dr. Monteau, and thank you for the presentations. The data that's come from Israel has been so interesting and I think helpful for other countries, particularly thinking about the two articles in the most recent New England Journal regarding myocarditis have been very, very interesting. The question I have is this. In Israel, you've used just the Pfizer, I believe, and not the Moderna vaccine. So one question is, how applicable, how would you have had a similar result, do you think, if you had used the Moderna vaccine instead of the Pfizer vaccine? Because that's really the question we're thinking about today. And so along those lines, was there any attempt to measure cellular immunity? You showed us a lot of data about antibodies. Can we get do you have any sense of the role of cellular immunity and waning immunity? Thank you.
So for the cellular immunity, we have this research that we hope to finalize shortly. And in that group of family members of confirmed cases, we will have data on cell immunity. We don't have it on a national level. The data that we showed here, or that Ron showed here, is public health surveillance data, is not connected to serology because we're not doing serology testing for all of the citizens. We do have a lot of research work from Israel by different groups showing the decline in serology and the effect of the booster dose. We are trying to get the data on cell immunity as well, hopefully finalized soon, so we'll have that answer.
And I'm going to park the question about how relevant.
And the question about Moderna. So
yeah, I'm going to park that question to later discussion because that's going to be something that we're going to have to discuss in terms of data that we get from about the way the Moderna vaccine has behaved elsewhere. Okay, so now we've got a break. I've eaten into the time for the break a bit, so we are going to come back in approximately 10 minutes.
All right,
that will be at 10:45.
All right, I'll put our timer up here for you.
right. Still good morning, or depending upon where you are in the country or the world. But welcome back to the FDA Center for Biologics Evaluation and Research meeting. This is the 169th VRPAC meeting. We just had a quick break, and now I'd like to get it back to our chair, Dr. Monto. Dr. Monto, you ready?
I am, and it's my pleasure to introduce the sponsor presentation from Moderna, going to be given by Dr. Jacqueline Miller, ID Therapeutics Area Head. Dr. Miller.
Yes, thank you, Dr. Monto, and good morning. My name is Jacqueline Miller, as Dr. Monto just said, and I am the Therapeutic Area Head for Infectious Diseases at Moderna. Thank you to the FDA and the VRPAC for the opportunity to present our safety and immunogenicity data for a 50 microgram booster dose of mRNA 1273, our COVID-19 vaccine, and thank you for everything you're doing to help fight the pandemic. Moderna has submitted a data package to the FDA for supporting use of a 50 microgram booster dose of mRNA 1273 for individuals 18 years of age and older, in alignment with recent FDA and CDC recommendations. We're emergency use authorization for all individuals 65 years of age and older, and individuals age 18 to 64 years at high risk of severe COVID-19 or with frequent institutional or occupational exposure to SARS-CoV-2. This is aligned with the recommendation evaluated a few weeks ago. How does the 50 microgram booster dose fit into the mRNA 1273 vaccination schedule? The first two doses are administered as a 100 microgram dose separated by 1 month. This was the emergency use authorization granted last December, and for which Moderna has filed a BLA, which is currently under review. Today, we're seeking your endorsement for a 50 microgram booster dose for the individuals I just described. A second schedule is depicted on the bottom row for significantly immunocompromised individuals who do not always develop neutralizing antibody titers after 2 doses. A third 100 microgram dose administered at least 1 month after the second dose is needed to complete the primary series. This indication already has emergency use authorization and is not the focus of today's presentation. This slide outlines the agenda for my presentation. I will start with why booster doses are needed. This rationale is supported by the ongoing vaccine efficacy analysis in the pivotal phase 3 study 301, long-term evaluation of antibody persistence, and observations of breakthrough disease observed in vaccinated individuals, which occurred in July and August of this year. And I'll then present data from study 201B, which evaluated the 50 microgram booster dose, including the rationale for dose selection, study design, the safety profile, and immunogenicity data against both the original virus and the Delta variant. So let's begin with a recap of the phase 3 data and the use of mRNA 1273 since the EUA. When we met last year, I presented the primary analysis results from the phase 3 study 301, the pivotal safety, efficacy, and immunogenicity study. The study enrolled 30,375 subjects who were randomized 1-to-1 to receive the vaccine or saline placebo. A 2-dose primary series of mRNA 1273 was observed to have an acceptable safety profile and a vaccine efficacy of 94.1% after 9 weeks of median follow-up time. Based on these data, the emergency use authorization was granted on December 18, 2020. Since that time, more than 190 million doses of mRNA 1273 have been distributed in the U.S., with nearly 70 million Americans being fully vaccinated. Additionally, according to the CDC, nearly 1.5 million Americans have received a third 100 microgram dose. Now I'd like to update the committee on additional longer-term data from our Phase 3 Study 301. After Study 301 participants were unblinded, those randomized to the placebo group were offered the opportunity to receive mRNA-1273. We then continued to follow all subjects for signs of COVID-19 through weekly e-diary contacts and monthly phone calls. If a subject reported disease symptoms, the site conducted a physical examination and PCR testing. At the end of the blinded phase of the study, an updated efficacy analysis was performed, and this was the basis of Moderna's BLA submission. This slide shows the Kaplan-Meier curve for COVID-19 disease occurring at least 14 days after dose 2. This is after a median of 5.3 months of follow-up, and vaccine efficacy remained high and durable at 93.2% in the per-protocol cohort. And then this is the Kaplan-Meier curve for severe COVID-19 disease, where vaccine efficacy also remained high at 98.2%. So during this period of time, through the end of March 2021, primary SARS-CoV-2 strains detected in the study were the original virus with the D614G mutation and the alpha variant. However, while the team was preparing the BLA submission, the Delta variant had emerged as a variant of concern in the United States. So the team conducted an exploratory analysis in subjects who previously received 100 microgram doses of mRNA-1273 in the Phase 2 Safety and Immunogenicity Study 201. These were 20 subjects boosted with 50 micrograms of mRNA-1273, and neutralizing antibodies were measured against the original virus as well as the Beta, Gamma, and Delta variants. Immunogenicity was first evaluated 1 month post-dose 2 with a research neutralization assay. In this graph, the bars represent neutralizing antibody titers to the various strains, and the circles represent the individual subject titers. The dotted line represents the limit of quantification of the assay. Subjects above the dotted line have antibody which can be reliably quantified, while subjects below the dotted line do not. All subjects evaluated at 1 month post-dose 2 had neutralizing antibodies against the original virus. And most also had antibodies against the Beta and Gamma variants, although at lower titers. 6 to 8 months later, see that antibody titers have waned. Nonetheless, all but one subject retained quantifiable neutralizing antibody titers against the original virus. In contrast, approximately half of the subjects had lost neutralizing antibodies to the Beta, Gamma, and Delta variants. And now, as seen on the right, 14 days after the 50 microgram boost, all subjects had neutralizing antibodies restored to the original virus, as well as to the 3 variants of concern, including Delta. The increases from pre-booster to post-booster ranged from 23-fold for the original strain to 44-fold for Gamma, and in particular, neutralizing antibody titers to Delta increased by 42-fold. This was the proof of concept that a fractional booster dose could restore neutralizing antibody even to variants not contained within the vaccine. And so, as we were learning more about the variants of concern, the Delta variant became the dominant circulating strain in the U.S. And we continued to follow the subjects enrolled in the Phase 3 Study 301 for breakthrough COVID disease. In the slides that follow, you will see the incidence rates in the subjects who were originally randomized to receive mRNA-1273 compared to those originally randomized to receive placebo. For brevity, I will refer to these groups as the early group and the later group, respectively, reflecting the time frame of their mRNA-1273 vaccination. Now this slide illustrates the time frames in which the early and later groups were vaccinated. We performed an updated analysis of COVID-19 incidence rates in August of this year because we had observed an increase in the number of breakthrough cases of COVID-19 in the study population during July and August of 2021. Prior to July, the maximum number of cases reported in mRNA 1273 recipients in a single month was 23. This increased to 81 cases in July and 169 cases in August, with 97% of these cases due to the Delta variant. At the time of this analysis, subjects in the early group had a median of 13 months of follow-up after their first dose, while the later group had only 8 months. And this enabled us to compare incidence rates in subjects who were vaccinated earlier versus subjects vaccinated later. So this is the comparison of incidence rates of COVID-19 in the July to August timeframe. And in light blue, you see the incidence rate in the early group, which was 77 per 1,000 person years, as compared to the later group in dark blue, which was 49 per 1,000 person years. Therefore, we observed a 36.4% decrease in the incidence of cases in those who were vaccinated more recently as compared to those who were vaccinated at an earlier time. Similar trends are seen when the data are stratified by age. In the younger cohort, 18 to 64 years of age, there was an observed reduction in rates of approximately 40%, and the reduction was lower in people over 65 at approximately 17%. But incidence rates overall were therefore higher in the group vaccinated earlier. And these findings are consistent with the waning antibodies I previously showed, particularly to the variants of concern. And they're also consistent with the findings of several real-world evidence studies, which have documented reduced vaccine effectiveness to the Delta variant. One way to increase antibody titers to the Delta variant could be to administer a booster dose of mRNA 1273. As part of the phase 2 development program, we had evaluated the safety and immunogenicity of a 50 microgram booster dose in the subjects who were originally received active vaccine in study 201. These data support our 50 microgram booster dose application. We chose the 50 microgram dose for the booster because we believe we should vaccinate with the lowest amount of antigen needed to induce an immune response, at least equal to that in study 301, which was linked to vaccine efficacy of 93%, which was durable for a median of 6 months. This has become a successful strategy for other booster vaccines, such as Tdap, because immune memory is reactivated. Reducing the booster dose to 50 micrograms would also increase the worldwide vaccine supply of mRNA 1273. This study was an extension of the original phase 2 study 201, which investigated 50 and 100 microgram doses as a primary series. When this study was unblinded to allow crossover vaccination of placebo recipients, subjects originally randomized to the 2 mRNA 1273 groups were offered a 50 microgram dose booster at least 6 months after their primary series. A total of 344 subjects received the 50 microgram booster dose, 173 after a 50 microgram primary series, and 171 after the 100 microgram primary series, which is the authorized series currently being administered. The co-primary endpoints were evaluated on the pooled data set, which included both groups. We also analyzed the 50 microgram booster dose after the 100 microgram primary series, because this reflects the schedule that people will receive under the EUA. The 100 microgram primary series group is a subset of the pooled primary series group. This slide gives the demographic characteristics of the 100 microgram prime subgroup as well as the pooled primary series group. Demographics were similar between the subgroup and the pooled group. There were more females than males enrolled, and the mean age was 52 years. Most subjects were white and not Hispanic or Latinx. Now let's review the safety data. The total safety database for the 50 microgram booster dose is 344 subjects. In the slides that follow, I will focus primarily on the 171 subjects who received the 100 microgram primary series. And I will compare these results to the safety data from Study 301, which is important to investigate potential increases in reactogenicity. Although the data are not shown for the 50 microgram primary series group, please note that the reported rates were numerically similar between the two primary series groups. Safety data were captured similarly to Study 301. Subjects reported local and systemic adverse reactions for 7 days and unsolicited reactions for 28 days after booster vaccination. SAEs, medically attended AEs, subject deaths, and adverse events leading to discontinuation are being recorded for 6 months after booster vaccination. This slide compares the reported rates of solicited local reactions within 7 days after the 50 microgram booster in Study 201B to those reported after the second 100 microgram primary series dose in Study 301. On the left-hand side of each panel is the booster dose. On the right-hand side is the second dose of Study 301. Grade 1 events are in blue, Grade 2 are in green, and Grade 3 are in orange. The reported rates of pain, erythema, and swelling were numerically similar between the groups, with no increases in severity after the booster. Axillary swelling and tenderness was the only solicited symptom reported more frequently after the booster. As with the primary series, the majority of events are mild to moderate in severity and lasted a median of 3 days or less. Overall, the rates of local reactions were generally similar between the booster dose and dose 2 of the primary series. This slide shows the systemic solicited reactions. For all systemic reactions, reported rates after the booster dose were numerically lower than after dose 2 of the primary series of Study 301. Again, these reactions were mostly mild to moderate in severity, with a median duration of 2 days or less. So now let's review the safety data by age group in Study 201B. Here, the bars on the left represent individuals 18 to age, and on the right, those over 65. Overall, subjects in the older age group tended to report lower rates and severity of local reactions. The sole exception was Grade 3 injection site swelling, which represented one subject reporting in the over 65 age group. And now we see a similar pattern by age for the solicited systemic symptoms. Most symptoms were mild to moderate in severity, and they were reported less frequently in the older adults. Now this slide solicited adverse events in Study 201B compared to those reported in Study 301. The first column shows the group boosted after the 100 microgram primary series, and the second column is the pooled group after both doses of the primary series. The third column represents the data from Study 301. Reported rates in Study 201B were similar to those in Study 301. To date, there have been no vaccine-related SAEs or deaths in Study 201B. Overall, the observed safety profile of the 50 microgram booster dose is acceptable. So now, let's review the immunogenicity of the 50 microgram booster dose, first against the original virus. We pre-specified 2 co-primary hypotheses to demonstrate the non-inferiority of immune response against the original virus strain in Study 201B versus Study 301. The pre-specified cohort for the primary endpoints was the pooled primary series group, which included subjects who received either 50 or 100 microgram dose for their primary series. Post-booster immunogenicity was compared to post-dose 2 responses from a subset of the subjects in Study 301. The first hypothesis was based on the geometric mean ratio, or GMR, which was pre-specified to have a lower limit of the 95% confidence interval greater than 0.67 and a point estimate of 1 or greater. The second hypothesis was based on group differences in seroresponse rates, or SRR, and a pre-specified lower limit of at least minus 10%. These criteria were selected to align with FDA guidance, and immunogenicity was also evaluated against the Delta variant. Vaccine effectiveness of the 50 microgram booster was inferred by immunobridging of the pooled primary series groups, Study 301 data. This was done to ensure sufficient study power for evaluation of the statistical criteria recommended by the FDA, since we had a fixed number of subjects originally enrolled in the phase 2 study to boost and could not increase further at that time. Our briefing book presented the pooled analysis, as this was the predefined primary subset. Additional analyses were also performed on the 100 microgram primary series group, and I will also share these data in the following slides. The first co-primary immunogenicity hypothesis regarding the geometric mean ratio of neutralizing antibodies to the original virus strain was met for the pooled data set. GMR was 1.7, and the lower limit of the 95% confidence interval was 1.5. Because the 95% confidence interval excluded the value 1, we conclude that the GMTs post-booster are statistically significantly higher than the GMTs post-primary series. Now this slide shows the same analysis for the GMR evaluated in the group that received the 100 microgram primary series. The results were very similar. The GMR was 1.8 with a lower bound of 1.5, and therefore the first co-primary immunogenicity hypothesis was also met for the 100 microgram primary series group. The post-booster neutralizing antibody titers were statistically significantly higher as compared to the post-dose 2 titers in the phase 3 study 301. Our second pre-specified compared seroresponse rates, which we initially defined as a 3.3-fold rise from pre-booster titers. This definition was based on the variability characteristics of this specific neutralization assay. And using this definition, the seroresponse rate was 94% in the study 201B group, compared to 99% in the study 301 group, with a lower limit for the group difference of minus 8.8 percentage points, which exceeded minus 10. Thus, the second pre-specified endpoint against the original virus strain was met for the pooled study group using the original seroresponse definition. And this is notable, especially because the pre-booster GMTs in the 201B group were so much higher at 126 than the pre-dose 1 titers in the study 301 subjects who were seronegative at the time of enrollment. The higher pre-booster titers make it much harder to reach the same fold rise as in a seronegative subject. The FDA requested that we evaluate a different definition for seroresponse, which I will evaluate and present in the slides that follow. This panel contains the data we just reviewed. So the light blue bar represents the seroresponse rate defined by a 3.3-fold rise in study 201B and in the pooled group, and the dark blue bar represents study 301. These bars represent the same study populations using a 4-fold rise as the seroresponse definition. Because a higher fold rise is required, the seroresponse rates are lower for both groups than with the first definition. We also noted that the VRBPAC committee reviewed a third definition at the prior meeting. This last analysis is a within-subject comparison of those who achieved a 4-fold rise increase in titers from pre-dose 1 at either the post-booster time point or the post-dose 2 time point in Study 201. Using this definition, numerically higher response rates are observed after the booster dose than after the dose 2 primary series in the same subjects. And importantly, regardless of the definition used, at least 90% of the subjects in the pooled group achieved a seroresponse rate post-booster. So the FDA also asked us to evaluate a seroresponse rate definition of a 4-fold rise only in the population that received a 100 microgram primary series. And this is the inferential analysis highlighted in the FDA briefing book. In this instance, the statistical criterion was not met. Nonetheless, the seroresponse rate was 88%, despite the fact that pre-booster titers were 150, which were 15 times higher than the pre-dose 1 titers in Study 301. And it should be noted that the post-booster GMTs at 1952 were nearly twice as high as the post-dose 2 titers of 1081 in Study 301, which were associated with vaccine efficacy. Now let's further examine those subjects in Study 201B who did not achieve a 4-fold rise. In subjects who failed to achieve a 4-fold rise, pre-booster GMTs were 492, which was more than 4 times higher than subjects who met the definition, with baseline titers of 108. Subjects in both categories achieved post-booster titers well above the level of Study 301 at 1081. And therefore, subjects who did not meet the 4-fold rise definition are still deriving substantial benefit from the 50 microgram booster dose. One of the key populations proposed for booster vaccination are adults over the age of 65 because of their increased risk from severe complications of COVID-19. And therefore, we performed an analysis comparing GMTs by age group. This slide presents the pre-booster and post-booster GMTs in subjects 18 to 64 years of age, those over 65, and the overall population who received the 100 microgram primary series, so the subgroup. And again, all post-booster GMTs are above the level set at Study 301, with adults over 65 achieving an 18-fold rise. And so similarly, we performed an evaluation of seroresponse rates by age based on the 4-fold rise definition in the 100 microgram primary series group. Post-booster vaccination, 88% of younger adults and 89% of older adults achieved a 4-fold rise, indicating no reduction in the over 65 age group. We also tested the serum samples from Study 201B for neutralizing antibodies to the Delta variant, as this is currently the variant of greatest concern. This slide shows the pre- and post-booster titers against the Delta variant in subjects 18 to 64 years of age, over 65, and overall for the group that received the 100 microgram primary series. In the younger cohort, antibodies increased 16-fold after the booster dose, and they increased 22-fold in the older cohort. These data suggest that the neutralizing capacity against the Delta variant can be substantially enhanced by administration of a 50 microgram booster of mRNA 1273, which would help address the current breakthrough cases due to the highly transmissible Delta variant. So here we see the seroresponse rates to Delta variant by age group and overall in the 100 microgram primary series group. The younger age cohort had an 88% response rate, which increased to 95% in the older cohort. This analysis supports the robust immunogenicity to the Delta variant of the 50 microgram booster. Now I'd like to summarize our safety and immunogenicity data on the 50 microgram booster dose of mRNA 1273. The safety profile of the 50 microgram booster was comparable to dose 2 of the 100 microgram primary series in Study 301. Injection site pain was the most common local solicited reaction, and headache, fatigue, and myalgia were the most commonly reported systemic adverse reactions. As with the primary series, most adverse reactions were mild to moderate in severity. Axillary swelling and tenderness was the only solicited symptom reported more frequently after the booster dose and in Study 301, and all other symptoms were numerically lower post-booster. No vaccine-related SAEs or deaths were reported during the study period. So to summarize immunogenicity, the co-primary hypothesis on the GMR was met for both the pooled data set as well as the 100 microgram primary series. The pre-specified hypothesis on seroresponse rate in terms of a 3.3-fold rise on the pooled data set was met. This criterion was not met for the 4-fold rise analysis in either the pooled or 100 microgram primary series population. Nonetheless, 88% of subjects achieved a 4-fold rise. The subjects who did not meet the 4-fold rise had pre-booster antibody titers more than 4 times higher than those who did have a seroresponse. A 13-fold rise in pre-booster titers was observed to the original virus, and the Delta variant antibody titers increased by 17-fold overall. The substantial increase in neutralizing antibody titers against both strains in both the younger and the older age groups. And taken together, these data suggest that a 50 microgram booster of mRNA 1273 will result in higher antibody responses than observed after dose 2 in Study 301, in which efficacy was demonstrated at 93%. This booster has the potential to address waning antibody titers and to reduce breakthrough disease due to the highly transmissible Delta variant. And the data that I have now presented support the 50 microgram booster dose at least 6 months after completion of the primary series. The proposed use is for individuals who are 65 years of age and older, 18 to 64 years of age at high risk of severe COVID-19, and those who are at increased risk because of institutional or occupational exposure to SARS-CoV-2, aligned with the committee's previous vote. We would like to thank our collaborators at the NIH, the COVID-19 Prevention Network, BARDA, the Montefiore Laboratory at Duke University, and the investigators and site personnel. And most especially, we would like to thank the study participants. This concludes my presentation. Thank you.
Thank you, Dr. Miller. And given the fact that you have finished a bit early, we have time for a few questions from the members. And I see Dr. Pergam.
Thanks for that presentation. I appreciate Moderna's efforts in putting that together. I had a question about how the drug is going to be put together and labeled specifically for the differing booster versus the primary vaccine, particularly when addressing the different populations who are getting boosters. Since the immunosuppressed population will be getting the 100 milligrams and the rest of the population will be getting 50, how is Moderna putting that together to make it clear? Because I could see issues coming with inappropriate dosing being given to specific populations. Can you discuss how Moderna is going to be organizing that specifically?
Yes, absolutely. So the current presentation is a multi-dose vial. So healthcare providers pull a 0.5 ml dose, which is the 100 microgram dose, from a multi-dose vial to administer. That same vial can be utilized to administer a 0.25 ml dose. and that 0.25 ml dose being lower is actually consistent with some other vaccines, particularly during the H1N1 pandemic, where lower doses of a multi-dose vial were administered to some population. We recognize that this will require some education and enforcement, and so we are preparing to send a Dear Healthcare Provider letter explaining how the doses are to be administered. In addition, our fact sheet is going to contain detailed information, and we have a 24-hour call center to support healthcare providers in their administration efforts. And there are also additional resources that will be available on the Moderna website. And then finally, our team that engages with primary care physicians is going to be going out and doing additional training to make sure that people understand the differences between the two doses. I think the important emphasis is that the 50 microgram is a booster. The 100 microgram that immunocompromised subjects are receiving is really a different indication. These are subjects who, in multiple studies, did not respond as well to the second dose and really need that third dose to reliably induce neutralizing antibody titers.
Thank you. Dr. Lee.
So one question I have is you noted obviously that with the criteria for immunobridging success, which included a sero response defined by a fourfold increase in titer, was not met, and that was in the report. In your presentation, you looked at a different threshold with 3.3. Can you sort of indicate why you chose that particular level as opposed to, I mean, we see what you have for 4, but where did the 3.3 come from?
Yes, thank you. The 3.3-fold rise is actually based on the inherent variability of the assay. So the assay itself has discriminating capability, and the statistical analysis in both booster titers during the validation of that assay indicated that you could reliably discriminate between levels of titers at a 3.3 threshold. And I'll point out that there are some other vaccines, particularly the meningococcal B vaccine, that also use a different definition for fold rises, so the 5-fold rise in that case. But we, accept the feedback that the 4-fold rise is going to be applied across companies, which is why we have also calculated using the 4-fold rise.
Thank you.
Dr. Gantz. Muted. Can't hear you.
You're muted on your phone, Dr. Gantz. There you go.
Okay. Can you hear me now? Okay.
Yes.
Thank you. Thank you, Dr. Miller, for that, for you and your team putting that together for us. I had a real question about really trying to identify that 18 to 64 age group because we're trying to really parse out their susceptibility for needing a booster. So you talk a lot about, you showed the breakthrough disease within that cohort, and it's actually quite high. We didn't see any outcomes for those breakthrough disease, so hospitalizations or severe disease, which is what we're trying to parse out. You also show the geometric mean titers pre-booster. They're pretty much the same as they are for that age group as they are for the greater than 65 age group. So I'm really trying to understand what we should be thinking about in terms of that age group and whether or not we really need to think about their also waning immunity. You don't seem to parse out the age groups when you're looking at the overall wane in antibody. I think that was slide 14 that you show against all the different variants, so we don't really know that for age group. And so I'm wondering if you could parse that out a little bit more for us and talk about what those levels actually mean for that particular age group.
Yes, I am actually going to show some additional data from that breakthrough analysis. So panel B, please. I would like to show you first the cases of severe COVID-19 between the more recently vaccinated participants and the later vaccinated participants by age group. So what you see on the slide is that all subjects are on the left-hand side of the panel. In the middle are the 18 to 64 years of age, and on the right are the greater than 65 years of age. And so what you can see here is that amongst all subjects, there was a 46 percentage point difference between the earlier and later group overall, with 30.9% in the 18 to 64 group, with only 11 cases. So this is the severe cases. And then in over 65, a 64 percentage point difference. And then can we go to panel A, please, because Dr. Gantz also asked about the characteristics of severe cases and hospitalizations. And so just to show you that the severe cases comprised 7.6% of the breakthrough cases. There were 19 of them overall. Notably, 3 hospitalizations and 2 subject deaths occurred in the earlier vaccinated group. Both of those deaths occurred in males over 70 years of age. Both of them had underlying COPD and other medical complications. And then, Dr. Gantz, your second question was with respect to antibody titers by age after the primary series, and I'm going to show you the original strain. So can we put up panel B, please? This is going to be after the 50 microgram booster for the 100 microgram series. So what you see, this time I'm going to reverse it a bit, move over to the right-hand side first. You see most to the right are the GMTs from study 301, and overall the GMTs for the pooled age group. And then in the left-hand panel, you see 18 to 64 years of age and 65 years of age. The antibody persistence, can we please pull up a slide that shows the GMT ratios by age group, please? The comparison of GMT ratios was actually higher in the older age group, and the antibody persistence was higher in the younger age group. And I'm just going to wait for the slide to come up to show you. So you know what? I will show that slide at the next Q&A and provide you with those.
Right,
steps.
Which helps me move to say that the next Q&A is going to be after lunch. So we will have some additional time to ask questions of Dr. Miller. We'll move now to the FDA presentation of the data. And we're going to have 2 speakers, Ina Morgan-Morgeau and Hui-Li Wang, with Dr. Richard Forche ready in the background to answer additional questions. So let's move ahead to the FDA presentations.
Good morning. My name is Dr. Tina Mojo. I am a medical officer in the Office of Vaccines Research and Review within the Division of Vaccines and Related Products Applications at the FDA. I will present FDA's review of the effectiveness and safety data following a booster dose of Moderna COVID-19 vaccine as submitted by Moderna under an emergency use authorization amendment. I'd like to start off by acknowledging the contributions of many of my colleagues within the Center for Biologics Evaluation and Research. My presentation is a reflection of all of their contributions. So my presentation will begin with background information, followed by an overview of the booster dose and 2-dose series studies, the immunogenicity and safety results, and then I'll conclude with an overall summary. The Moderna COVID-19 vaccine, also known as mRNA 1273, has been available under emergency use authorization since December 18, 2020. It is authorized for active immunization to prevent COVID-19 due to SARS-CoV-2 in individuals 18 years of age and older. The authorized regimen is a 2-dose series administered 1 month apart with each 0.5 ml dose containing 100 micrograms of mRNA. A third 0.5 ml dose is authorized for administration at least 28 days following the second dose in individuals with certain immunocompromising conditions. Moderna has submitted an amendment to their EUA to support authorization for booster administration of Moderna COVID-19 vaccine at 50 micrograms, 0.25 ml dose, at least 6 months following a 2-dose series to the following populations: individuals 65 years of age and older, individuals 18 through 64 years of age at high risk of severe COVID-19, and individuals 18 through 64 years of age whose frequent institutional or occupational exposure to SARS-CoV-2 puts them at high risk of serious complications of COVID-19, including severe COVID-19. Regulatory background for this submission dates back to May 28, 2020, with initiation of Phase 2 study P201 Part A, which I'll refer to as P201A, evaluating 2 dose levels of the 2-dose series of mRNA 1273. On July 27, 2020, the Phase 3 randomized placebo-controlled safety and efficacy study P301 was initiated. On December 18, 2020, FDA issued an EUA for a 2-dose series of Moderna COVID-19 vaccine in individuals 18 years of age and older. On January 28, 2021, the booster dose phase of study P201, which I'll refer to as P201B, was initiated. And on August 12, 2021, the Moderna COVID-19 vaccine EUA was reissued to include a 3rd dose for immunocompromised individuals 18 years of age and older. So the next part of my presentation will provide an overview of the design of the booster dose and 2-dose series. So study P301 is an ongoing, randomized, observer-blinded, placebo-controlled study conducted in over 30,000 participants 18 years of age and older. Participants were stratified by both age and risk for progression to severe COVID-19 into one of 3 groups, shown on this slide here, and randomized 1-to-1 to receive 2 injections 28 days apart, either mRNA 1273 at 100 micrograms or a placebo control. Data from study P301 supported the EUA for the 2-dose series of mRNA 1273 at the 100 microgram dose in adults 18 years of age and older. The 15,184 recipients of the 100 microgram mRNA 2-dose series were used as a comparator group for overall safety following the booster dose, and the 1,080 participants who were randomly selected as an immunogenicity cohort in P301 were used as a comparator group in booster dose immunogenicity analyses. In the context of this EUA submission, study P201 is an ongoing 2-part study. Part A is the observer-blinded, randomized, placebo-controlled 2-dose series phase, and Part B is the open-label booster dose phase of the study. Part A was conducted in a total of 600 participants without pre-existing conditions that would place them at risk of severe COVID-19. Participants were stratified by age into 2 cohorts and randomized according to a 1-to-1-to-1 ratio to receive 2 injections 28 days apart of mRNA 1273 at either a 50 microgram dose or a 100 microgram dose or a placebo control. At the conclusion of Part A, all participants were offered a 50 microgram booster dose at least 6 months after completion of the 2-dose series during the booster phase of the study. Of the participants who completed Part A, 344 agreed to and actually received an open-label booster dose in Part B of the study. This included 173 participants in the 50 microgram primed group and 171 participants in the 100 microgram group. Only the 171 booster dose participants primed with the 100 microgram series, shown in bolded text on the slide, contributed to our analyses of the immunogenicity analyses. In addition, these participants contributed the main safety data for the booster dose safety analyses. The median interval between completion of the 100 microgram two-dose series and the booster dose was approximately 7.2 months, ranging between 5.9 and 8.6 months. Booster dose effectiveness is being inferred by immunobridging analyses comparing two immunogenicity endpoints, geometric mean neutralizing antibody titers, or GMTs, and seroresponse rate against a pseudovirus expressing the SARS-CoV-2 spike protein from a USA Washington 1 2020 isolate carrying the D614G mutation, which I'll refer to from this point forward as the D614G strain. Immunogenicity analyses compared each co-primary endpoint at 28 days after the booster dose in study P201B to the corresponding endpoint 28 days after dose 2, which would be study day 57 in the P301 random immunogenicity subset, which is the reference study population in whom vaccine efficacy was demonstrated. And just a note, neutralizing antibody titers were 50% inhibitory dose ID50 titers measured with a validated pseudovirus neutralization assay against the D614G strain by Duke University Medical Center. This slide summarizes the immunogenicity analysis of the GMT co-primary endpoint against the D614G strain. The primary analysis evaluated the ratio of GMTs after the booster dose in study P201B to the corresponding GMTs after dose 2 in study P301. The immunobridging success criteria required that for the GMT ratio, lower limit of the 95% confidence interval had to be greater than 0.67, a 1.5-fold margin, and that the point estimate of the GMT ratio had to be greater than 1.0. This slide summarizes the immunogenicity analysis of the seroresponse co-primary endpoint against the D614G strain. Seroresponse for an individual participant is defined as a 4-fold or greater rise of neutralizing antibody titers from baseline to 28 days post-vaccination against the D614G strain, where baseline titers that were less than the assay's lower limit of quantitation, or LOQ, were set to the LLOQ. For P201B booster dose recipients, baseline was defined as the titers prior to the booster dose on the day of booster vaccination. For P301 two-dose recipients, baseline was defined as prior to dose 1. For the immunobridging analysis, the percentage difference was calculated between the seroresponse rate at 28 days post-booster dose in P201 and the seroresponse rate 28 days after dose 2 in P301. The immunobridging success criterion required a lower limit of the 95% confidence interval for the difference in seroresponse rates to be greater than or equal to negative 10%. P201B statistical analysis plan also pre-specified immunobridging analyses with hypothesis testing for the B16172 or Delta variant. These analyses are not yet available because the assay for the Delta variant is not yet validated. We will, however, present descriptive analyses submitted by Moderna using an exploratory assay for the Delta variant. At this point, I'll move on to review the booster dose study results, starting with immunogenicity data. In study P201B, of the 171 participants who were administered a booster dose, 149 were included in the per-protocol set, which is the primary analysis population for immunobridging comparisons. A total of 15 participants were excluded from the full analysis set due to the lack of baseline or post-baseline immunogenicity data. An additional 7 subjects were excluded from the per-protocol set due to SARS-CoV-2 infection or a major protocol violation involving incorrect dosing at the booster dose visit. Of note, one 100 microgram prime booster dose participant who did not receive dose 2 was included in the per-protocol population, as P201B participants were not required to receive both doses of the 2-dose series to be included in the per-protocol set. In study P301, of the 1,080 participants randomly selected for inclusion in the immunogenicity subcohort, a total of 1,055 participants were included in the per-protocol set for the primary immunobridging analyses. Exclusion from the P301 per-protocol set was most commonly due to HIV infection, followed by errors in the administration of dose 2, and 1 participant with other protocol deviation. This slide presents the demographics of the per-protocol immunogenicity subset for studies P201B and P301. Compared to study P301, participants in study P201B were less racially and ethnically diverse, had a lower percentage of males, a lower median BMI, and a lower percentage of participants who were in the category of obese with the BMI 30 or greater.
Our audio feed into the public's. All right, we just want to make sure we're good.
Your microphone has been turned on.
Thank you very much. So this slide shows the results for the GMT co-primary endpoints, again for the D614G strain. And we see neutralizing antibody titers against the D614G strain at 28 days after the booster dose in P201B. That's in this column here. And 28 days after completion of the 2-dose series in P301. The GMT ratio of study P201B over P301 was 1.8 with a 95% confidence interval ranging from 1.5 to 2.1, which met the pre-specified success criteria of a lower limit of the 95% confidence interval being greater than 0.67 and the GMT ratio point estimate being greater than 1. This slide presents the results for the seroresponse co-primary endpoint for the D614G strain. The difference in seroresponse rates between the booster dose recipients in P201B and 2-dose series recipients in P301 was negative 10.5 with a lower limit of 16.7%, missing the pre-specified immunobridging success criterion of a lower limit of the 95% confidence interval greater than or equal to 10%. In post-hoc analyses, participants with lower pre-booster neutralizing antibody titers appeared to be more likely to achieve 4-fold or greater rise in titers after the booster dose compared to participants with higher pre-booster titers. For instance, P201B participants who met the 4-fold rise in titers had a baseline GMT of 109, whereas those who did not meet the 4-fold rise in titers had a baseline GMT of 2. And seroresponse rates and baseline GMTs in P201B participants by age subgroups also appear to be consistent with this observation. Participants who were 65 years of age and older had a lower baseline GMT but a higher seroresponse rate compared to participants 18 through less than 65 years of age. This slide shows the exploratory descriptive analyses of neutralizing GMTs against the Delta variant after the booster dose in study P201B among the 100 microgram prime booster dose participants and after dose 2 in study P301 participants who received 100 microgram two-dose series. These data suggest numerically higher GMTs were achieved 1 month after the booster dose, data with some caution because they are limited by the use of a non-validated assay against the Delta. Assessment of the incidence of SARS-CoV-2 infection in study P201B was an exploratory endpoint. SARS-CoV-2 infection was detected by virologic or serologic evidence at scheduled visits or for potential SARS-CoV-2 exposure and/or symptoms. Through the August 16, 2021 cutoff date, a total of 38 booster dose participants had positive tests, 20 in the 50 microgram primed group and 18 in the 100 microgram primed group. All participants who tested positive did so at pre-planned study visits. Of the 18 booster dose participants who were primed with the 100 microgram two-dose series and who tested positive, 2 occurred prior to when a maximum antibody response would have been anticipated after the booster dose, both being positive on day 8 after. The remaining 16 infections were identified at day 29 or later. Only 1 of the 18 participants was symptomatic, and no SARS-CoV-2 infections were reported as severe. Limitations of this analysis include the exploratory nature and the lack of a control group. Case definitions for COVID-19 were not pre-specified and were not provided to study sites nor used in the analyses, and information related to COVID-19 cases was not clearly collected systematically. Responding to an FDA request, Moderna performed a post-hoc analysis of protocol-specified COVID-19 cases in the ongoing P301 efficacy study, which accrued during the period between July 1st and August 27, 2021, corresponding to the Delta variant surge. The analysis compared rates of COVID-19 among participants originally randomized to mRNA-1273 and those who completed the two-dose series early in the study versus those who were originally randomized to placebo and then crossed over to mRNA-1273 and thus completed the two-dose series later in the study. Study participants who were included in the analyses were those who remained at risk for first occurrence of COVID-19 following receipt of the two-dose series. Although not independently verified by FDA, the post-hoc analyses appear to indicate that the incidence of SARS-CoV-2 during the analysis period among participants who completed the two-dose series early in the study was 77.1 cases per 1,000 person years versus 49 cases per 1,000 person years among participants who completed the two-dose series study. The median duration of follow-up was 13 months post-dose 2 among those who completed the two-dose series early in the study, and 7.9 months post-dose 2 among those who completed the two-dose series later in the study. 19 severe cases, COVID-19 cases were reported during the analysis period, 13 of which occurred among participants originally randomized to mRNA-1273, giving an incidence of 6.2 per 1,000 person years, and 6 occurred among participants originally randomized to placebo with an incidence of 3.3 per 1,000 person. Overall, 15 of these 19 severe cases occurred among participants who were 65 years of age or older and or who had a risk factor for severe COVID-19. The 4 remaining cases occurred in participants between 42 and 64 years of age and who were not at risk of severe disease. And of those four, 3 out of the 4 were originally randomized to the mRNA 1273 group. We'll now move on to review safety results. This slide shows the median length of safety follow-up after the booster dose in all P201B participants through an August 16, 2021 cutoff date. Among the 100 microgram primed booster dose participants in the middle column here, we see that the median duration of follow-up was 5.7 months, ranging from 3.1 to 6. So our review of safety results will start with the immediate reactogenicity, defined as reactions occurring within approximately 30 minutes after study injection. Results are shown for P201A and P301 participants who received a 100 microgram two-dose series of mRNA-73 and 100 microgram prime booster dose participants study P2. Overall, immediate reactions were reported by a numerically higher proportion of P201B participants at 13.2% compared to P201A participants at 5.1%, but the rate in P201B is notably similar to that in P301, which had a rate of 9.9%. A total of 22 participants in the P201 group reported any immediate adverse reaction. Of these, 1 was reported as severe, 1 case of severe injection site pain. Breaking down these reactions by local versus systemic, 10.2% of participants reported immediate local reactions, which consisted mostly of injection site pain, followed by erythema and acceleration arm, and 4.8% of participants reported immediate systemic reactions, which consisted of headache, fatigue, arthralgia, myalgia. This slide shows the rates of solicited local reactogenicity by age group within 7 days after dose 2 among the 100 microgram two-dose series recipients in P201A, and within 7 days after a booster dose following a 100 microgram two-dose series in B. The most frequent local adverse reaction reported in both age groups was injection site pain, in which this was reported by a similar proportion after the booster dose versus dose 2. Among participants 18 to less than 65 years of age, rates of axillary swelling or tenderness of the vaccination arm, which were mostly mild in severity and transient, were higher after the booster dose at 24.8% compared to dose 2, 11.6%. When comparing the rate of axillary swelling or tenderness after the booster dose to the corresponding rate after dose 2 in the larger P301 population of 18 to less than 65-year-olds, the rates were more similar, 24.8% versus 16%. In participants 65 years of age and older, there were no notable trends in the frequency of local reactogenicity after the booster dose compared to after dose 2. Rates of local reactogenicity were generally lower in participants 65 years of age and older compared to those 18 through 64. Across both age groups, severe local reactions after the booster dose were reported by 0 to 5.3%. No grade 4 solicited local reactions were reported in either group after the booster dose, in either age group. Are you still able to see my slide?
Yep, hold on a second. Somebody moved the slides here. I'll put it back on yours. Give me a second here. There you go, Dr. Mongo. There you go.
So, yeah, rates of local reactogenicity were generally lower in those 65 and older compared to, I think I was going over the severe reactions, and Overall, the median day of onset of local reactions was generally between day 1 and day 3, and the median duration of local reactions was generally no longer than 3 days in both age groups. So we'll move on to review the slide, which shows the rates of solicited systemic reactogenicity, again shown by age group within 7 days after dose 2 among those who got the 100 microgram 2-dose series in study D201A, and within 7 days after a booster dose among those who received the 100 microgram 2-dose series in PD. The most frequent systemic adverse reaction reported in both age groups was fatigue, followed by either headache or myalgia, and then arthralgia and chills. In participants 65 years of age and older, which had a relatively small denominator, the rates of myalgia and arthralgia were numerically higher after the booster dose compared to after dose 2. However, the rates of myalgia and arthralgia after the booster dose were notably similar to the corresponding rates after dose 2 in the larger P301 population 65 years of age and older. Across both age groups, severe reactions were reported by 0 to 7.9%, and there were no grade 4 reactions reported after the booster dose. Overall, the median day of onset for systemic reactions was day 2, and the duration of these reactions was generally no longer than 2 days in both. This slide provides an overview of the unsolicited adverse events and serious adverse events reported in study P201B. Through the August 16, 2021 cutoff date, there were no unsolicited adverse events that were not already captured as solicited local and systemic reactions and which were not considered causally related to Moderna COVID-19 vaccine. A total of 20 subjects, or 11.7%, reported unsolicited adverse events through 28 days after the booster dose. The most common unsolicited adverse events included headache and fatigue. One case of Bell's palsy was reported and considered unlikely to be related based on temporal implausibility. That event occurred 5 hours after booster dose. There were no serious adverse events reported within 28 days after booster vaccination. As of the August 16, 2020 cutoff date, 5 SAEs were reported in 4 participants with time to onset more than 30 days following the booster dose. That included 1 case of tendon rupture, 1 case of spontaneous abortion, 1 case involving deep vein thrombosis and pulmonary embolism, and 1 case of pericarditis. None of these SAEs were considered likely to be related to the vaccine because the timing of the events in relation to the vaccination did not suggest a causal relationship, and or a more likely alternative etiology was identified. And no participants were withdrawn due to adverse. So I will now conclude with a summary of P201 immunogenicity and safety data. In terms of immunogenicity, immunobridging analyses against the D614G strain met the pre-specified success criteria for the GMT ratio, but not for seroresponse rates. In post-hoc analyses, participants with lower pre-booster neutralizing antibody titers were more likely to achieve a 4-fold or greater rise in neutralizing antibody titers after booster vaccination, compared to participants with higher pre-booster neutralizing antibody titers. Immunogenicity data to support effectiveness of the booster dose against the Delta variant are limited to exploratory analyses using a non-validated. In terms of safety, there was no evidence of increased reactogenicity following a booster dose relative to dose 2, with the exception of axillary swelling or tenderness of the vaccination 18 to less than 65 years of age. Unsolicited adverse events did not reflect any new safety concerns, and through the August 16, 2021 cutoff date, there were no deaths or SAEs considered causally related to Moderna COVID-19. That concludes my presentation. Thank you.
Good morning. I'm Quili Wong, Associate Director for Innovation and Development, Office of Biostatistics and Epidemiology at the FDA Center for Biologics Evaluation and Research. On behalf of our multiple collaborators in the FDA BEST system, today I'll present some pre-market data. Today I'll present some preliminary results on post-market data of myocarditis and pericarditis following mRNA COVID-19 vaccination in the FDA BEST system. Information on myocarditis and pericarditis has been updated to the fact sheet for COVID-19 vaccines for Moderna. Post-marketing adverse reports have indicated or suggested risk around within 7 days following the second dose, highest in males 18 through 24 years of age. We evaluated this in the FDA Active Surveillance System, the Biologics and Effectiveness Safety System, or BEST. The FDA CBER Active Surveillance Program, through multiple partners as illustrated here on this slide, proactively monitors the safety and effectiveness of biologics, including COVID-19 vaccines. For the safety surveillance of COVID-19 vaccines, the BEST system works with, in this case, the 4 large nationwide health plans seen here in the yellow circles. So collectively, these 4 BEST medical claims databases here contains data from every state in the United States, in this case claims databases, and with covering at least an approximate around 80 million enrollees per year. For our analysis that I'll be sharing here today, that is around 21 million doses, vaccine doses. That's 12.6 million doses for Pfizer and 8.5 million doses for Moderna. In this presentation, when I state myocarditis and pericarditis, we define that as myocarditis and or pericarditis. We identify that using diagnosis codes for reimbursement, and the risk interval is 1 to 7 days after each dose. We estimated the incidence rates, and we compared incidence rates between Moderna and Pfizer. So for incidence rates, we estimated this in the Moderna and in Pfizer vaccine brand by groups, by sex, and by dose. In dose, that will be any dose, post-dose 1, post-dose 2, in post-on regression. It adjusted for age, sex, and where sample size permits, week of vaccination, history of prior COVID-19, and urban-rural status. This slide shows you the number of events seen here in the first 1 to 7 days of receipt of any dose in the FDA BEST system. As you can actually see here, that it's actually the highest in the younger age group, that's 18 to 25. It's also the highest in males, and not shown here, it's actually also the highest in the after second dose, so that'd be males 25 to 18 to 25 years of age. This slide here illustrates, graphically summarizes the incidence rates of myocarditis and pericarditis in the first 1 to 7 days of receipt of any dose of mRNA COVID-19 vaccines for the 4 databases. So the vertical axis here is by age. So the uppermost there is the youngest age group, 18 to 25. The horizontal axis here is the incidence rate, and that is per million person per day. Overall, as you can see here, you see colored dots and whiskers, and that denotes the incidence rate and the corresponding 95% confidence interval for each of the databases here. In general, we can see that the incidence rates vary across the 4 databases with wide confidence interval. As you can also see, the highest actually is in 18 to 25 years of age. And in our analysis, we saw that the highest risk is actually in 18 to 25 years, male post-dose 2. With that, one thing also to note that these events here have not yet been confirmed with medical charts, and medical chart confirmation is underway. For the highest risk for highest incidence rates of myocarditis and pericarditis. We're looking at the age group of 18 to 25 males after dose 2. Similarly, we see here that the dose here actually, for this post-dose 2, the incidence rate here vary across these 4 databases, and this actually range from 5 to 37 per million person days. We compared between Moderna and Pfizer, these incidence rates. We used a retrospective comparative cohort study design, and what we did was that we compared the post-vaccination rates in the first 1 to 7 days of each dose. We also adjusted for some of the covariates where sample size permits that we used in the incidence rates. Among the males 18 to 25, there's a total of 1.16 million mRNA vaccine doses, of which 750,000 Pfizer, 410,000 is Moderna. And for this analysis, there's a total of 68 events that we see here. As you can tell, most of the events are actually in dose 2. Although we've done analysis by dose, the conclusions are somewhat actually similar for any dose and dose 2. So in my next slide, I'll be showing you results for any dose. In this case, this will be comparing between Pfizer and Moderna, the incidence rates. This slide shows you the incidence rate ratio of myocarditis and pericarditis comparing Moderna versus Pfizer, and that would be the reference. And this is for, as mentioned, the highest group, the group that's the highest risk is male, 18 to 25 years, any dose. What you see here actually on the horizontal axis is your incidence rate ratio, and that once again is that compares between Moderna and Pfizer. The dotted line here actually denotes the rate ratio of 1 that indicates that there's no difference in risk between Pfizer and Moderna. Hence, so the incidence rate ratios on the right of this dotted line then represents a higher incidence rate ratio for Moderna, and on the left, a higher incidence rate ratio for Pfizer. As you can see here, the first top 4 incidence rate ratios estimates in our 4 databases here, that there's actually heterogeneity among those. And in all 3 of these actually, they were no, so it crosses the null, so there was no statistical significant elevated risk here. One of these here, data part number 4, there's an elevated risk, and that's based on 20 events. That is also a federated data system, which means that we'll be able to take advantage of master protocol and common data elements to combine these estimates. And this is particularly helpful for rare outcomes, for example, in myocarditis/pericarditis. So we summarize this incidence rate ratios, and this is represented in the rate dot with the whiskers here in random effects meta-analysis. Here we see that there isn't a statistically significant elevated risk. However, this could be as low as 0.56 and as high as 2.6. In summary, in our U.S. study of 4 large claims databases covering 18 million persons with 21 annually, with 21 million mRNA vaccine doses, our preliminary results have shown that incidence rate is highest in males, age 18 to 25, post-dose 2. However, as you can tell, there's a wide range of incidence rates among these 4 databases with wide confidence intervals. For incidence rate ratios, estimates this compares between the Moderna and Pfizer. The current preliminary results do not support a significant difference for males 18 to 25 years. We do want to note that these estimates have very large uncertainty. As you can tell, this is due to small numbers of observed events for this rare outcome. And we also partially we adjusted for some potential confounders, so we cannot exclude that these estimates may actually be biased. It has come to our attention and our understanding that maybe some results from other surveillance systems. As of this meeting, while in communication with some of them, we have not actually independently reviewed or verified the underlying data or their conclusions. We do want to note that our understanding is that the results that we just shared with you, it probably comes from one of the largest studies for in terms of for this very rare outcome, actually. And also the results that we just shared with you, we've taken in context of the various limitations that I've actually summarized during this result interpretation. I'd like to thank all the multiple and various collaborators who've contributed to this work and who has worked with us, the FDA BEST Coordinating Center, Acumen, and our data partners who contributed to this analysis, CVS Health, Optum, IQVIA Health Corp, Blue Health Intelligence, all of our FDA colleagues, and federal partners. This concludes my presentation. Thank you.
Thank you both very much. Presentations were very clear and helpful. We have a very few minutes now for questions. We have a much longer time after lunch for more broad questions of both the sponsor and the FDA presentations. I'd like to restrict the few minutes we have now for questions concerning the most recent presentation, the myocarditis, pericarditis presentation. So if you aren't asking about those, please lower your hands. Keep them up if you want to ask about this most recent presentation, and then we'll get back to it later. Dr. Moore. I believe this is about the myocarditis issue because the data is being presented on the 206 study is really quite complicated to me. First, I want to say thank you so much to the FDA for their analysis of the Moderna data. And I think it may be just me, but perhaps other members of the committee are confused as well. But I found that the FDA's clarification made a great deal of sense of the data that was being presented. But much of the data that was presented was on a vaccine that we have not authorized, and no one is actually receiving and will not receive a booster, and that is 2 50-milligram doses followed by a 50-milligram booster. That's not EUA approved. What is approved is 2 100-milligram doses followed by a 50-milligram dose. The reason why I say that that may be related to the myocarditis issue is because if we're looking at any serious adverse effects and we're mixing all those people together, we're going to be underestimating because there's a dose response. If there's a dose response effect on myocarditis, there's going to be less if you're mixing a lower dose vaccine that is not being used together with the remaining data. Similarly, for immunogenicity, with a lower dose vaccine, you're going to have a lower, one assumes, basal reactivity, and a boost will obviously increase the relative amount of immunogenicity compared to the vaccine that is currently being given. I just wanted, while the FDA personnel were here, I just want to know, am I confused, or did I more or less describe the data as it was presented and what is being seen? We should just be looking at the 149 people in the 206 study that had 100 milligrams of vaccine for their primary series. Yeah, could we have some clarification? Dr. Fink.
Hi. I can clarify that the primary analyses that FDA considered in its review of the Moderna submission was the cohort of study participants who were vaccinated with the two-dose series of 100 micrograms each, followed by a 50 microgram booster dose, which is what Moderna is requesting for emergency use authorization. We additionally considered safety data mainly in terms of serious adverse events for the additional cohort of subjects who received a 50 microgram two-dose series prior to a 50 microgram booster. I would mention that really the sample size is sufficient for characterizing common adverse reactions, but in order to assess for rare adverse reactions such as myocarditis, one would really need a significantly larger safety database by orders of magnitude, and that is really for post-authorization surveillance to address.
And Dr. Fink, the material that Dr. Wong presented was the authorized dose, correct?
That is correct. The material that Dr. Wong presented was from best analyses of the 100 microgram two-dose series as used in the U.S. under emergency use authorization.
Thank you. Final question before lunch break from Dr. Rubin.
Thanks for the next presentation. Just a quick question. Do you have an idea of the specificity of the diagnosis from the diagnostic codes from previous work where you've looked at diagnostic codes?
Thank you. Currently, we're doing chart review for that, but we do not have that currently right now.
Okay, we have a full 45 minutes after lunch and the public presentations to get back to all these. So note your questions and we'll take them on. We have 45 minutes for a robust discussion. So we break now for lunch and also for the open public hearing. The full meeting, other than the open public hearing, resumes at 2 p.m. Eastern.
All right. I'm going to take us to lunch, so thank you while we get set for lunch. Okay, and welcome back from our little lunch break to the 169th VRPAC meeting. Dr. Monto, looks like you're ready. Take it away. Oh, hold on a second. Somebody unmuted somebody. All right, Dr. Monto, take it away.
Okay, welcome to the open public hearing session. Please note that both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, to, at the beginning of your written or oral statement, advise the committee of any financial relationship that you may have with the sponsor, its product, and if known, its direct competitors. For example, this financial information may include the sponsor's payment of expenses in connection with your participation in this meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationship. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Over to you, Prabha.
Thank you, Dr. Monto. Can you all hear me?
Yes, we can.
Okay, thank you. Before I begin calling upon the registered speakers, I would like to add the following additional information for the record. FDA encourages participation from all public stakeholders in the decision-making process. Every advisory committee meeting includes an open public hearing session during which interested persons may present relevant information or their views. Participants during the OPH session are not FDA employees or members of this advisory committee. FDA recognizes that the speakers may present a range of viewpoints. The statements made during this open public hearing session reflect the viewpoints of the individual speakers or their organizations and are not meant to indicate agency agreement with the statements made. With that guidance, I would like to now state that each speaker has 5 minutes to make his or her remarks. The first two speakers will utilize PowerPoint slides while others simply make oral remarks. Thank you. And the first speaker is Benjamin Newton. Can we have his slides and his presentation, please?
Hi, thank you. My name is Ben Newton. I'm here to speak today about how we can save the most lives. We should approve boosters, heterologous boosters, and vaccines for children. Slide 2. What could we have done? We could have authorized tests as soon as they were developed. Instead, we sent cease and desist letters to the first people detecting community spread. We could have authorized vaccines in July of 2020 based upon safety data in phase 1 and 2 studies and animal trials. Instead, we waited months after 90% efficacy was demonstrated. We could have authorized microdoses so that 100 times as many people could have been protected at any given time. Instead, even though we knew that a 50 microgram dose of mRNA 1273 elicited the same antibody response with fewer side effects, we insisted on a 100 microgram dose, killing tens or hundreds of thousands who couldn't be vaccinated. We could have lived in an alternate universe where Delta never developed, but we chose to be precisely wrong instead of approximately correct. Slide 3. As you all know, the FDA has an animal rule. It is possible to approve vaccines without full-scale human testing of efficacy by using human safety data and animal efficacy data. We chose not to use this rule for COVID-19, which cost tens or hundreds of thousands of American lives. Slide 4. In July of 2020, animal challenge trials had already made its way to the New England Journal of Medicine. It was widely known that vaccines equaled faster viral clearance. Slide 5. In August of 2020, we saw in Nature that microdoses protected animals, so 1/100th dose would provide significant protection against severe disease. there was no risk of vaccine-enhanced respiratory disease. We could significantly decrease dosing safely for children because there was not a Goldilocks zone. Any tiny dose was better than no dose. Slide 6. We looked at the Moderna and Pfizer data from their original EUA filings and saw 90% efficacy 14 days after the first dose. Once the DSMB had this data, they likely contacted the FDA to ask for a pause of the trial, and the FDA said no. How many additional people died because of that single decision? Slide 7. When was 90% efficacy reached? About August of 2020, you can see from trial enrollment. To know for sure, you would have to FOIA the underlying data, though the FDA refused my request for the data. Slide 8. Merck developed an antiviral drug, and the FDA paused the trial once 50% efficacy was reached. Vaccines reached 50% efficacy in phase 1 or 2 trials by matching participants to the general population. For an endemic respiratory disease, there is a 100% chance of catching it, which means that the standards for treatment and vaccine approval should be identical. Slide 9. Adenovirus vaccines require heterologous boosting. Other regulators knew this and encouraged heterologous boosting months ago, even for heads of state. Slide 10. Since April, we have had a very helpful rubric. Once you know the amount of antibody increase from a boost, you can accurately predict the change in efficacy. Slide 11. Chinese regulators on June 4th approved vaccines for children aged 3 and older. The American Academy of Pediatrics on August 5th recommended that we approve pediatric vaccines based upon serobridging data. Pfizer on September 20th released data suggesting vaccines for children were safe and effective. DSMBs have already seen everything necessary to approve children's vaccines. Just because we have not seen the data doesn't mean the data doesn't exist. Pulling less hard on a syringe does not require anything complex from an approval standpoint. Slide 12. Everyone here went into medicine to save lives, but today we are killing people, not by our actions, but by our inactions. If you withhold care from someone who needs it, that is no different than providing bad care. We falsely believe that it is safe to wait when waiting kills and maims thousands of people each day. Is there any potential that vaccines could be more dangerous than COVID? No. In fact, the most significant risk associated with vaccination is not even acknowledged by the FDA. It is the risk of driving your car to get vaccinated. Today, the FDA is preventing J&J recipients from receiving heterologous boosts. The people who took that vaccine acted in good faith and took whatever was available when we all knew that the Moderna vaccine was the best one from phase 1 data alone. The FDA is preventing many Moderna and Pfizer recipients from receiving boosts, and the FDA is preventing children from being vaccinated. We are failing to protect those too weak to protect themselves. Today, a child died because the FDA prevented her from being vaccinated. One father, just like me or you, lost his daughter because he wanted to send her to school. I thank you for your time.
Thank you. The next speaker is James Rios.
Hi, my name is James Rios. I have no financial relationships to disclose. I'm pursuing my master's.
Go ahead, please.
I'm pursuing my master's in economics at Florida International University, and I'm currently in the midst of an internship with the Vaccine Considerations Project. While the Vaccine Considerations Project has encouraged and supported me in applying for and preparing for this presentation opportunity, all of the assessments and recommendations I'll be sharing are my own and may be different from the neutral stance of the Vaccine Considerations Project. All the peer-reviewed research papers and other reference materials that I used to create this presentation are available live on vaccineconsiderations.com right now. If you have the ability, I encourage you to follow along on vaccineconsiderations.com right now. Next slide, please. My intention is to open a discussion that will address the need to increase trust in new vaccines. States across the country are encountering hesitancy and resistance to getting vaccinated among their populace. The overall success of new vaccines will rely on more than the public fully accepting these vaccines into their everyday lives. It is critical that the FDA and these new vaccine producers create communication guidelines in order to identify, clarify, and explain potential risks. Here are a few of the suggestions from the 2019 CDRH Communication Guides. 1. Further expand the reach of communications. 2. Clarify the FDA's role. 3. Constant outreach and accurate information to promote understanding, trust, and adaptation. I encourage a full mechanism to be developed moving forward. Focusing on increasing trust and credibility through mechanisms and systems that produce consistent and scientifically accurate information regarding the vaccine will reduce uncertainty regarding the vaccine. This will hold long-term implications as people learn to trust the information they consume through these systems. Next slide, please. Even before the pandemic, it was common for patients to seek information about health conditions and treatment options from health-related sites and sources of information on the internet. As the pandemic began to spread, individuals once again turned to the internet for information. According to many experts, including Dr. Akpan, the effect of the lagging responses by government and public health agencies to prioritize the dissemination of information about the coronavirus outbreak drove many back to the sources they were familiar with. The vacuum in the supply of information regarding COVID-19 was then filled by popular media producers on social network platforms, news platforms, websites, and blogs, with unsubstantiated, incorrect data or misinformation. To understand consumer perspectives, recent studies have employed an infodemiology approach or method which is designed to measure and track health information demand and supply by analyzing search queries or social network communications. Other studies have focused directly on patient education and intervention or internet technology application. The overarching conclusion in these studies is the individual is becoming a savvy patient consumer. A savvy consumer is a consumer who is media literate, knowledgeable about marketing and targeting, as well as cynical about advertising and can see through the traditional sales pitch. Next slide, please. In trying times, some have come to expect extreme solutions as the only methods for progress. However, I do not believe we're at such a point. This committee and others like it are charged with putting the patient consumer first above all else. I implore you to continue to do so by making it a priority to build trust and credibility parallel to addressing efficacy and safety concerns. Increasing levels of trust and credibility should become an iterative process at every level through business development, regulatory approval, and finally communications with the patient consumer. This is why I encourage the committee to continue these events and increase its focus on the mechanisms and systems most efficient at taking on the tremendous task of organizing consistent and scientifically accurate information regarding the vaccine. Taking these steps now could prevent future hesitancy with new medical technology as patient consumers begin to trust these reputable sources. Next slide, please. As a reminder, all the peer-reviewed research papers and other reference material that I used to create this presentation are available live on vaccineconsiderations.com right now. I encourage you to dig deeper. Thank you to the committee, and thank you all for your time.
Thank you, Mr. Rios. The next speaker is Karen Azarian.
Hello, my name is Karen Azarian. I have no financial relationships with the sponsor, its products, or any competitors. The committee's decision whether to recommend a Moderna booster for each of the three populations in your question today will require making a single risk-benefit assessment for different groups of people within each population. One of those groups is the community of people who have intellectual and/or developmental disabilities. I'd like to highlight the high risk of severe COVID, the high exposure, low vaccination rates, and current lack of requirements for vaccines and rapid testing among people with IDD and the people who support them as factors that should be considered when weighing the risks and benefits of a booster. I respectfully ask the committee to consider the public health impact of your decisions specifically for people with IDD. And if you decide not to recommend emergency authorization for the broader populations at this time, to recommend it for people with IDD who received the Moderna vaccine more than 6 months ago, and for those who support them. People with IDD are at high risk of being infected with and dying from COVID. and are often included in high-risk groups as a result. As Jonathan Gleason and others wrote in the New England Journal of Medicine Catalyst, March 5th, 2021, a cross-sectional study of over 64 million patients across 547 healthcare organizations, quote, reveals that having an intellectual disability was the strongest independent risk factor for presenting with a COVID-19 diagnosis and the strongest independent risk factor other than age for COVID-19 mortality. A person with IDD who's been fully vaccinated and who lives in a certified group home in New York, for example, is supported by staff who have a statewide vaccination rate with at least one dose of 36.3%. They may attend a day program where the staff have a vaccination rate of 34.4% and where they interact with peers who have a vaccination rate of 47.7%. There are currently no vaccine or rapid testing requirements that apply to either staff or individuals with IDD in New York, other than in state-run homes. All figures are from New York's Office of People with Developmental Disabilities as of September 10, 2021. As you decide whether to wait for more data or how to balance competing public health interests, I ask you to consider that in New York, the case fatality rate for people with IDD for COVID is 7.7%. Even mild cases can have a disproportionate impact on the system of supports. And as the pandemic takes its course, a person who has IDD often can't avoid exposure or maintain social distancing in their home. Many of the people with IDD in New York who completed the Moderna series did so in January and February, more than 8 months ago. The committee may question whether the data sufficiently demonstrates the need for or the effectiveness of a Moderna booster. Nevertheless, I ask the committee to consider the factors I outlined for people with IDD and those who support them. I believe they support recommending an emergency authorization of a booster for this committee, whether homologous or heterologous, as was done last month for people who are immunocompromised, if not for each of the broader populations. Thank you for the opportunity to speak and for your work.
Thank you, Ms. Azarian. The next speaker is Mr. Burton Eller.
Good afternoon. My name is Burton Eller, and I am the play caller from the National Grange in the advocacy arena. I'm the Director of Policy and Advocacy. The National Grange is America's oldest agricultural, rural life, and small-town citizen advocacy organization. An important factor impacting the health of rural Americans is a significant number of disparities that increase our vulnerability to certain conditions and at the same time impede our access to care and treatment. Here are a few examples. Since 2015, 181 rural hospitals have permanently closed, depriving surrounding populations with timely access to everything from emergency care to disease management and prevention. Despite recent advances, 20% of our population still cannot access high-speed broadband, which essentially eliminates their access to virtual clinical care. In comparison to urban and suburban areas, there are far fewer providers of rural health and their resources. Moreover, rural patients often have to drive significant distances to reach those that are available. The COVID-19 pandemic has brought new challenges to the importance and urgency of addressing these disparities. Throughout its existence, the National Grange and its state and local chapters have advocated for educational outreach, sound public policy, and adequate resources to protect and advance rural health. That has not changed, nor will it. Today, as the expansion of protection through boosters is being considered, we want to thank the FDA for its work and leadership. We respectfully ask that the committee keep in mind during its deliberations the access challenges that face the population we are proud to serve and the frontline healthcare workers who care for us. As we represent rural Americans across all generations, we look forward to FDA's upcoming assessment of COVID-19 vaccines for our younger children as well. We welcome the actions of FDA in this and all matters so important to our health. Thank you for the ongoing commitment to protecting Americans against COVID-19.
Thank you, Mr. Eller. The next speaker is Tyre Phillips.
Thank you. Good afternoon. My name is Thayer Phillips of Seniors Speak Out. I have no financial relationship to disclose. For the 20 years before I became eligible for Medicare and the 8 years since, I have been an advocate for the concerns of older Americans. As a military veteran, I have a special interest in all our veterans. I want to start by thanking this committee for your unending commitment to ensuring the COVID-19 vaccines are safe and effective for as broad a population of Americans as possible. From the early days of the pandemic, it was clear that the threat of COVID-19 was particularly high for people 65 years and older due to our weakened immune responses and increased likelihood of chronic conditions. Ensuring these most vulnerable members of our society had access to effective and safe vaccines to prevent the onset of serious respiratory illness was a critical first step toward slowing the spread and impact of this deadly virus. While some chose to ignore the science-based recommendations, it quickly became apparent that these vaccines were the right medicine to conquer this deadly virus. For our part, older Americans have stepped up to the plate and taken action to protect both ourselves and our families from COVID-19. Older Americans lead the country in protecting ourselves from COVID, as those 65 and older have the highest rate of vaccination among all age groups, with 89% having received at least one dose, compared with 68% for people ages 18 to 64. Now we once again look to the FDA for guidance on how to continue to take the appropriate steps to provide ourselves with the strongest protection possible against COVID-19 with the booster vaccine. We are encouraged by the current vaccine's ability to greatly reduce the risk of hospitalization and death from COVID-19 and know the lives of thousands of seniors have been saved as a result. As the science continues to evolve, we believe ensuring broad access to the booster dose will provide an added layer of protection so that we as a nation can continue to watch the rate of COVID cases decline. We know that we are not only taking this action to protect ourselves, but also to help stop the spread and impact on younger generations who are not yet eligible for the vaccines. We are grandparents, aunts, uncles, teachers, mentors, and friends who are eager to see all generations obtain protection from this virus. Just as you have worked diligently to ensure safe and effective vaccines are available to a broad population of Americans, we look forward to seeing the youngest generation have access to needed protections as well. I thank you for this opportunity to speak on this important issue.
Thank you, Mr. Phillips. The next speaker is Ms. Linda D.
Hi, good afternoon. My name is Linda D. I've been an AIDS activist for 35 years and have served as a community representative on many CEDAR Antiviral Advisory Committee hearings. I have no conflicts. I usually address scientific and regulatory issues at VRBPAC meetings. Today I intend to shine a light on Moderna's failure to provide mRNA 1273 vaccine to low and middle income countries with few exceptions. Unless we begin vaccinating the entire world in earnest, SARS-CoV-2 mutations will continue to develop. We will continue to need boosters, and the pandemic will never end. It will certainly not be over by next year. International variants have continued to plague us, including variants from the United Kingdom, Brazil, South Africa, and now the Delta variant from India, which is the most transmissible and virulent to date. If anything, international travel has steadily been increasing with no signs of decreasing. Messenger RNA vaccine technology was developed by U.S. government researchers. According to the New York Times, our government contributed $300 billion to Moderna in research and clinical trial support, and another $1.5 billion for pre-ordered, unproven vaccines. Taxpayer dollars also pay Moderna $15 to $16.50 per U.S. dose. Moderna's 2019 revenue was $60 billion. Their projected income for 2021 is $20 billion, with approximately $14 billion in profits. Moderna's market value has tripled and is now about $120 billion. Forbes lists Moderna's founders, two Moderna founders among the 400 richest people in the United States. Yet Moderna has provided its vaccine to wealthy countries to the exclusion of low and middle income countries more than any other vaccine manufacturer. Moderna has provided 8 times less vaccine than Pfizer and 25 times less than Johnson & Johnson to World Bank classified low-income countries. The few middle-income countries that do have contracts with Moderna are paying more per dose than both the United States and the European Union. The Biden administration has expressed dismay about Moderna's international vaccine allocations and has called for Moderna to produce more vaccine for international donation and to license their technology to overseas manufacturers that are able to produce vaccine for international use. Licensing their technology would be the quickest way to begin vaccinating the rest of the world, but it would also mean Moderna might lose potential profits from the development of mRNA vaccines for other diseases such as cancer and HIV. VRBPAC recommending the authorization of a 50 microgram booster dose of 1273 will also increase the availability of vaccine doses. While Pfizer has agreed to sell low-cost vaccine doses to the U.S. for overseas donation, Moderna has not. Meanwhile, only 10% of people in Africa and the Middle East have been vaccinated. Moderna has stated that low and middle income countries will receive vaccines after its commitment to developed countries have been fulfilled. Moderna has not delivered any of the 34 million vaccine doses promised to the United Nations COVAX program or the 500,000 doses promised to Botswana. Other international shipments are not slated until next year. If we're going to successfully combat COVID-19 and prevent the possibility of our current vaccines from eventually being overtaken by even more virulent variants, we must ensure that the entire world has vaccine access. It is not only the right thing to do, it is also the scientifically sound thing to do to end the pandemic by reducing continuous viral replication and possibly even reducing the necessity of continuous administration of boosters in the future. Thank you for the opportunity to comment and for your dedicated service.
Thank you, Ms. D. The next speaker is Dr. Michael Carome.
Good afternoon. I'm Dr. Michael Carome, Director of Public Citizens Health Research Group. I have no financial conflicts of interest. Public Citizens supported the initial emergency use authorization of the primary 2-dose series of the Moderna COVID-19 vaccine because clinical trial data demonstrated that the vaccine was highly effective and safe. Importantly, data from observational studies summarized by the CDC at the September 2021 meeting of VRBPAC indicated that the primary series of the Moderna COVID-19 vaccine continued to afford robust protection against severe COVID-19 disease and death in the U.S. Although there may be a role for a booster or third dose of the Moderna vaccine in certain populations, such as individuals 65 years of age or older, who are at least 6 months post-completion of the primary series, we want to highlight 3 limitations regarding the data submitted in support of Moderna's request for an EUA for such booster doses. First, the efficacy of booster doses of the vaccine against symptomatic or severe COVID-19 disease was not evaluated in the phase 2 clinical trials of the booster. Second, the subject population enrolled in the phase 2 clinical trial was not representative of the racial and ethnic diversity of the U.S. population. Specifically, with regards to race, the subject population was 95.3% white, only 2.3% black or African American, 0.9% Asian, 0.6% American Indian or Alaska Native, and 0.3% Native Hawaiian or other Pacific Islander. And with regards to ethnicity, was 93.8% not Hispanic or Latino, and only 7.6% Hispanic or Latino. In contrast, the U.S. population, according to the 2020 U.S. Census, is 61.6% white, 12.4% black or African American, 6% Asian, 1.1% American Indian or Alaska Native, and 0.2% Native Hawaiian or other Pacific Islander, and 81.3% not Hispanic or Latino versus 18.7% Hispanic or Latino. The significant overrepresentation of white and not Hispanic or Latino populations and underrepresentation of black or African Americans, Asians, American Indians, and Hispanic or Latino populations raises concerns about the generalizability of the clinical trial findings to a large proportion of the U.S. population. Moreover, the lack of diversity in the enrolled subject population indicates a failure of Moderna and the trial investigators to ensure that selection of subjects was equitable and satisfied the basic ethical principle of justice articulated in the 1979 Belmont Report that, upon which human subject protection regulations are founded. Third, although no serious safety signals were identified during the clinical trial of the proposed 50 microgram booster dose of the Moderna vaccine, the safety database for this booster dose is very small, including only 171 subjects who received a 50 microgram booster dose administered at least 6 months after completion of a primary series of 2 100 microgram doses, the authorized doses under the initial EUA granted by the FDA, and 173 subjects who received a 50 microgram booster dose administered at least 6 months after completion of a primary series of 2 50 microgram doses, a dose not authorized by under the EUA. For the former subject group, median follow-up was just 5.7 months and a range of 3.1 to 6.4 months. Finally, while the U.S. already is implementing widespread distribution of COVID-19 vaccine boosters, the vast majority of people in low and middle income countries have had no access to any COVID-19 vaccine, let alone the highly effective mRNA vaccines. The world continues to suffer from an artificial scarcity of high-quality COVID-19 vaccines because governments are permitting drug corporations to maintain monopolies. It is ethically imperative that the U.S. government move to rapidly ramp up global vaccine manufacturing so that every person on the planet can be vaccinated. Thank you for your attention.
Thank you, Dr. Karom. Thank you all for your comments, and this concludes the OPH session, Open Public Hearing session. Now I hand the meeting back over to Dr. Manto. Dr. Manto, take it away.
We are at the end of the open public hearing. It would be great if we could start question and answer session at 1:45 Eastern. To Prabha and Kathleen, do you think that's going to be feasible?
Dr. Manto, it is now 1:20 p.m. in Eastern time, so if you take 10-minute break, we can start earlier, then maybe around 1:30.
And the committee members are online?
They are.
They know to start.
They are.
That's the thing that worries me.
They're online.
They're all online, sir.
Okay, 1:30 start. Well, wonderful. Just a 10-minute break right now.
All right, welcome back from that real quick short break. Dr. Monto, are you ready to kick off this last leg of today's meeting?
I am, and I want to thank the staff for expediting this return to our deliberations. We've got a long day, and moving things forward is always very helpful. So now we've got the question and answer session, and it's questions and answers for both FDA and for the sponsor, who are all back online. So Dr. Carilla, you are leading us off.
Thank you, Arnold. Yeah, I have a question for Moderna. I don't know if Jacqueline is back in the hot seat, but she is. Okay, thank you. Yes, so with regard to your immunobridging analysis, it seems that that is predicated on the assumption that the protection is mediated exclusively by antibody response, specifically neutralizing titer. And it's clear that even when your neutralizing titer levels drop, you're still seeing some degree of protection. And that's not surprising, particularly for severe disease, because we would expect that there would be some very good, hopefully some good cellular memory responses that would be kicking in. And so my question really gets to the heart of, at a lower dose, what is the impact on all of those other protective effects? You're predicating everything just on the neutralizing titer dose. So one aspect is, are you actually going to be impacting the decay kinetics of the antibody response, which seems to be why you get breakthrough infections in the 6 to 8 months? So is it going to come sooner? And secondly, what's the potential impact on the waning immunity with regards to more severe disease, hospitalizations, and death?
Yes, so Dr. Carrillo, that is a very interesting and relevant question, and if I implied that neutralizing antibody, that I believe that's the only element of protection that the vaccine is inducing, then I apologize, I misspoke. We have phase 1 data demonstrating the induction of both CD4 and CD8 cells, so there clearly is some T cell work that is induced. The other point in a collaboration with the Cove PN, where we looked at of risk, I mean, there was an estimate that at least 40% or so of protection in our recent publication is likely due to T cells. And there's one final line of evidence that there's T cell immunity, and it comes a bit from the exploratory analysis I showed you in the core deck, where you saw the increase in neutralizing responses not only to the original strain, but also to beta, gamma, and delta. Those samples were actually taken at day 15, and in the Cove study, we really didn't see full neutralizing antibody titer until 2 weeks after the second dose. After the first dose, even 1 month afterwards, did not see neutralizing antibody titers in about half of the subjects. So that brisk return is certainly an indication that immune memory has been established. That said, we are still concerned about the breakthrough disease that we've been observing in the participants in the Cove trial, and particularly the breakthrough cases that we're starting to see in severe disease in the older adults, which is why these data that we've investigated earlier in the year, we now have submitted for emergency use to enable booster vaccination. We are going to investigate immune memory further. We have an ongoing collaboration with Washington University, and as we continue to study the impact of booster doses and the possibility in the future of variant booster doses, one of our ongoing studies is actually going to be looking at germinal centers, memory B, and memory T cells. So in summary, I think you're right that T cell immunity is contributing here, but nonetheless, we continue to see breakthrough cases.
One follow-on. Do you have any evidence or experience with perhaps other mRNA-based vaccines that you've worked with that would suggest that a 6-month boost is likely to lead to better durability than what you've seen with what is likely a suboptimal dosing interval of 1 month?
So we have ongoing vaccine programs in CMV. CMV is the most advanced program that's in a multi-dose usage. So subjects in phase 1 and phase 2 clinical trials have been vaccinated at dose 1, then 2 months later for dose 2, and then 6 months after dose 1, 4 months after dose 2 for dose 3. And in CMV, we have also observed the induction of T cells. We have antibody persistence data out to 6 months after that third dose. And we see persistence, but again, this is smaller sample sizes. And I think that question will probably be answered better in the phase 3 trial that we're about to launch.
Okay, thank you.
Thank you. Dr. Gans.
Thank you very much. It's wonderful for this opportunity to ask a question. I did have a question about breakthrough. So, Arnold, one question now, and then I'll come back around if I have another question. Is that our idea?
Thank you. Appreciate
okay.
that.
So I guess my question then right now will relate to safety. So we've seen a lot of data on the original safety for the 2-dose, but there has been 1.5 million doses of the Moderna. We've seen other data related to Pfizer. And I'm wondering if someone can give us any follow-up on safety data in the large cohort of people who have already received. I realize they're immunocompromised or whatever. I know it's not necessarily relevant as a group during today, but I'd like.
Dr. Miller, can you answer that, or should we refer that also to FDA?
No, I'll be happy to at least start, and I'll share with you the data that we're aware of. So we have had the emergency use authorization for the third dose in immunocompromised population since about the middle of August. And in that subset, we have been reported to our pharmacovigilance database 355 total events. The most commonly reported adverse events that we have heard about really align with the symptoms that we solicit as part of the clinical trial process. So fever was the most commonly reported event, and it's followed by headache, arthralgia, chills, and myalgia. So overall, I think it's been a bit of a short time period for us to really have data in that regard. We are generating additional data in immunocompromised patients. So we have an ongoing study in 240 renal and liver transplant patients, and we are offering all of those patients a third dose. So we will be reporting the safety data from that clinical trial as well. And Dr. Gans, if I may, you had asked me a question before the break, and I was able to pull up the slide showing the geometric mean ratios by age with the immunopersistence. So panel A, please. Thank you. So what you see in the top row of the table are the antibody persistence results in the 18 to 64-year-olds in the left column, and the greater than 65-year-olds in the right column. The study 301 is pre-vaccination, and that's why the titers are so low at 9 and 10. But in the older adults, the pre-booster titers were 91. In the younger cohort, they were 177. You see the post-vaccination titers on the slide, and it results actually in very comparable geometric mean increases. So 1.7 for the younger cohort, 1.9 for the older cohort. Thank you.
Dr. Hawkins.
Great. Great. Thank you very much, Dr. Miller, and to all the presenters. I'm a consumer representative and a physician, private practice. Can you respond to the criticism often levied against Moderna, including today in the open public hearing section? What is Moderna's commitment to COVAX and other steps it will take to help control the pandemic? in countries suffering disproportionately? And can you give specifics?
Yes, I'd be happy to address that question. So recently, I'm actually going to refer you to an open public letter that was published by our CEO, where he laid out a five-pronged strategy to address COVID-19 disease in the developing world. The first element refers to our announcement in October 2020 that we were not going to pursue patent enforcement of our mRNA technology for the duration of the pandemic. The second has to do with the 50 million doses of COVAX, or a vaccine that we've delivered to COVAX through September of 2021. And that was made possible by our pursuit of the emergency use authorization letter from the WHO. So we've been meeting with the WHO and the SAGE working group throughout our development. We have an agreement to supply doses to COVAX, 500 million doses to COVAX in 2022. And we have just announced that we will be building a manufacturing facility in Africa. And this is important because it will be a localized manufacturing facility in Africa for Africans. We also have plans to distribute 1 billion doses to low-income countries in 2022. And you know, even though it includes greater complexity, we're reducing the dose to 50 micrograms in order to try to make more vaccine available for the world. So that frees up 1 billion extra doses if we can have a booster dose.
Thank you very much for that. Thank you very much for that. Do you have a timeline on that manufacturing plant in Africa?
My sincere apologies. I'm in the R&D group, so I have to check back with other colleagues to be able to answer to that.
Thank you very much.
Dr. Perlman.
Yeah, so I just had a question about the myocarditis. So I don't think we understand why that occurs, and the fact it seemed like it might have been occurring less after the third dose than the second dose. So I don't know if that's true, but it seemed like that was the case. So does that give you any insight into possible mechanisms? Because of course the concern is if you had immune response to either the vehicle or the product of the RNA, that would get worse potentially with repeated immunizations. But it seems like it's not. So does that tell us anything? Does Moderna know anything about possible mechanisms there?
So the mechanism of action question is one that's really important to us as well because patient safety is of the utmost importance. So after the third dose, I think you mentioned we don't have a lot of cases yet. I would say we also don't have a lot of exposure yet. And I wanted to mention that for that reason, we actually are offering the 50 microgram booster to all of those subjects in COVE, or the phase 3 study 301. The reason to do that is to investigate the vaccine in a larger safety database, as well as to generate additional immunogenicity. And as part of that effort, we have enhanced the clinical trial procedures to detect myocarditis. So for example, we're now screening subjects for myocarditis-specific symptoms after vaccination. We are collecting serum samples that we're banking in case a subject should develop symptoms later and we need to test troponins and compare to a baseline. We've convened an adjudication committee composed of cardiologists independent from the company who will be evaluating these patients and advising us on what we should be doing to investigate further. So the question, part of your question about mechanism of action though, because in 25,000 subjects we are probably not going to be able to tell too much about myocarditis since this is such a rare event. We believe that understanding the immune response that's actually induced by the vaccine is really a critical component. So in addition to the mechanistic study that I described in collaboration with Washington University. We're also looking to do a mechanism of action study comparing multiple antigens in our mRNA technology and then looking at system serology afterwards. So hopefully as we continue to generate these data, we'll be able to elucidate a greater understanding.
Okay, thank you.
Thank you. Dr. Levy.
Thank you. I have a question that's actually both for FDA and for Moderna, and it has to do with the data that's being presented today on antibody responses to the mRNA vaccine, to the booster dose. That makes a lot of sense to look at because we have a lot of evidence in animal models and some evidence emerging in humans that an antibody response is relevant to protecting us against coronavirus infection and disease. That said, what's being presented is very specific types of analyses, fourfold rises and other types of cutoffs to judge a, quote, zero response. And all of this kind of begs the question of, do we know the correlative protection? There was already a question about antibody responses versus cell-mediated responses, and I appreciated the response from Moderna on that. But I'm taking a step back and asking both FDA and Moderna, what is their best estimate of the antibody response level that protects against infection and against severe disease? I know research is ongoing, but we're talking about a lot of very specific data on antibody responses, and we need a context to contextualize those data. So I'm wondering if FDA and Moderna could comment on that. Thanks.
So I'm happy to start, but then I will hand over to the FDA. So Dr. Levy, your question, I think, allows me maybe to expand a bit further on the publication I spoke about earlier. So as part of the Study 301 and in our collaboration with the COVID-19 Prevention Network, we utilized the immunogenicity subset and examined actually correlate of protection. So we had baseline results in all subjects. We made sure to sample subjects once they had a case of COVID-19, and therefore we had a subset of immunogenicity in patients that were non-cases, and we're able to analyze antibody titers looking at individuals who received placebo that got infected, individuals who had placebo did not get infected, and importantly, mRNA recipients who had breakthrough disease versus the rest of the pool of mRNA recipients. And so we've published that work on the MedBrick server, and it has been submitted for peer review publication. But what we found was that for 50.8% of the subjects, or the vaccine efficacy compared to individuals that were vaccinated and unvaccinated with messenger RNA was 50.8% if the antibody titer in the breakthrough case was undetectable. It was 90.7% for an antibody titer of 100, and it was 96.1% for a titer of 1,000. And so while this is not at all a validated correlate of protection, the data would need to be submitted to FDA and undergo additional statistical review. We believe that 1,000 benchmark really represents a reasonable threshold that we should be targeting. And it also aligns nicely with the GMT that we saw post-vaccination in the Cove study.
And also to the comments from Dr. Alroy in Israel, but that's a different product. It's the Pfizer product. But again, they're not there yet to announce an exact correlate, but she talked about breakthrough when the titers were in the hundreds. Does FDA have a comment on this?
Hi. So I can comment. So I wish I could tell you what FDA thinks is the correlative protection. That would make all of our lives so much easier, wouldn't it? But at this point, FDA's position is that we don't have enough information to understand what specific threshold of any immune response is fully predictive of protection. And in the meantime, we're tasked with evaluating data and taking action to address public health needs. And so to do that, we are relying upon established regulatory science and precedent in which we use an immunobridging approach based on an immune marker, which, although it may not be scientifically established to predict protection at a given threshold, we have reasonable enough confidence in the clinical relevance. And we use that immune marker to bridge back to a dosing regimen and a population in which efficacy has been demonstrated.
Has FDA made an estimate of this number and is not free to talk about it? Is that the situation?
No, we are continuing to await additional data, both from vaccine manufacturers as well as U.S. government partners and elsewhere.
Okay, so to recap, this fourfold... Yeah.
Let's go on to some other questioners. Dr. Chatterjee.
Yeah, my question is actually for Dr. Miller. I believe that you presented data that the boost with the booster dose is less prominent in those participants who had a higher pre-booster antibody level compared to those who had lower pre-booster antibody levels. Do you have any kind of an explanation for that? Because when I think about those data, I think about, okay, this is not a live virus. This doesn't need to replicate. So why are we seeing this difference in people who had higher pre-boost antibody levels versus those who had lower pre-boost antibody levels?
Yes, Dr. Chatterjee, thanks for that question, and I think it might help if we put the slide back up. So could we please put up panel 8? Thank you. So I believe this is the slide you were referring to where we showed that the subjects who did not achieve the fourfold rise had a pre-booster titer of 492. With respect to the reason why subjects may not have responded as well, I'm going to start, but I'm also going to ask for my colleague, Dr. Darren Edwards, in the research group to contribute as well to the response. So overall, when there are pre-existing antibodies, our technology works through expression of the protein antigen on the cell surface. And pre-existing antibodies can, I believe, bind to that cell surface protein. But I'm going to ask Dr. Edwards to come up and explain further.
Thank you, Dr. Miller. My name is Darren Edwards. I'm the Director of Immunology within the Infectious Disease Group at Moderna. As Dr. Miller alluded to, the mechanism of action of our vaccine is to deliver the spike protein mRNA to cells where it is translated into protein and inserted into the cell membrane of the expressing cell. That protein, while present not only in the injection site, but also in the draining lymph node, is able to activate the immune system. However, it can be impacted by the presence of pre-existing antibody. And that is a potential reason why in the group that had a high baseline, we see a lower neutralizing antibody level after the booster.
Thank you.
Okay.
As we go forward, I just want to remind the committee that one of the discussion question we're going to be asked later on, and we aren't going to have a chance to do a question and answer of the sponsor at that point, about other ages going down in the discussion topic to 18. So let's keep that in mind as we ask our questions. Dr. Moore.
Hi. So, I mean, clearly this is not amazing new thing, is that this epidemic won't end until we end transmission, regardless of how effective on an individual basis the vaccine is. And what we saw was that the FDA reported they had that Moderna had 18 cases post-booster that were PCR or antigen positive. So it looks we don't have a control group, so we don't have a vaccine efficacy for asymptomatic or pre-symptomatic infection and the protection against that. But that's a really, really, really critical thing for the ending of this epidemic. Do we have an idea of what it would take to be able to ship to a Delta booster, because people have already had 2, if I understand it correctly, Wuhan 1 sequence injections. Now they're getting a 3rd Wuhan 1 sequence. And if you did ship to a variant of concern booster, would you anticipate that you would have increased protection against asymptomatic infection or pre-symptomatic infection? And since that's our best guess of inhibiting transmission.
Yes, so I think your question maybe gives me the opportunity to review some data first from an ongoing vaccine effectiveness study, because we take your point that because all of the placebo subjects have received vaccine, it's not a true efficacy study anymore. But we are currently working with Kaiser Southern California in a large-scale vaccine effectiveness study where we're able to compare vaccinated versus unvaccinated individuals. And as was noted earlier this morning, this kind of analysis has some limitations because unvaccinated individuals don't necessarily have the same behaviors as vaccinated individuals, but it still, I think, provides at least a value in understanding the data that we're seeing. So may we please show panel A? While we're waiting for the slide to show up, I'll just say that we have been following vaccine effectiveness in approximately 1.1 million Kaiser members, and the effectiveness has been estimated not only overall but also by variants of concern. So the slide that you see now in the orange includes vaccine effectiveness against all PCR samples that have been detected that were not of the Delta variant. And I guess I should note here that unlike most effectiveness studies, we actually are sequencing every subject that is a case in this observational study, and we'll be continuing this study into the period should the booster dose be authorized. In green, you see the vaccine effectiveness against the Delta variant, and as you can see, I mean, the vaccine effectiveness is still high, but the Delta variant is clearly lower. The other, I think, important point about the Delta variant is after initial vaccination, the vaccine effectiveness actually was much higher. So Delta effectiveness was 94.1% between 14 and 60 days after vaccination. This declined to 80%, 151 to 180 days after vaccination. And the waning of that effectiveness was less pronounced for the other variants. So indicating that as the antibody titers wane, we are seeing also a concurrent waning in vaccine effectiveness. And then, I'm sorry, could you please remind me of the second part of your question?
Well, the question is that obviously if you have, right now we're in the middle of a Delta epidemic. So if you have a better genetic or a better antigenically fit booster, we're not really, at least I'm not terribly worried that we're shaping the immune response such that it will not recognize earlier variants, because people have already seen those earlier variant spike proteins because they've had 2 doses of the Wuhan vaccine that has roughly 95% vaccine efficacy. So if they get a booster with a new antigen that is shaped towards Delta, then it seems like your efficacy will be much better. Now, the Kaiser study, if I remember correctly, had a 72% estimated vaccine efficacy against asymptomatic infection. You've got 18 people out of 149 that are point positive at some point after booster, maybe it was 16, I'm sorry, there may have been 2 people that were early on that have not really reached full antibody response after booster. But nonetheless, it's about 10-12% of those people are positive for SARS-CoV-2. I'm sorry, we don't have a comparison group to that group, but if you invert that ratio, if we had a hypothetical comparison group, then that would be an attack rate in that group of 30-40% during a comparable period, I would think. And so that seems just really, really high, and that's one reason why I think the efficacy looks somewhat low in protecting from asymptomatic carriage.
Yeah, okay, thank you so much for reminding me of the question. And you're correct, but I want to emphasize that the 18 cases that were detected, these were primarily cases that were found from the nasal swabs that we conduct routinely at dose 1 and dose 29. So you're absolutely right that they were contributing to asymptomatic infection. The other part of your question was with respect to variant-specific boosters, and we actually are investigating the possibility to further boost individuals with variant sequences. And we think that this is really important, even if we don't administer booster doses for quite some time, to understand whether the messenger RNA sequence can be replaced out with a comparable profile to what was observed in the large-scale study. And can you put up panel B, please, because it gives me a chance to speak a bit about the ongoing work we have with boosters.
Right. Let's not spend too much time on it, though. We're getting short. Go ahead, please.
Oh, okay. Well, Dr. Monto, then I'll just summarize by saying that we agree that it's absolutely important to understand if a beta or a delta sequence could better protect against the variants of concern, and that's why we've committed to studying it.
Thank you very much. Dr. Offit.
Right, thank you. So a question for Dr. Miller. Jackie, Tony Fauci has said that were this not a pandemic, this would have been a three-dose vaccine. The reason he said that is that he likens this vaccine to the inactivated viral vaccines, like the inactivated polio vaccine, the hepatitis B vaccine, or hepatitis A vaccine, where you need to have an interval of 4 months plus in order to get decent frequencies of memory cells, because that's going to allow you to have protection against serious illness and to have durable protection. The question is this that vaccine? Because, as you said, it's not quite an inactivated viral vaccine. You have viral proteins that are being made in the cytoplasm, which likens it more frankly to a live attenuated viral vaccine, where a single dose can induce long-lived memory responses. So the thing you said earlier that I think is really important is that when you do this third dose and you're looking at the effect of the third dose, I think it's really important to look at the memory B cell response to answer the question, do you really boost memory B cells? Because if you look at the data by John Weary and Shane Crowdy and La Jolla, John Weary at Penn, what they find is that 6 months after your 2-dose vaccine, you have reasonably high frequencies of memory B cells, which, if anything, increase over time, suggesting long-lived immunity induced by two doses. So it may never have really been a 3-dose vaccine. And if the goal is to try and protect against the unfortunately named breakthrough infections of asymptomatic infection or mildly symptomatic infection, which I wish we'd never use that term because it implies failure, and that's not a failure, then we're going to be talking about giving frequent boosters, which I don't think is a reasonable strategy for this vaccine. But so I think it's really important to look at, can you boost memory with that third dose?
Yeah, thank you for that, Dr. Offit, and we agree, which is why we are engaging in that particular mechanism of action study. And I'll just mention that we're also utilizing a bivalent vaccine in that study. So we are looking at the beta delta in a combination vaccine to also understand if you give a different antigen, what does the memory B cell look like to that variant of concern. I think to your question about what we call the schedule, I take your point that one person's primary series and another person's booster series, I suspect that there's a continuum of improvement in protection and immunogenicity with every dose. And I guess what I would say about longer-term boosters is that I'm not sure that a booster that you give in the middle of a continuing pandemic that's due to a lot of different factors necessarily will determine what will happen in the future. And so, the data set we're bringing here today is really to address a specific problem, which is the breakthrough severe disease that we're beginning to see in the patients that have been.
Thank you.
Thank you.
Dr. Lee.
So, Dr. Miller, this is something of a follow-on to Dr. Chatterjee's comment about the fact that your serial response seemed to be greatest among those that had the lowest pre-booster levels. And I guess one of the questions I have is whether you actually looked at the association between time from their last second dose to when that happened. And so what I'm leading up to is the fact that maybe 6 months, we've drawn a line in the sand of 6 months, which is completely arbitrary, and whether or not it would be optimal for people to wait longer to get the boosters, et cetera, because the waning hasn't occurred as much in some, and they don't benefit that much about it. And so I'm just interested in your comments on that observation. Thank you.
Yeah, that's a great question. And unfortunately, in this phase 2 trial, subjects were really vaccinated in a relatively narrow time window, so 6 to 8 months, and that particular analysis will not be as helpful. What I would say is that's why we think that investigating the booster dose in the phase 3 study, COVE, is so important, because by that time, subjects will have been in the earlier group. Now it's even later than July and August, so closer to 14 and 15 months past their initial vaccination, while subjects who were originally the later group, originally allocated to placebo group, are going to be about 9 to 10 months after vaccination. And I think all of those data together may build a picture. And I think you'll see some data tomorrow presented by colleagues at DMID regarding a booster dose within the 4 to 12 week window. So hopefully that will also help inform the discussion.
Thank you.
Thank you. Dr. Meisner.
Thank you, Dr. Monto. And Dr. Miller, I would like to ask you a question about sterilizing immunity. And I think, as you just said, it's so important to look at breakthrough disease or disease that occurs in people who are fully vaccinated, rather than just an infection from whom one can get a positive PCR or recover virus. And it seems to me it's going to be very difficult with the mRNA vaccines to achieve that objective. That is, asymptomatic infections in someone who had pre-existing immunity, because these viruses are mostly stimulating IgG and circulating immunity. Have you looked at IgA? I guess there's no reason to think that there would be secretory IgA made, but is it reasonable to expect that these vaccines would prevent essentially colonization that results in asymptomatic disease in someone who's immune?
So, Dr. Meisner, I'm going to turn your question back over to Dr. Edwards in just a minute, but I will tell you that in the phase 3 study, and again, this is a different moment in time, so it was when the original Wuhan strain and the alpha variant were circulating, but at the very end of the placebo-controlled period, so when subjects were in the process of crossing over, they had a final visit. That was the final efficacy that I described to you, and in the interest of time did not speak to the asymptomatic infection rates, but we had an efficacy of about 60% against asymptomatic infection. I think the question about sterilizing immunity is probably best addressed, and IgA best addressed by Dr. Edwards. Thank you.
Thank you, Dr. Miller. So I think some of the best evidence that we have on the ability of our vaccines to elicit secretory IgA and the mucosal tissues is from our non-human primate studies that we have run with our wonderful collaborators at the NIH. Several of those studies have been published, and amongst the observations that we've made is the presence of IgA in the nose and in the BAL, in the lung samples that we've collected. Now, more recently, we are now looking in non-human primates over the course of an entire year to look at the durability of protection during that time period and the immunogenicity that's observed during that time period. We don't yet have specific IgA measurements over that time period, but the results should be available in the near future, at which time it will be published. So it will be interesting to look not only acutely after vaccination, the presence of IgA, but what levels are present over a long period of time.
Thank you.
Thank you.
Which time it will be published. So it will be interesting to look not only acutely after vaccination, the presence of IgA, but what levels are present over a long period of time.
Thank you.
Thank you. Dr. Gantz.
Thanks for allowing me to come back on. And it's so great to hear from my colleagues because they had a lot of questions answered. Anyway, I think there's a lot of evidence that we're now seeing that despite our desire to see this memory response, I think we are starting to see a signal that is suggesting to us that despite what we understand, what might be the control of this virus, so breakthrough of serious disease. I take Dr. Offit's point that we're not trying to just disease that we can see by PCR. However, that is important for control of transmission. But anyway, I did want to understand more because we are starting to see a signal in individuals, and I just different from what we were seeing previously. So I think unfortunately for the Moderna, I know that the breakthrough was only 19 cases, so have a low number, but the pool of people we were looking at was very low too. So the 4 individuals who were not accounted for by age greater than 65 or those under 65 who had pre-existing conditions, which I think would be taken care of by the people that you've listed for your extended EUA. The 4 individuals who don't fall into any of those categories, would they actually perhaps fall into a category of exposure? So I'm wondering if you know anything more about those individuals that could help us understand they too would have been protected by being provided and considering occupations that are high exposure applications. We do know inoculum correlate of disease.
Yes, Dr. Gantz. So I think you're right that we don't have a sufficient number of cases in this particular analysis to be able to refine our analysis to that level of degree. I think the phase 3 study, has larger sample sizes of those kinds of populations. I think I'll clarify that so our intention in our labeling information is to say that the booster dose is indicated for those 18 years of age and above. There's no reason to necessarily exclude someone that either FDA or particularly CDC, who make the vaccine recommendations for which population should be vaccinated. We want to give them the ability to recommend the vaccine booster for who they think needs it.
Thank you. Let's go on to Dr. Nelson.
Thank you. This question's also for the sponsor.
There will be 2 more questioners before we move on. So Dr. Nelson and then 2 more.
Great. This is indeed a question for the Sponsor and Dr. Miller, thank you again for enlightening us this afternoon. This has to do with the relationship between pre-existing immunity and the risk for adverse events by a booster dose. My understanding of the data presented earlier was the reactogenicity is measured by common adverse events in the combined data set for the 300 recipients of the 50 microgram dose. Doesn't appear to be significantly different than after dose 2 compared to the primary series. So what was found was that the risk of myocarditis and pericarditis does appear to be increased after dose. So it's unclear to me, probably most, whether the level of current humoral and cellular immunity at the time of boosting is directly related to this risk or the risk of any other systemic adverse. What I didn't see in the briefing material, this isn't a criticism, it's a question, is there data that stratified the risk for systemic adverse events by pre-event titer? Data of this type will help us do the risk-benefit analysis for broader populations who are largely immunocompetent, such as the third population that we're asked to address today, that is the 18 to 64 at higher risk for institutional occupational exposure. Premise being, with the immunocompetent possibly having a higher baseline cellular and humoral memory response from the 2 doses, are they at significantly higher risk for a booster dose?
Yeah, so thank you for that question. Unfortunately, we don't have that analysis, but it's a really excellent suggestion. So again, thanks.
Thank you. Dr. Fuller.
Dr. Fuller, are you there?
Be able to follow breakthrough cases that would be captured. But in people whose baseline swabs or who had baseline evidence of previous infection, they actually tended to report overall that they had lower reactogenicity, although some specific elicited symptoms, so the individual symptoms, some of them were higher. You know, I think we will learn a lot more about the third dose and a lot more than we did in the original iterations of phase 3 when we give this 50 microgram booster, because there certainly was a lot of breakthrough disease in the original placebo group. They've actually now continued potentially in the study, and we'll be vaccinating them with this additional dose. So in terms of education of people, though, I think, you know, regardless of whether they had COVID before or they did not, it's important that patients understand what to expect before they get the vaccine. And that's why we really invested in looking in the comparison to the phase 3 data. The phase 3 data are the data that are currently represented in our vaccine fact sheet. And I think going through that fact sheet, letting people know what they might experience, let them know that it at least in our initial studies, has been similar to what they saw after dose 2, is probably the best guidance we can give them.
Thank you.
All right, thank you.
Dr. Rubin.
Thanks, Dr. Monto, and I'm honored to get the last question, if that's really the case.
It is before we have more comments.
Yep. The presentation today started with included presentations from our Israeli colleagues about their Pfizer vaccine results. And in fact, when Pfizer's vaccine came up for consideration, the fact that that there was widespread use and some efficacy data from Israel, I think influenced many of us to think that this was a reasonable idea. And now we have more of those data, but they're Pfizer data. So I want to ask Dr. Miller a slightly unfair question. Do you think we can generalize from data from this other vaccine to what you might see in Moderna? Because I will say that the safety data in particular are very thin. They're really, as was pointed out in the public comments, they're really only 170-ish people got the same dose that we will be giving to if we approve a third dose.
Yes, so Dr. Rubin, we don't have real-world data similar to those that were generated in Israel, and I will say I guess we're indebted that Israel decided to be the front runner so that we have those data to review today. But what I will say is I think the 1.5 million Americans who have already been vaccinated with 100 micrograms as a third dose, and these are admittedly immunocompromised but also medically vulnerable individuals, contributes to at least some of the understanding of a safety profile. And that safety profile is in a different population, but reasonably conservative given that they got twice the dose. We're going to continue to follow the subjects that I described in the Kaiser study once they are, if they are offered their third dose. And that will be another way in which we can continue to evaluate what happens in terms of vaccine effectiveness. And then certainly from a safety perspective, all of the ongoing pharmacovigilance activities that are currently underway will continue and include subjects who have received a third dose. So I would say I think the data, much as they did with the original messenger RNA submission where we had 30,000 subjects worth of data, but now we have over 190 million doses worth, that will grow the database in a similar fashion.
Thank you very much. We are going to terminate the question and answer session right now because in reality we do not have only our voting topic. We will have after our voting topic a discussion which may be a rather robust discussion of steps forward for all of the vaccines. So we will go into our committee discussion, and this discussion will be focused on our voting question. So can we get the voting question up so that we can at least focus our discussion on this?
This is the discussion topic today, Dr. Monto?
No, that is not the discussion topic. I want the voting.
There's a voting question for you,
topic.
Sir.
No, you got the wrong. You've got tomorrow.
I think tomorrow's.
Oh, hold on a second. Did we put up tomorrow's? All right, let me stop sharing that real quick. Somehow we made them a...
Can we vote on tomorrow?
1 second, sir. No, we're not voting on tomorrow's. Hold on a second. Committee discussion. I just want to make sure I grab the right one. There it is.
We want the voting on Moderna. With a 65 plus.
All right, hold on. I just want to make sure it's the right one. Discussion topic should be, yes, it's loading in now, sir.
We don't want discussion, we want voting. We're going to just do the discussion after.
It's both, sir, it's both. It's both.
Oh, you got both?
The slide
okay.
that, yes. Just make sure it's the right one.
Okay, that's it.
There you go.
So there is our voting question. And what we're going to do now is discuss this, have the vote, have any explanations of votes afterwards by those who want to explain their vote, and then go on to the discussion topic, which is not going to have a vote. And that's going to be trying to harmonize any recommendations across the board in terms of different age groups and things of this sort. So reserve your broad thinking to the discussion, and let's focus now on the question of that we've got in front of us, which we will vote on. Okay. Dr. Nelson.
Last time. Thank you.
He put his hand down.
Oh, okay. Dr. Meisner.
Thank you, Dr. Monto. And I would like to ask a question about the third bullet, going back to something I mentioned earlier. Are there data indicate that individuals who have occupational exposure to SARS-CoV-2 are at high risk of severe COVID-19? For example, for healthcare workers, are they at increased risk of severe infection? My only point being, I think we have to be sure that we can justify everything we're saying. I'm not aware of data to support that, but maybe I need to be educated.
This mirrors the approval that we gave for the Pfizer vaccine. So anybody at FDA or elsewhere ready to answer that question, Dr. Fink?
Let me try to explain a little bit about how FDA arrived at this authorization statement for Pfizer. And you're right that when we held the VRBPAC on September 17th, and when we constructed this authorization statement, there were not specific data, nor do I think there are specific data now that speak to the risk of severe COVID among individuals with increased exposure in institutional or occupational settings. But I think it's important to highlight a couple of principles. First of all, this third bullet includes the words severe COVID, but it also includes serious complications of COVID. And as Peter Marks explained earlier in the day, there are sequelae of COVID, including long COVID, thromboembolic events, and other sequelae that may not meet someone's definition of severe COVID, and yet would be considered serious conditions that would be applicable to the statutory criteria for emergency use authorization. I think it's also worth mentioning that at the time of COVID, following primary vaccination, one can hypothesize that it might be the same groups as would be at higher risk of severe COVID prior to a primary series, but we don't know this for sure. We didn't have data, and yet such groups may exist. And so really the intent of structuring the authorization in this way is to provide a regulatory allowance for groups that could reasonably be considered at risk of serious complications of COVID, for which there would be benefit to a booster dose being made available under emergency use authorization. The point of emergency use authorization is that it is intended to address current emergency situation. It can be changed as circumstances evolve. And furthermore, ACIP can evaluate data to make recommendations for use of the vaccine that has been made available under EUA, and those recommendations can change as circumstances evolve. And so really this authorization was designed to allow for flexibility in making the vaccine available under EUA to individuals for whom it could provide a benefit and where the benefit would outweigh the risks.
Thank you very much.
Thank you, Dr. Lee.
Can I have a follow-up?
Yeah, go ahead.
Thank you, and thank you, Dr. Fink, for that thoughtful answer. I appreciate it. The only point I'd like to say is that I think it's so important that that these recommendations are evidence-based. And I agree it's the ACIP which will make this decision. But it's so important because this is such a controversial issue. If we can't defend these recommendations based on evidence, I think it's going to further complicate getting this vaccine into every single adult American, and that's really what we want to do.
Thank you, Dr. Meisner. Dr. Lee.
So I think one of the questions I'm a little bit troubled by is that, as Dr. Moore pointed out, the data we have that have the individuals that had the full dose of Moderna followed by the booster is really only limited to about 149 patients, which is a fairly limited group, and also only meets one out of the two criteria that were pre-specified for the emergency use. So I guess one of my questions I have, as you can see I have a little bit of hesitation, maybe for Dr. Fink, is would the requirements for full authorization of the booster mimic the ones that we have now for the EUA, or would they be more stringent? Have they been formulated, or what is sort of the thought at FDA? Were we to grant an EUA, what would be the requirement for them to get a full authorization for the booster?
I had to unmute myself there. So thank you for that question. I would really like the committee to focus on the question as it pertains to emergency use authorization. It is an entirely valid question to ask where we are ultimately going. We've heard discussion today about what the appropriate regimen would ultimately be, perhaps under different circumstances when we're not in the middle of an active pandemic. I really would like the committee to focus on considerations for emergency use authorization right at this moment in time.
Thank you.
But it actually doesn't meet them. That's what I'm getting at. Thanks.
Thank you. Dr. Hildreth.
Turn my camera on. Okay. Thank you, Dr. Monto. I want to go back to Dr. Meisner's comment about bullet number 3, and that is that oftentimes individuals who have occupational exposure are brown and black people who work under conditions where they're exposed. And as we know, they're more likely to have underlying conditions that predispose them to severe COVID-19. So as far as I'm concerned, that's the only justification needed for bullet number 3, the higher percentage of people with underlying conditions who have occupational exposure. So for me, bullet number 3 is very important and should remain a part of this voting question. Thank you.
Dr. Sawyer.
Mine is more of a comment. I don't really have a question. I've been listening to all of the discussion and the excellent questions that have been raised. I'm of the opinion that we need boosters. I find the Israeli data compelling, as well as the breakthrough cases we're identifying in the United States. I agree that the amount of safety data presented specifically from the company was very minimal, but I do think that we can take some reassurance from the 1.5 million U.S. citizens who've already received this vaccine at a higher dose and we have good surveillance systems in place to have detected any new or unusual side effects. I also think we can probably extrapolate from the Pfizer data in Israel and the experience in Israel in that in all other ways these two vaccines are quite similar. And lastly, I think since I'm of the opinion that we need these boosters to be available for use in some populations, I think it's best to put it in the hands of ACIP to determine exactly who should get it and under what circumstances. I'm not wild about a bunch of 20-year-olds running out and getting a booster dose unless they're at increased risk of hyperexposure or severe outcomes.
Thank you, Dr. Gans.
Hi, thank you. I just wanted to make the comment alongside of my colleagues how important I think it is to protect. So we use vaccines protective. I'm not sure that we want to allow signals to be higher. I couldn't agree more that the Israeli data related to a messenger RNA vaccine, what we're also considering here today, is very compelling. They've done a really good job of showing us that it stops transmission that aren't vaccinated and actually impacts severe disease. Their hospitalizations did fill up with people who were outside of ones that were considered necessarily in the first round to be at risk for hospitalization and severe disease, so I think we have to be careful about that. I couldn't agree more with my other colleagues also about exposure and really protecting those people who are on our front lines as well as those who are in industries that are bringing them at higher risk. I think that Dr. Fink's comment about what was happening pre-vaccine is very important. There were healthcare providers who were really getting sick outside of those age groups and without underlying conditions, probably because of, again, inoculum effect and how much they were being exposed. We do have PPE now, and we do have masks. However, some individuals are just in situations where the conditions are such that these are even. So I also find it very important to continue to include this in our recommendations on way. And I couldn't also agree more with Dr. Fink to say we are in the middle of the pandemic. So stopping this virus from spreading is also important. We're starting to see, once again, our hospitals filling up with children who've been exposed through community transmission, and another way of protecting them is this Delta. There's a lot of evidence that the level of protection, whatever it's going to be, is not being met over time with the regimen. There's also a lot of data to suggest that 2 doses without a boost is not really a regimen that's sustain us. I'm in favor of this and probably the broader discussion.
Thank you, Dr. Gans. Dr. Marks, I see you have your hand up. You're muted.
Sorry about that. I just wanted to remind the committee that for emergency use authorization, ideally, this committee will try to be relatively specific about what they would like to see so that we can put into place the correct wording on our authorization. And that has to do with some of the legal liability issues and how that works. And it helps avoid some of the issues that can come up then when CDC, if they were need to change that language. Bottom line is what I'm saying is that some of the deference that we are able to give to the ACIP when we do biologics license application approvals is a little more complicated here. So it's not to say that ACIP will not decide to further manipulate these recommendations, but to the extent that we can try to come to a place that we think will be acceptable for ACIP, that will be appreciated. Over.
Thank you, Dr. Marks. Dr. Chatterjee.
Thanks, Dr. Monto. I'd like to make 3 points. The first is I agree with several of my colleagues with that bullet number 3 on the voting question. I do think that besides the individual risk, which is what we are assessing here obviously, but there is also the societal risk, particularly for healthcare workers, for frontline essential workers who, as Dr. Hilgert pointed out, have individual risks as well. But I think this was part of our discussion a month ago that having a lot of these folks come down with disease, whether it is mild or more severe, is still a problem, because even if they were asymptomatic but they were detected, that would take them out of the workforce, and that certainly is a concern for us as well. The second point I want to make is about the inclusion of racial and ethnic minorities in these studies, and this was a point made by, I believe, one of the open public hearing speakers, that Moderna should look at those populations and their risk and their safety with regard to the booster doses, because there are very, very limited data. There are limited data overall, but particularly in those populations, the data are very, very small. And then the final point I'd like to make is about the Israeli data, and I too am impressed with the work that they're doing. The point I'd like to make is that what they're seeing in Israel isn't necessarily what we're seeing here in the United States. They have shown very compelling data that the booster dose clearly disrupted the third wave of their pandemic. Our numbers are going down before very large proportions of our population have received the booster dose. So I think when we extrapolate data, we have to be very mindful of what the epidemiology is in individual countries and even in local areas.
Thank you. Dr. Kurilla.
Thank you, Arnold. Yeah, just a couple of comments. One is that I certainly recognize the desire for the FDA to put out an EUA for the Moderna boost that essentially mirrors what was done for the Pfizer. And I'm certainly comfortable with that. I think that the same reasons with the waning of immunity, particularly the antibody decay rates that these people are experiencing, places, particularly those populations, especially the elderly and those at the high risk of severe COVID disease, are the ones who are most at risk, and they're relying extensively on their neutralizing titers to really prevent infections. They have much more limited capacity to prevent the severe disease complications. That being said, I have some degree of reservation about the Moderna booster, the 50 microgram, because as was demonstrated by Dr. Miller, even in the absence of neutralizing titer, they are still manifesting more than 50% protection, which means there's things other than neutralizing titer that are doing something. And I don't know if the FDA has any sense of how that will change going from 100 to 50. So that is a little bit of an unknown, and that may actually have a tremendous impact on the durability. The other thing I would say, both with regards to the mRNA vaccines, is that the durability of both of these has been adequately demonstrated in terms of very limited durability, anywhere from 4 to 6 months or 6 to 8 months. And whether that is a consequence of a suboptimal dosing interval, whether that is a dose of the vaccine itself, or whether that is a fundamental inherent issue with the mRNA platform, I think is unknown, and it's going to be very critical to understand whether or not a 6-month boost actually does change the trajectory of the antibody response and provide some better durability than simply anywhere from about 4 to 8 months of antibody responses, and that's all we tend to see. So I think it's going to be very critical going forward to be monitoring this ever so closely, because I'm not convinced that we have actually identified the optimal primary vaccination regimen for these vaccines. Thank you.
Thank you, Dr. Kurilla. Dr. Moore.
Yeah, so there's one point that I'd like to make, and that's that the beauty of the mRNA vaccines, obviously, is because you can change the base pairs as you make the vaccine. So you could, in theory, with making a new 50 milligram, which there's no formulation right now ready for public distribution. Presumably, at least theoretically, I haven't done it, obviously, but theoretically you could change the sequence. So the real question that I have is to Doctors Marks and Fink, is that approving this EUA, does that give you more flexibility administratively to be able to request or demand that booster doses are addressing the variant of concern? That's one thing. Two, I don't quite understand why this is not Delta, because that's what we're facing right now. And three, we've got to remember the Israeli data is really, really quite clear. I was unconvinced by the data, including the early and late vaccination. I can talk about that more, but I don't want to waste the committee's time. I'll talk individually about that, why that's not convincing to me. But the Pfizer data is quite convincing in Israel, but they're different vaccines, since there's, as Dr. Perlman reminded me, there's about 3 times as much mRNA in the Moderna vaccine as there is in the Pfizer vaccine. So the question is to Dr. Fink and Dr. Marks, approving this EUA, does this somehow give you value added in terms of the public health response to be able to quickly respond to variants of concern with a booster?
Dr. Fink, Dr. Marks, Dr. Marks.
Yeah. So thank you. So I think we have in our guidance for emergency use authorization, Appendix 2 discusses how we would deal with variants of concern. Additionally, the World Health Organization is now convening on how to try to decide globally how we'll deal with variants of concern. I think that I would make the decision on this based on what you think the benefit to the patient would be, and not to our ability to move forward with further variants, because I think we do have a reasonable procedure in place for moving through to variants, and some of the sponsors, in fact, I think all the ones I can think of are working with one or the other of the variants of concern to show that they can make a vaccine that will generate an immune response. Now, I think the other question you asked, which somebody else can chime in if they think I've gotten it wrong, for the reason for going with the prototype vaccine here rather than moving to Delta was that the neutralization with these prototype vaccines against Delta are quite good, and the feeling was not to move to a new vaccine if you could neutralize equally well with the response to this variant. Again, it's less churn and burn on the manufacturing, also less exposure of people to potentially antigens that they may not need to see. But it looks like someone from Moderna might also want to speak up here.
Yes, Dr. Miller.
Yes, so Dr. Marks, you actually do have it right, but I just wanted to add some other historical context to how we got here. So we actually made the decision in February of 2021 to begin manufacturing and study variants of concern, and that was really based on data that we observed with the Beta variant. Actually, some of the data that you saw in one of the slides I presented, where we noted a 6.9-fold decrease in neutralizing antibody titers relative to the Wuhan strain. But it takes some time to swap out the sequence, make GMP manufacture, move forward with clinical trials. The exploratory analysis was actually a phase 1 to then be able to move into phase 2. So the data you're reviewing today really came from the population that we had available at that time to vaccinate, and that was the phase 2 study. So they really are the only population, other than the much smaller cohort in phase 1, that were available to be boosted. The mRNA 1273 vaccine was the only one that we had available for use in clinical trials, and we're pleased to see that there is cross-protection to the other variants. But to the question that's been asked, yes, I mean, I think we need to see what happens in terms of the epidemiology and constantly reflect on what the next steps need to be. But that's why we are investigating variants of concern. And, this submission is really the start of our evaluation. And maybe if you'll indulge me since I have the floor, I'll just say completely agree that we need additional data, completely agree that we need data in more diverse populations. That is why we are continuing to vaccinate individuals from the Cove study who are now further out from their primary vaccination. And Cove, if you'll recall, had a much greater degree of diversity. And the final point I want to make is that for these variant vaccines that we're investigating, we also are boosting subjects from Cove and moving forward employing the same diversity and inclusion initiatives that we did in the phase 3 study. So thank you for the opportunity to comment.
And Dr. Marks, to close off this part of the discussion.
Whoops. There we go. Dr. Moore, one other thing, and you might know this already, but the Israel's data were obtained pretty much in the setting of 99% Delta variant over this past summer. So the real-world evidence study there from their boosters is largely from a Delta variant with that was boosted with their prototype vaccine. Over.
Hey, Dr. Perlman.
Dr. Perlman, you there?
Yeah, I'm just turning on it. So I just wanted to make a couple of points. One is I think that it would be great if Moderna actually could do investigations of dosing intervals and mucosal vaccines. That's what we've talked a lot about in the last bit of time. I don't know what they're doing with that, but that's just a small comment. The second thing is in support of the notion of this 18 to 64-year-old vaccination for people who have institutional or occupational exposure. I think another issue that we were thinking about when we approved this for Pfizer was that we can't afford to have healthcare workers, even if not sick, be positive and infected and have to stay home from work because there's parts of the country where there's just a shortage of healthcare workers and there's burnout everywhere. So that was, I think, another part that's not quite in the statement, but I think was in the thinking of some of us anyway. And the other thing was that one thing I have had trouble trying to put together is the Moderna vaccine was actually a little more efficacious than the Pfizer vaccine, yet we're talking about the same 6-month interval. So I'm not sure that that's really it's necessary because the vaccine does seem to be a little more efficacious, but it's hard for me to put that together mathematically to know what the best way to do that. And the final thing was, I think from a pragmatic point of view, even with what I just said, in some ways I support this EUA because we've already approved it for Pfizer, and I don't see how we can possibly not approve it for Moderna and not have most U.S. folks be completely confused. So I know that's not really part of what we're supposed to think about, but I think it's a pragmatic issue. That's all.
Thank you. Dr. Nelson.
Thank you, Dr. Monto. Just a few comments and one technical question regarding this vote. I'm very reassured that it's not a new preparation, actually half a dose of an existing formulation. I think it'll be very reassuring to the public. And two, I agree with our colleagues about the many unknowns regarding the durability of response, and specifically Dr. Kurilla's comments that does the lower dose have an implication for durability after this booster dose. Next, I do remain concerned about the sluggishness with which we are acquiring knowledge about the risk factors for some of these adverse events, systemic adverse events, and communicating with the NIH and sponsors to assist in rapidly identifying these risk factors will make these decisions a lot easier in the future. And finally, very supportive of this EUA intent of making the vaccine available to these very three valued and determined at-risk populations. And with respect to the wording, I'm very happy to see the specific wording of at least 6 months. It allows some discretionary use with respect to the timing of this booster dose, given some of the issues we've discussed today. And then my last comment, or really a question, is a technical one. Before any EUA was authorized last year as part of this committee, we were informed that the data that we were to review to provide that EUA was to be based on individuals who were studied. So I was struck by the lack of underrepresented minorities in the data set of these 300 plus for this specific vaccine. I just wanted confirmation from the FDA that we're allowed to use the bridge data from the initial primary series as part of our deliberations and not have to factor in the absence of these underrepresented minorities. And I appreciate the sponsor's commitment to acquire that data going forward.
Thank you for that question. I of course agree that ideally we would have more diverse representation in all of the data that we have available to evaluate, to make regulatory decisions. That being said, we do have fairly robust data from the primary series that does not suggest significant differences between racial and ethnic groups or genders with regard to vaccine efficacy or vaccine safety. And I do think it's fair, and it is the FDA viewpoint as well, to rely heavily on those observations from the studies with the two-dose series in understanding how a booster dose would be effective and also its safety across diverse populations.
Thank you.
Thank you. Dr. Hawkins.
Thank you very much, and I appreciate all the comments before. So I'm a physician caring for adults that are primarily African American, Hispanics in Los Angeles, California. I've been in practice for 35 years. I believe the results presented today will be encouraging to the many patients who have received available vaccines. They've benefited, look forward to recommended boosters. They look forward to recommended boosters. I also hope the presentation will result in and will be encouraging and instill more trust, including areas of safety and efficacy in hesitant citizens. I still have a substantial number of those. Physicians and medical groups are following CDC vaccination strategies, and overall acceptance has improved. However, challenges still persist, and I think that approval will help us along the way. Thank you.
Thank you. Dr. Rubin. You're muted.
It's your individual phone, Dr. Rubin.
Got it, sir? Dr. Rubin, okay, just unmute your regular phone, sir. Okay, let's go to someone else.
Okay, let's go on to Dr. Hawkins.
I've already spoken.
Oh, okay.
I've already spoken.
Dr. Pergam. Thank you, Dr. Pergam.
Thanks. Thanks, Arnold. I think one thing that everybody's been talking about is this third group. I want to reiterate that I'm very supportive of that third group being part of this. And specifically to Dr. Perlman's comment, the healthcare workers, I think it's critical that we prevent infection as much as we can. And if there is a benefit to that booster in preventing primary infection, then that will be critical at protecting healthcare institutions from outbreaks, et cetera. I also want to comment, just as a side note, that there was some concern that the number of groups here would suggest that a large population of the United States would be eligible for boosters. And one difference between the Israeli data and the United States data so far, at least, has been the uptake of boosters. At least what I've seen that's been published by the CDC so far, only about 10% of those 65 and older have received boosters to date, and only about 4% in the United States have received boosters. So it has not been as some had expected that large numbers would be going to get boosters. And I think one thing that I think will be important is really, if we are going to be making boosters available, is to increase efforts to get these to specific communities at risk.
Thank you, Dr. Pergam. Dr. Meisner.
Thank you, Dr. Monto. And I just wanted to clarify my comments because I'm not sure I was clear. I certainly agree that healthcare workers and institutionalized individuals should be eligible for a booster. My issue was that the statement says their employment or their living situation puts them at high risk of serious complications. And I was just asking, I don't think there are any data to say that, for example, a healthcare worker has a higher risk of serious complications just because of his or her employment. So it's the wording that troubles me, not the intent. I think if it puts them at risk of or increased risk of COVID infection, I think that's fine. The second point is I agree with the comment that people are getting Moderna's booster in a number of different places, and I have a little bit of trouble with saying yes, you can get it if you got the Pfizer the first time for the first primary series, but you can't get it if you got the Moderna for the primary series. I don't think that's really fair. Over.
Thank you. Any comments from FDA about Dr. Meisner's concerns about the wording? The problem is that's the wording we approved last time, correct?
Yes.
In terms of amending.
Dr. Monto, that's correct. I think when you come to your next question, we'd like to give you lots of latitude to make comments on how we could improve that.
Thank you. Dr. Rubin.
Second try working this time?
It is.
Excellent, thank you. I would echo what many people said, and I'm not going to repeat. The data are not perfect, but these are extraordinary times, and we have to work with imperfect data. I just want it to be said once here, and as it was said in the public meeting, that the effect of a booster is much less than the effect of vaccinating unvaccinated individuals, and that means both here and abroad. So I think that we want to clearly send the message or include the message that if we're going to get out of this thing, we have to be vaccinating the unvaccinated.
Thank you. I think that message has been reiterated. Whether they're listening is the problem. Okay, we do not have any more hands raised. So are we ready to call the question? Kathleen?
I believe so. Let me just provide some instruction. Mike, are you back and able to pull up the questions? Okay, great. So thank you, Dr. Monto. We have 19 voting members and 1 non-voting industry representative attending today's meeting.
So only these 19 voting members, excluding the industry representative as seen on this slide, should vote in today's meeting. If you are not an official voting member, please refrain from voting as your vote will not be counted. And in regards to the voting process, Dr. Monto will read the final question aloud for the record, and afterwards all members and temporary voting members will cast their vote by selecting yes, no, or abstain. You'll have 2 minutes to cast your vote, and after the question is read, we will broadcast the results and read the votes aloud for the record. And please note that once you've cast your vote, you may change it within the 2-minute time frame. However, once the poll has closed, all votes are considered final. So unless anyone has any questions related to the voting process, we'll have Dr. Monto read the voting question aloud for the record.
Okay. Do available data. Okay.
Go ahead, Dr. Monto.
Do available data support the safety and effectiveness of Moderna COVID-19 vaccine for use under EUA as a booster dose, 50 micrograms mRNA 1273 at least 6 months after completion of a primary series in the following populations: individuals 65 years of age and older, individuals 18 through 64 years of age and high risk of severe COVID-19, and individuals 18 through 64 years of age whose frequent institutional or occupational exposure to SARS-CoV-2 puts them at high risk of serious complications of COVID-19, including severe COVID-19.
Thank you, Dr. Monto. And Mike, if we could pull up the voting pod. Great. So go ahead and cast your vote if you are an official voting member at this time.
Is the voting pod up?
I don't see it.
It is.
The voting pod is up. It should say voting question 1, yes, no, or abstain. Okay, let me just look at the results here. Okay, I believe that we have all of the results in for all 19 voting members, and I will read them aloud for the record. Dr. Randy Hawkins voted yes. Dr. Cohn voted yes. Dr. Pergam voted yes. Dr. Nelson voted yes. Dr. Moore voted yes. Dr. Fuller voted yes. Dr. Levy voted yes. Dr. Wharton voted yes. Dr. Hildreth voted yes. Dr. Sawyer voted yes. Dr. Carilla voted yes. Dr. Monto voted yes. Dr. Perlman voted yes. Dr. Lee voted yes. Dr. Meisner voted yes. Dr. Gans voted yes. Dr. Offit voted yes. Dr. Chatterjee voted yes. Dr. Rubin voted yes. So we do have a unanimous 19 out of 19 yes votes, and that concludes the voting portion. So we can close this out, and I will hand it back to Dr. Monto. Thank you.
Thank you very much. If anybody wants to explain their vote, raise their hands. What we're going to do after that is we're going to take a merciful 5-minute break before we go on to the discussion topic. So we'll have a few minutes to stretch between any explanation of votes and the discussion topic. Okay, Dr. Moore.
I think that, it's kind of clear that I've got some real issues with this vote, but nonetheless, I just want to explain why I voted yes on it as more gut feeling rather than based on really, truly serious data. And I think that it's very important for companies that are coming to VRPAC on dealing with this EUA that they really take seriously the idea that we need to see good, solid data, and it needs to be explained well, which, to be honest with you, this submission was, to me at least, and perhaps it's just because I'm old and befuddled, but it was not explained well until I read the FDA review, the second half, and that, on the other hand, had a clarity and a crystal precision to it that really made it clear what the issues are. But the data itself is not strong, but it is certainly going in a direction that is supportive of this vote.
Thank you, Dr. Moore. We're going to break until 3:20 Eastern. And then at that point, Mike, you'll put up the discussion question. Break for about 6 or 7 minutes.
All right, so just a short break for 7 minutes. All right, let me put the timer up. All right, welcome back from that quick break. And Dr. Monto, you ready to take us into the discussion topic and wrap and get towards the end of the day?
I am, and remember, this is not a voting topic. As Dr. Marks told us, we have free reign to say whatever we want to. We can be a little less focused than we were during the discussion of the voting questions. And I won't just read this to you because you all can read the PowerPoint. And what we're going to be doing is talking about how comfortable we would be in extending some of these booster recommendations down to age groups down to 18, not including anyone at this point under 18 years of age. This reflects some of the requests that have actually been made to FDA from the manufacturers. So, Dr. Chatterjee's got her hand raised. Dr. Chatterjee.
Thank you. We discussed this a little bit at the last meeting when Pfizer's vaccine was up for discussion. And I think the concern I have, there are a couple of concerns I have. One is that I am not convinced that the epidemiology of the pandemic at the moment in the U.S. supports this request. We are seeing cases going down without booster doses play yet, and in this population, the people who are vaccinated appear to be protected. The disease primarily seems to be occurring especially in its more severe form, in those who are unvaccinated. And the comment was made earlier today that is really the group that we need to focus on getting them vaccinated. So that's the first point I want to make. The second point is with regard to the robustness of the data. The numbers of participants in these booster trials, these booster studies, are very, very small. And we're talking about basing a decision that will impact tens, if not hundreds of millions of people based on data that have been provided by both the companies. If you add them together, they don't come up to 500 people. So I am very concerned about the paucity of data on which this decision will be made.
Thank you, Dr. Chatterjee. Dr. Offit.
Yeah, I'd just like to agree completely with Dr. Chatterjee. I feel like we're sort of going down the line here of booster dosing based largely on data generated from Israel. And although I think the data generated in Israel certainly was clear for the 70 to 79-year-old, I am just less impressed with who I put, frankly, in the same category as an immune-incompetent host. I am less impressed with the data regarding the younger person. There's just too many variables in there that I think may not have been considered, not the least of which, as Dr. Chatterjee said, we're seeing a decline in this country right now, too, and it's certainly not because of booster dosing. I mean, we could claim that. So I do worry about this broad use now of boosters. Certainly, I don't agree with doing this down to 18 years of age at all. I mean, maybe at 30, I would feel a little better because I think the 18 to 29-year-old is at higher risk of myocarditis with any clear evidence of benefit. I'm impressed by the fact that we continue to have excellent protection against moderate to severe disease in this country through Delta and for all age groups. And I just think that we continue to send wrong messages out there by using terms like breakthrough and by making people feel that they're not protected unless they've gotten a third dose. And where as Dr. Rubin said so accurately, the problem in this country is vaccinating the unvaccinated. I can tell you at HUP, the Hospital of the University of Pennsylvania, at CHOP and those over 12, the people who are in the ICU aren't there because they haven't gotten a third dose. They're there because they haven't gotten any dose. And I just worry that we haven't clearly defined what the goal of this vaccine is. Because if the goal of this vaccine is to prevent asymptomatic or mildly symptomatic infection, that is a goal for which we have set no other vaccine. I mean, if we're trying to prevent what is inevitable, which is a decline in neutralizing antibodies and an erosion in protection against mild or asymptomatic infection, that is a high bar to which we hold no other vaccine. And I understand we're in a pandemic. I understand that may mean somewhat less
shedding, but I think if you really want to control shedding, we just have to vaccinate the unvaccinated. So I'm uncomfortable with how we sort of trip down the line here regarding sort of now the thought of universal booster dosing, which I just think is wrong. Thank you.
Thank you. Dr. Rubin.
Thank you. Am I on?
You are.
Oh, thanks. Sometimes it talks to me and sometimes it doesn't. So I agree entirely with Dr. Offit. And I guess I phrase it slightly differently, which is, and Dr. Chatterjee, but I think that I phrase it slightly differently, which is that in order to demonstrate we should be giving vaccine to much younger patients who are not otherwise at risk, we need to have some sort of risk-benefit analysis done. And that risk-benefit analysis could include the fact that the vaccine inhibits transmission and therefore can break the cycle of transmission. That would be at least one factor to consider. But we don't have that. We don't really have a good idea of the benefit of boosters for this group, and there's a good reason to think that there isn't much benefit. And we know that there's some safety signal, and I'm not sure that we want to just explore it willy-nilly by giving it to a lot of people.
Thank you, Dr. Gans.
I want to thank my colleagues for bringing forward some really great thoughts about it. I would argue that I don't think that we have to do talking about not facts.
Dr. Gans, you're breaking up.
Dr. Gans, we're not hearing you right now. Yeah, Dr. Gans, we're not hearing you. So let's go to somebody else. I think our headset unplugged.
Dr. Carilla.
Yeah, thank you, Arnold. Yeah, I agree with my colleagues. As I've expressed previously, I think that in my mind, the need for the boosters is primarily in those individuals who are at high risk for serious disease, which overlaps pretty well with individuals who don't respond very well with adequate cellular immune responses, which I think is most important for protecting against severe disease. For the younger population, they seem to be responding not only quite well to these vaccines, but they're actually holding up. So I don't necessarily see the need for a sort of let it rip campaign for boosters for everyone who's ever been vaccinated. I'll respectfully disagree with several of my colleagues. So I was not as impressed with the Israeli data as a justification. They may be attributing their profile of their third wave to the introduction of boosters, but I think if you look at their first and second waves, which was pre-vaccines, it qualitatively looks very similar. And in fact, if you look at the Delta wave that went through India, which has less than 20% fully vaccinated people, and it's very similar to what we're seeing here. The Delta wave seems to have entered into a population, and it goes through, and then it moves on, and it's just been a wave moving throughout the country. So I don't think that the boosters really should be I guess the question I'm really getting at is, what do we want the boosters to do? As Dr. Offit was saying, if the intention here is to actually have an impact on the transmission with some sort of, aspirational sterilizing immunity type of function, I don't think these vaccines are really demonstrating that. What they are very good at is preventing severe disease, and I think that if we can actually migrate the pandemic down from being a very severe case to situation to something that is more akin to influenza, I think that the vaccines will have done what we really need for them to do, which is to prevent the overwhelming of the healthcare system and to protect the people who are most at risk of serious disease. The younger populations don't seem to have as much of a problem, and I'm not as really worried, even if they, are not boosted, from the standpoint of the other factor we're not paying attention to is as this pandemic evolves, we are looking currently as if people are vaccinated or unvaccinated, but there's also people who have been infected, and no one has really talked about whether breakthrough infections, I know that some people don't like that term, but having been vaccinated and then having experienced an infection because of waning immunity, what sort of immunological responses does that manifest, and is that the equivalent of being boosted? Those are questions I think that are going to become more critical, because eventually everyone is either going to have been vaccinated or have been priorly infected or both, and really understanding what their immunological status is across the age spectrum and across the healthcare spectrum, I think, is going to be very important. We can't just look at this as boost people every 6 months. It's not going to work.
Dr. Meisner.
Thank you, Dr. Monto. I completely concur with everything that's been stated up to this point in terms of younger adolescents and children. If we look at the CDC hospitalization rate for COVID-19 associated hospitalizations in children under 18 years, it's less than 1 per 100,000. And the rates of myocarditis are variable depending on the study, but probably at least 5 to 10 per 100,000. So before we recommend a vaccine for young children and adolescents, I think we really need to know exactly what Dr. Rubin said. What is the risk benefit ratio? And I think giving a booster without a large number of participants and subjects, I think may not be the best thing to do. Thank you.
Dr. Levy.
Thank you. I think there are 4 elements here I'd want to know more about before a decision on recommending boosters all the way down to 18 years of age. We've talked a lot about risks to young individuals, particularly young males, vis-a-vis myocarditis in relation to the risk of COVID symptoms. What we haven't said too much about is if a vaccine helps reduce transmission of coronavirus from a young individual to their parents or grandparents, there are both indirect and maybe direct benefits to that individual as well. So that calculus gets more complicated and should be considered and analyzed. Is it possible that boosters in the right context could help us get to herd immunity? Several of the other committee members brought that up. The Israeli data spoke to that possibility. That's intriguing. Another unknown in my mind is solid studies about long COVID in children. Does it exist? What is it like, and how frequent is it? Do we have phenomena where children initially don't have many symptoms, but then there are longer-term effects? To my knowledge, the literature is still muddled on this, and there's a lack of rigorous studies, and we would look forward to information from CDC and FDA for their national analysis on that. Finally, we're asked to consider these questions without regard often to whether recommending something would become making it available to a particular age group versus it's turning into a mandate. And that's not really the purview of our committee, because that goes to CDC, and then states in our federal system implement their approach to all of this. But nevertheless, it would impact my view of it in terms of the public health impact. So those are 4 areas I think should be considered and explicitly analyzed and discussed ahead of any such vote by this committee. Thank you.
Thank you, Dr. Gaines.
Apologies for being booted off last time. So I don't know how much you got, but I really agree and want to thank my colleagues for this discussion. And as the question is stated, I mean, it's really asking if we have the current data, and I think we need more. But I would add to the amount of data that we need, because I think it's very important to get this question right. So, you know, the fact that other vaccines are used, we don't call it a boost, just say series. So we really have to get right, what is the series for this? And so we really have to understand these breakthroughs and really understand the disease long-term ramifications. So we need immunologic data on these breakthroughs that we keep hearing we're going to get, and we've been actually hearing that for quite some time. So it sounded like there were some preprint information. We need that to move forward. So we need both the information not only around humoral immunity. Everyone has brought up that we have to understand what actually is your cell immunity. So we really need to appeal to our colleagues looking at this to really understand that. The other piece of information that I think is going to be really important, again, as the Delta variant is actually causing a different distribution as well as different severity of disease, and we need to understand, we're not going to have long-term data, but we need to understand the indications, even if you have mild disease, whatever that is, what does that actually do? Short-term is long get, because allowing people to get infected because we can't achieve sterilization is different than affording them the ability not to have damaged tissues from infection as mild as it is. So I think we need several points that we need that we're all asking for, battling with, so that we can make sure that we understand this. But I think it's very important for us not to ignore signals that are out there. It's true that Delta is dropping, but it's also true that there's a different disease form, and we are seeing people hospitalized who don't necessarily meet the risk factors that we understood the original. I think it's very important not to ignore signals early so that we can cause prevention, and that's what this question is asking. Did we hit it right first and understand it? That's what I think we need, but I also would say, and I don't know if this was something that I said before and was heard, but this question does not need to be answered in an or question. We can immunize people who are not vaccinated and still. And then we need to also consider the protection of the very youngest people in our society who.
Thank you, Dr. Gans. And as we go forward in our discussion, I think we should not think about this as one enormous population group down to age 18. The risk-benefit may vary in some of the older, still young, but let's say down to age 40, as compared to the 40 to 18-year-olds. We are seeing breakthrough, to use an unwelcome term, we are seeing infections with hospitalization in those age groups, and we will be getting data as the boosters are rolled out in the older population. So let's keep that in mind and not look at this as a single question, but perhaps a question that can be broken into stages. So going further to Dr. Cohen.
Thanks, Dr. Monto. That is actually one of the points I was going to bring up as well. And I'd really like to bring up the age group of 50 and older. One of the topics that came up during the ACIP meeting where this was discussed is that 65 is really a construct for being older or not, and given the incredible impact that COVID has had on many older communities of color, it's even especially important that we protect older persons of color who may not actually meet that 65 age cutoff. And so I would like to consider at least moving down to age 50, where the risk for myocarditis after 1 dose and 2 doses and in the 3rd dose from Israel is back to baseline.
Okay, Dr. Hildreth.
Okay. Thank you, Dr. Monto. I want to reference a point made by Dr. Gans, and I said this last time. What would be really helpful would be to have some objective measure to know when boosters are needed. An immune correlate. It could be a certain neutralizing antibody titer or certain T-cell response. That way we could know when boosters are needed regardless of the risk factors, because after all, the first problem to be solved is keeping people protected from infection. And to know when the antibody levels are high enough to protect them would be very helpful. So I don't understand how after hundreds of millions of people infected and almost 1,000 trials that we don't have that information yet. So I think an immune correlate would be really helpful in all of this. So thank you.
Dr. Moore. J&J adenovirus vaccine, whereas something like 1 out of 50,000 to
your microphone has been turned on.
Young men will be affected by, apparently, by the mRNA or the RNA vaccines. Hence, there's one thing that one way to approach that, of course, is to restrict use of the, or at least suggest restricting use of each class of vaccine to those that have the highest risk of severe adverse effects from them.
Thank you. We don't have any hands raised. Dr. Marks, would you like to make some comments before I try to summarize the discussion?
No, thanks very much to the committee members. I think we heard pretty loud and clearly that there was not a lot of appetite for moving down the age range very significantly, if at all. And I think we'll go back and try to understand what might make the most sense, if anything, based on your feedback. And if anyone wants to chime in on anything else in that regard, we're happy. But I think to hear that. But I think I think if that's the summary that we would take from this. And we do hear very loud and clear this need for benefit risk considerations here. It is a very challenging pandemic. Having been doing this now for about 2 years, the problem here is that we don't know what we don't know, and making any predictions about what's going to happen in the next months is very challenging. There are models that predict that we could potentially have another wave of COVID-19 as people go inside this winter, and we have either the current variants or one other one come up. So that is part of what is going into our minds here about being prepared. I think we can't simply look right now at what's going on with the pandemic curve and just call it a day. We have to be able to think about what might happen, and I would encourage people to look at any one of, there are several very good modeling groups, academic as well as from the CDC, which are concerned that we could see another wave. So that's part of what's going into our thinking here, is that we do have to think ahead, but very, very grateful for the committee's, I think it seems pretty uniform feedback here.
Dr. Pergam, I see that you have your hand raised. I may have missed it.
That's okay, Dr. Monto. This is more just a question of sort of how the process works, and maybe this is for Dr. Marks. Currently, the FDA guidance is that it's these particular groups that would be eligible. If the ACIP decided to change the age range, would that be a decision that they would make independently, or do they need our group to vote to make those changes first to allow them to drop to those lower levels? Are they only allowed to vote on sort of what we've approved from this committee? I just wanted clarification on that.
I'm going to actually defer part of this to Dr. Cohen, because it's nice to have her on the line to be able to But in general, the idea here is that ACIP for these emergency use authorizations could potentially, there are a lot of options. They could potentially narrow. There's another vehicle they could use called emergency use instructions, which could work differently. But ideally, they would have, what we have would be something that would be broad, and they would potentially narrow or refine further. But Dr. Cohen, do you want to try to refine what I've said a little bit?
Sure. I'll just confirm that ACIP, under the constrictions of the EUA, unlike a BLA, ACIP really can't expand or be broader than FDA conditions of use. However, we can be more narrow. So, for example, FDA could go down on age and ACIP would not have to. But if FDA does not change and go down on age, ACIP could not address it.
Thank you. I think that's pretty clear. And what I would suggest, Dr. Marks, as we go forward, and I'm not looking for more meetings. These are quite tiring and time-consuming for all of us. But I think we need to develop some rationale for going down in age groups as we gain experience with these, with the booster doses in an older and other populations, populations at high risk, which will include younger individuals. I think part of the problem is basically one of risk-benefit, and I don't know that the benefit has been sufficiently defined. So as we go down in age and gain experience in terms of the risk and to a lesser extent the benefit, because we may not see that if in fact the wave that we're currently getting out of does not return, then we can revisit the topic and try to refine it in terms of different age groups and what might happen in the older of the young and the younger of the young, not going below 18 years of age. So I think that would be my summary. The concern that I have is that we don't want to wait until we see real more severe infections in the under 65-year-old general population, because getting this vaccine out takes time and requires extreme logistic efforts. So that's my summary. At this point, thank you all. Thank you to the staff of FDA. Thanks to members of the committee. And I'll turn this over to Prabha for the official closing. Until tomorrow, that is.
Thank you, Dr. Monto. Thank you, everyone, all the members and consultants and the meeting participants and speakers. Thank you for a very productive meeting. And then we are actually closing earlier than anticipated. So we will be ready for fresh and nice for tomorrow morning and another topic. So thank you, and the meeting is adjourned now at 3:50 p.m. Eastern Time. Thank you.
Thanks very much.